पढ़ रहे हो कि नहीं पढ़ रहे हो?
वैसे नोट को स्टार्ट कर
रिसर्च करते हैं
मतलब वो डायरेक्टली एक ही नोट को इम्पल्स भेजो
इम्पल्स एक नोट का मतलब नहीं जो उसके ऐसे नोट डिवाइड कर लिया
तो एक ही नोट को ऐसे नोट भेजो
So what is happening?
Drainage is nothing but छोटा सा दिवाली धमाका okay और blood vessels में capillaries start rupturing due to tremendous pressure यहाँ पे छोटे पाइप है ना प्रेशर बहुत ज्यादा आ गया तो क्या होगा बस्ट होगा जब वो इंटरनली होते हैं उसको बोलते हैं इंटरनल ब्लड अगर बॉडी में हुआ एक्सीडेंटल मौत होता है ठीक है मतलब कैन बी टेकन केयर डाउटफुल तो बहुत कुछ फर्क पड़ेगा
So breakdown is clotting of blood.
इमेजिन में ब्लड क्लॉटिंग होगा ही होगा कि पर क्लॉट फॉर्म इन द ब्लड वेसल्स इज नॉट गुड ऑब्वियसली इट वुड लीड टू डेथ चला गो अहेड आगे बढ़ू ओके so now I want you to open there is a system given lymphatic system मिला ओके सर डायग्राम जो साइड में दिया हुआ है that diagram in the volume is only a representation diagram picked up from ICSE book for you to understand okay technically थोड़ा बहुत अजीब हो जाता है डायग्राम को समझना पर it is a diagram which need to be understood second thing चलन ये बहुत ज्यादा ना थियरी पार्ट है इसमें जैसे हार्ट में डायग्राम होता है एक्सप्रेशन बहुत कुछ है इसमें थोड़ा समझना आपको खुद को इमेजिन करना होता है ओके सो व्हाट आई वुड लाइक टू डू इज पहले अपन ना ये पूरा एक एक लाइन पढ़ते हैं so that I explain you line by and then I explain you once again the end so I want somebody to read loud धीरे धीरे पढ़ लेते हैं
lymphatic system as the blood flows in the capillaries of the tissues the plasma and the leukocytes leak out through their walls some of this fluid may be reabsorbed into the blood vessels but most of it enters another set of minute channels named lymph vessels where it is called
okay now then ध्यान देना a thick vessel known as artery. Artery divides into small tubes known as arterioles. Arterioles further divided to form capillaries. Capillaries unite to form vein. venous. Diagram, I think it's very clear. Now, what type of blood flows over here? Oxygenated blood flows in this. What is the pressure of the blood in the arteries? High pressure or low pressure? The blood is flowing at a high pressure. What about arterioles? Pressure decreases. What about capillaries? Low pressure. Sure. Why?
Because in the arterial it was already decreased.
In the no. Why do you decrease in the artery? Why do you decrease further in the capillary?
So because it is divided into
Good. So pressure divides. The pressure in the capillaries is very high. Reason? Pressure divide हो रहा है, but simultaneously narrowing भी तो हो रहा है. Narrow the tube, greater is the pressure. So capillaries में what is the pressure? Very high. इतना समझ में आया? अब मैं सिर्फ इस पार्ट को मैग्नीफाई करूं, एक कैपिलरी. So this is the capillary. What did we say about capillaries? Capillaries are single epithelial cells. एक single layer होता है. ये epithelial cells ऐसे हैं. Now, the single layer cells. In capillaries के बीच में there are in the between the cells there are small pores present. मेरे जो black dot बना है वो क्या है? The cell meets उनके बीच में एक gap है, and that gap is known as capillary pores. Now, blood is coming from here, which is at a high pressure. It comes over here. What will happen? It comes out the cap. It's simple ना. अपना एक पाइप है, पाइप के मैंने होल बना दिए, तो पानी डाला, तो होल से पानी बाहर आएगा. What is blood made up of? Plasma and cells. Okay? तो अब सबसे पहला क्या बाहर निकलेगा? प्लाज्मा, बिकॉज़ वो लिक्विड कंपोनेंट है. सबसे पहले तो प्लाज्मा बाहर आएगा. अब सेलुलर कंपोनेंट में पहला सेल कौन सा था? एरिथ्रोसाइट्स, आरबीसीज. साइज बहुत बड़ी है और की साइज बहुत छोटी है. कैन दे कम आउट? नो. सो आरबीसीज विल रिमेन in the capillaries. फिर platelets. Platelet size again बड़ा है compared to the whole size. Can they come out? No. So platelets भी inside. What about WBCs? Good. They are largest among all the two. सबसे बड़ा WBC size. साइज में सबसे बड़ा WBC होता है, but they can come out because they are irregular. This can squeeze and move out of the. So यहाँ पे leukocytes. Is it clear? Now, when this plasma and leukocytes comes out, surrounding the capillaries, okay, then this is referred as capillaries के बाजू में जो है उसका बोलते हैं टिश्यू. अब इसमें से कुछ वापस ये ब्लड वेसल्स में एब्जॉर्ब हो जाता है, थोड़ा बहुत वापस एंटर्स इन, बट मेजॉरिटी ऑफ इट सिंपली मूव्स फर्दर. जहां जगह मिलेगी वहां पे वो आगे बढ़ते रहेंगे. सो दिस इज द फ्लूइड ओवर हियर. ये कैपिलरी है अपना ब्लैक बोर्ड. कैपिलरी से फिल्टर हुआ, यहाँ पे प्लाज्मा और WBC आ रहा है. जगह कहाँ पे मिली? यहाँ पे मिला, यहाँ पे मिला, यहाँ पे मिला. So when they start moving, they start moving and they enter into another set of tubes. यहाँ से they enter into another set of tubes. Then, when this fluid enters into another set of tubes, then it is referred as, यहाँ पे वो fluid आपको इसको बोला जाता है lymph, and the set of tubes is known as lymph vessels. So what is lymph? Lymph is nothing but a fluid derived from blood itself. Blood में से plasma and leukocytes is what we say as lymph. इतना समझ में आया? Everybody. Now, हमने blood circulatory system पढ़ा था. What are three main parts of blood circulatory system? Blood vessels और heart. अच्छा, lymph बना, filter हुआ, tissue fluid is also known as lymph only. अब ये lymph enters into another set of tubes. What is the tube known as? Lymph vessels. Now, lymph always flow towards the heart. तो हमेशा heart की तरफ ही move करेगा. So when it is moving towards the heart, in between it moves across lymph nodes. तो हम इसमें पढ़ेंगे lymph vessels, lymph nodes. Okay. Now what are lymph nodes? So आपके डायग्राम में ऐसे दिया देखो यहाँ पे. Now what exactly are lymph nodes? The lymph node कुछ नहीं है, but simple language में we can say एक junction. Okay. यहाँ से पाइप आया, कहाँ पे जा रहा है? ऊपर की तरफ, हार्ट की तरफ. बॉडी में सिर्फ एक ही पाइप होगा lymph vessel का. यहाँ से जाएगा, यहाँ से जाएगा, यहाँ से आएगा, यहाँ से आएगा. So from various lymph vessels open up into a big space. What is that big space known as? Lymph nodes, from which a fresh lymph vessel arises, moving towards the. Okay, so it's like अपन वो ये जो point है junction, वहाँ से उड़ाया, सामने से उड़ाया, यहाँ से उड़ाया, ये कहाँ पे जा रहा है? Towards wash. Okay, so this is what we say as lymph nodes. At any given point, where will be maximum lymph present? Lymph. Is it clear, everybody?
अगर lymph हार्ट की तरफ अगर नीचे से ऐसा है, उसको तो कुछ पंप नहीं कर रहे.
Good. Question is right. Okay. सर lymph हुआ मतलब blood की तरह. ये सब lymph blood vessels हुआ. अब वो नीचे से ऊपर की तरफ आ रहा है, तो पंपिंग ऑर्गन चाहिए. व्हाट इज द पंपिंग ऑर्गन इन आवर केस? हार्ट, ब्लड पंप कर रहा है. सो लिम्फ में पंपिंग ऑर्गन नहीं है, लिम्फ हार्ट नहीं है. सो व्हाट अलाउज इट टू पंप फॉरवर्ड इज थ्राइसेड का ये स्पेशलाइज्ड वेसल्स, नॉट ब्लड वेसल्स. या अपने चैप्टर के स्टार्टिंग में पढ़ा था कि ओपन सर्कुलेटरी, क्लोज सर्कुलेटरी. ये कौन सा एग्जांपल है? ओपन सर्कुलेटरी. अच्छा, ये मेरे बॉडी के सेल है, एक, दूसरा, तीसरा. There are 3 cells which are formed over here. ऐसे. चौथा सेल मान लो इसके नीचे है ऐसे. बीच में क्या क्रिएट हुआ? क्या? चल, lymph vessels नहीं है. ये बॉडी के सेल्स की अरेंजमेंट इन सच अ वे है कि उसके वेसल इज अ गैप क्रिएटेड इन बिटवीन, एंड दैट एक्ट्स लाइक अ lymph vessels which are open ended tubes. मतलब ये यहाँ पे ओपन होते हैं. It could be open at any point. Okay, so वहाँ से lymph enter हुआ, बाहर निकल भी सकता है. But because वो जगह ऐसी बनी है, बॉडी से ऐसे अरेंज हो चुके हैं कि उसके वजह से एक गैप क्रिएट हुआ है which is acting like a tube-like structure. That is why we say it as a lymph vessels. Now, what are these body cells? Body cells could be any cells of your body. उसपे muscle cells भी होते हैं. And what is the muscle cell property? Contract and relax. So lymph vessels में बाजू में muscle cells होते हैं. The contraction of the muscle cells pushes the lymph upwards. So वहाँ पे organ नहीं है. वहाँ पे the nearby muscle cells which constitute the lymph vessels help in pushing the lymph upwards. Clear with this? Okay. Can you continue the next point?
Lymph is a colorless or slightly yellowish viscous fluid which is derived from the tissue fluid and is present inside a special tube called lymph vessels.
Learn exam में ये definition की लिखी थी. What is lymph or define lymph? तो ये क्या लिखोगे? Lymph is a colorless or slightly yellowish viscous fluid which flows into specialized tubes known as
Lymph
vessels. Yeah, you will write in your own words. Lymph is a colorless or yellowish color fluid which is made up of plasma and leukocyte which flows into a specialized tube known as lymph vessels. Am I clear? Viscous is denser. Okay, so comparatively less viscous. Next.
vessels on the way drain lymph into lymph nodes from where fresh lymph channels arise and ultimately pour the lymph into the major anterior veins.
Okay. So lymph vessels ultimately pour it into the nodes from where the fresh lymph vessel arises, which ultimately pours into anterior veins. Now children, diagram में अगर आप ध्यान दोगे आपके बुक के ऐसे दिया हुआ है और फिर यहाँ पे हार्ट दिया हुआ है और हार्ट के ब्लड वेसल पे जाके वो कनेक्ट कर. So lymph vessel heart में open नहीं होता. Lymph vessel as it move towards the heart, it will ultimately join back into either of the vena cavas. चाहे वो superior vena cava हो, चाहे वो inferior vena. Reason simple है. ये आपका हार्ट का ये आपका मान लो ब्लड वेसल का पता है. आयोटा से घूमते हुए फिर ये कैपिलरीज और फिर वेन बैक टू द हार्ट. ओके, अब चिल्ड्रन यहां से 5 लीटर्स ऑफ ब्लड बाहर निकला. ये कैपिलरी पार्ट है. मान लो कैपिलरी में क्या होगा? द ब्लड फिल्टर्स आउट टू फॉर्म इन शॉर्ट क्या बनेगा? लिम्फ. अपन इसको ग्रीन रेड कर दिखा रहे हैं, येलो करो तो लिम्फ. यहाँ पे जब बाहर निकला 2 लीटर बाहर निकला. हाँ, यहाँ पे कितना ब्लड होगा? 3 लीटर. एक मोमेंट के लिए मानना. फिर 3 लीटर गया, यहाँ पे आया, फिर 2 कम हुआ, 1 -2 में आएगा. नो ना. जो बाहर निकला, ओके, अब यहां से फिर घूम फिर के कहां पे आया? यहाँ पे. तो यहाँ पे माइनस हुआ, यहाँ पे प्लस हुआ. व्हाट इज द नेट गेन? Same remains. That is why it is very important. The lymph ultimately goes back and joins the vein caves so that the overall blood volume remains same. Am I clear? Everybody? Okay. That is what it is in that point. Next point.
Lymph flows unidirectional. That is from tissues to heart.
मैं कहा था the lymph always flows in direction. किसी भी बॉडी पार्ट से वो हार्ट की तरफ ही जाएगा. लोअर towards the heart, अपर towards the heart. अपर में बहुत कम होते हैं lymph vessels, लोअर में अलग. अपर पार्ट में ब्रेन में there is another fluid CSF which is fucking like a lymph. तो वहाँ पे उसका रिप्लेसमेंट कर देगा. Next.
The lymph flows in these vessels due to contraction of the surrounding vessels.
Already explained. Surrounding, what are the lymph vessels basically? The body cells itself. So surrounding muscle cells है, Because of its contraction, the lymph flows upwards towards the hard part.
It is also called extracellular fluid as it makes the cells and tides outside.
Okay. अब चलन अलग अलग बुक पे अलग अलग. अगर मैं इसको मैग्नीफाई करूं तो ये क्या है? इसके बाजू में सेल्स होंगे, बॉडी सेल्स, और ये बॉडी सेल्स के बीच में क्या होगा? लिम्फ, जो सेल के बाजू में. Because it is present between the cells, it is known as intercellular flow. Because it is present outside the cell, it is also referred as extracellular flow. Okay? इंटर दो सेल के बीच में, दो सेल के बीच में है, सेल के बाहर है, सेल के अंदर भी फ्लूइड होता है, उसको क्या बोलते हैं? सेल के बाहर एक फ्लूइड है, वो क्या है? लिम्फ. अब जरूरी कौन सा है? साइटोप्लाज्म. लिम्फ जरूरी है? नहीं. इसके उसको एक्स्ट्रा सेलुलर फ्लूइड which baths all the cells of your body. हर एक सेल आपकी लिम्फ के आजू बाजू में होगा. ठीक है? Next.
Composition of lymph. Lymph does not possess any
Composition made up of plasma and leukocytes. Leukocyte में further analyze किया. बढ़ो.
lymph does not possess erythrocyte and
Erythrocytes,
erythrocytes and blood platelets, but consists of leukocytes.
Okay, leukocyte लिखा है क्या? Leukocytes लिखा है. Leukocytes. Okay.
Water is 94% in solids, which is proteins, fats, carbohydrates, enzymes, antibodies, etc. is 6%.
I think clear with it. कोई बताने की जरूरत है? नहीं. आगे.
Function of lymph.
Okay. चल. अब वहाँ पे जो composition लिखा है 94% water, 6% solids. Okay. 6% में उन्होंने क्या है? Cells, antibodies, etc. चल. WBCs में majority of the cells are another type of WBC known as lymphocytes. WBC are further divided into 5 categories. ये 5 कैटेगरी में एक जो कैटेगरी है lymphocytes, सबसे ज्यादा लिम्फ में वही होते हैं. लिम्फ, लिम्फो, समझ में आया ना? वर्ड. So lymphocytes. Reason? It is this lymphocytes which are responsible for antibody production. तो आपका जितना भी अपन ने पढ़ा, WBCs are referred as soldiers of the bodies. Okay? WBCs में जो बाकी 4 टाइप होते हैं ना, वो अपने हवलदार, वॉचमैन, उस कैटेगरी. What are lymphocytes? ब्लैक कमांडो. छोटे मोटे चीज के लिए ब्लैक कमांडो आते क्या? कि कल तेरे घर पे चोरी आए, पुलिस को बुलाएगा कि कमांडो को आएंगे, सर चोरी हुई है, तेरे यहाँ पे मारा मार चल रहा है, तो ब्लैक कमांडो जाएंगे सॉल्व करने के लिए. There is a major issue, terrorist attack. तब ब्लैक कमांडोज ही आएंगे ना. एनएसजी, नेशनल सिक्योरिटी गार्ड. यहाँ पे वही चीज है. छोटे मोटे चीजों के लिए डब्ल्यूबीसी के टाइप है. बड़ी चीज फॉर एंटीबॉडी प्रोडक्शन, देन लिम्फोसाइट्स कम्स इनटू पिक्चर. सो वो हमेशा लिम्फेटिक सिस्टम में जाना है. Am I clear? Next is function.
Functions of
Don't read it. Then I will explain. अच्छा ना, फंक्शंस पे ध्यान दोगे. 4 फंक्शन दिए हुए: न्यूट्रिटिव, एब्जॉर्प्टिव, ड्रेनेज एंड डिफेंस. अब अपन एक एक करके पढ़ेंगे. चल, बाय डिफॉल्ट फंक्शन सबसे लास्ट वाला है. बाय डिफेंस, व्हाट इज दिस? The plasma plus leukocytes, and leukocytes are soldiers of our body, so they by default provide immunity to the, they are parallel immune system to our blood circulatory system. So that is difference. Do I need to explain that? No. Second, nutritive. ट्रेन से जाते हो? आया ना? गुड. कहाँ पे गए हो? CST उधर आप गए होगे ना. अब मान लो CST से आ रहे हो, कहाँ पे कट डाउन करोगे? वाशी से फिर तो ऊपर करना, ऊपर करने से फिर चल के या रिक्शा से, इज द ट्रेन रीचिंग टू योर हाउस? ऐसा बेडरूम में आता है वो? आई डोंट थिंक सो। क्या रिक्शा से भी आज रिक्शा वाला बोलता है तो बेडरूम में लेके आ जाता है, सबका बेड है ना? इट कैन ओनली गो टू अप टू सर्टेन पॉइंट, और फिर वहां से आपको वॉक डाउन जो भी है। ब्लड वेसल्स जो होते हैं कैन नॉट गो टू ईच एंड एवरी कॉर्नर ऑफ द वर्ल्ड। जैसे कि यहाँ पे ब्लड वेसल ड्रॉ किया, इज दिस ब्लड वेसल रीचिंग टू दिस सेल? नो। सो व्हाट इज रीचिंग टू दिस सेल? लिम्फ। इसके बाजू में क्या है? हर एक सेल के बाजू में क्या है? लिम्फ। Whatever nutrients the blood vessel brings, that would be diffused into the lymph. Because what is lymph? Plasma plus substances dissolving in also. So plasma में dissolved हुआ substances will then enter into the cell. So it provides nourishment to each and every cell of the body where blood vessels can't reach. इतना समझ में आया? तीसरा फंक्शन पहला फंक्शन अगर समझ में आ गया होगा तो तीसरा फंक्शन उससे से रिलेटेड है, उल्टा करना है। सेल भी बहुत कुछ बनाता होगा, वेस्ट प्रोडक्ट्स है, एंजाइम्स है, बहुत कुछ बनाता है। इज दैट सेल डायरेक्टली गिवन टू ब्लड? ना, ब्लड से तो नहीं आ रही ना। वो क्या करेगा? It will pour it into the lymph. Lymph क्या करेगा? It is moving towards the heart. So as it is moving towards that, it drains back the extra fluid into the circulation, along with waste and all this. What do you mean by extra fluid into the circulation? Children, imagine करना है आपकी बॉडी की हर एक सेल में which process is going on? What is the end product of respiration? Carbon dioxide, water, 38, 80. अपने बॉडी में कितने सेल हैं? अपन बोलते चलो बिलियन बिलियन बिलियन। हर एक सेल में रेस्पिरेशन हो रहा है, हर एक सेल में वाटर बन रहा है। अपन एक कैलकुलेशन करने के लिए बोलते हैं हर एक सेल में 1 ml वाटर बन, 1 ml into बिलियन 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 कितना होगा? एटलीस्ट कम से कम 100, 200, 400, 500 लीटर तो वही चलेगा। उतना पानी अगर अपने बॉडी में होता तो अपन कैसे होते? ऐसे होते या पंपकिन की तरह गुब्बारे का पानी, एक बड़ा सा गुब्बारा, इसमें पानी होता आया ना? वैसे है नो। whatever cells extra fluid जो सेल वाटर बना रहा है, that is drained into the lymph, and that extra fluid along with the waste proteins which are produced by the cells, all that would be drained back into circulation. फिर वो ब्लड वेसल में जाएगा, ब्लड वेसल से हार्ट में जाएगा, हार्ट फिर उसको डिपेंड करेगा किडनी से बाहर निकालना है या किधर से बाहर निकालना। सो चाहिए उतना रखेगा, नहीं चाहिए उतना। Is it clear? That is the drainage function. Any doubts? Sure. Shall I move ahead? Last point. ये उसका यूनिक फंक्शन एब्जॉर्प्शन। अपन ने स्मॉल इंटेस्टाइन का डायग्राम देखा था। What are these? Villi. Surrounding to the villi are blood capillaries, not blood vessels. So parallel to the blood capillaries are lymphatic vessels, जिनको नाम दिया जाता है the lymph vessels present in the small intestine are referred as lacteals. What is the function? They absorb the fats from the villi into the lacteals. Then, बहुत ही यूनिक है. Okay, fats, चर्बी. Okay, अगर सोचो स्मॉल इंटेस्टाइन को एब्जॉर्ब के डायरेक्टली ब्लड वेसल में जाता तो क्या होता? स्मॉल इंटेस्टाइन से एब्जॉर्ब हुआ और वो ब्लड वेसल में जाता. What would happen? It circulates the blood vessel. Blood vessel में majorly fat deposition, blockage ज़्यादा जल्दी होता है. But that's the nature's way to keep the blood vessel at least somewhat free of fats. चाहता है blood vessel में थोड़ा बहुत, but comparatively less. Reason because majority will go into the lacteal. Lacteal से वो body के body parts में जाते जाते distribute करते जाएगा, छोड़ देगा. Okay, when it will go to liver, वहाँ पे deposit करेगा, fat depots, arms वगैरह सब जगह पे deposit करेगा. So it absorbs all the fats from the small intestine into the lacteals. These are the four important functions of the blood. Is it clear, everybody? That completes the lacteal vessels and that completes circulation in human beings. Any doubts? आगे बढ़ू. चलन आगे का पार्ट सबसे इम्पोर्टेंट, सबसे फनी पार्ट भी है. हेडिंग क्या लिखा है? अब जो ट्रांसपोर्टेशन इन प्लांट्स के नीचे सब हेडिंग लिखी है. पहला सब हेडिंग, नेक्स्ट सब हेडिंग, थर्ड हेडिंग. ओके, अब वो कंडीशन फॉर इजी डिफ्यूजन, उसके बाद प्लांट ट्रांसपोर्ट सिस्टम में दो वर्ड हाईलाइट किए हुए हैं, जाइलम एंड फ्लोएम. एक्चुअली वो पूरा इतना जो पढ़ा है ना, हेडिंग लिखा है, उसमें से अपने काम का जाइलम एंड फ्लोएम. ये बाकी सब बकवास पे एग्जाम में पूछेंगे नहीं. जस्ट नीड कहानी की तरफ पढ़ना है, आगे बढ़ना है. अपने काम का सिर्फ दो वर्ड, जाइलम एंड फ्लोएम. व्हाट इज जाइलम कंडक्टिंग? What is phloem conducting? Good. Good. Next page. हाँ, वो टर्जिडिटी वाला 10 पॉइंट. उसके बाद हेडिंग लिखा है absorption of what plants. ठीक है, तो उसमें दो वर्ड हाईलाइट किए हुए. I hope 9th standard tissue chapter. Xylem is made up of 4 cells. Brackets, vessels, xylem parenchyma, xylem sclerenchyma. याद है ना? अच्छा, उसके बाद नहीं, उसके बाद ना कहीं पे एक लाइन, उसके बाद एक पॉइंट छोड़ के नेक्स्ट, the cells of the root in contact with the soil actively take up ions. मिला? Online children, please pay attention. वहाँ पे just below the absorption of water by plants, कौन सा पॉइंट है वो नंबर? 6 पॉइंट. चलो 6 पॉइंट को मार्क करना इंपॉर्टेंट, और वहां से अपने काम का आंसर चलना. उसके ऊपर से जितना भी था अपने कुछ भी काम करना. एक्चुअली आंसर वहां से स्टार्ट होता है. Okay, and I said transportation of water in plants, my answer starts from there and not from the above. Above part is basically just the expression, brief understanding. Am I clear? Okay, so children, first part में we come back to the first page again. यहाँ first part में I want you guys somewhere to read out that part. James. Madam, can you read that part?
Raw materials and source. Main source is soil. Raw materials, nitrogen, phosphorus, and other minerals.
Okay, in short, what you're talking about is some transportation in plants. Mein, what is the transport thing? Either water, minerals, or food. अभी अपन पहले in general बोल रहे हैं soil is the main source of nutrients. So whatever plant get, it is getting from the soil. Okay, soil के अंदर all the nutrients are present. 9th standard macronutrient, micronutrient, geography किधर किधर तो पढ़ा होगा. Okay, so nitrogen, phosphorus, potassium, sulfur, many are things. Okay, all that are the raw materials required by the plant, which are derived from the soil.
Main part of plant for absorption of minerals and water are roots.
I think everybody knows this. Next.
Condition for easy diffusion. The distance between soil contacting organs and chlorophyll containing organs should be small.
Again.
So that every raw materials can easily diffuse to all parts of the plant body.
Okay. अच्छा, इसका मतलब समझना है इनको. ये आपका प्लांट है. Which is the source? Source क्या है? Soil. Absorbed by roots. Roots से कहाँ पे जाना चाहिए? Chlorophyll containing part, that is leaves. Now, sir, the distance between the root to the chlorophyll containing part. Okay, अगर ये बहुत छोटा होगा, like मान लो grasses, root or leaf. Leaf का तो यहाँ पे green का जरूरत है. So is the distance big? No. When the distance is small to move, it can easily move by diffusion. छोटे distance के लिए diffusion काम आएगा. But when the distance is very huge, coconut tree, mango tree, the banyan tree, distance बहुत बड़ा होता है. Can simple diffusion take place? No. So then I need to go for a alternative method, and that is what I am going to study in this chapter. Am I clear?
If distances are large, diffusion processes will not be sufficient to provide raw material to leaves and energy levels.
Okay, distances are large, simple diffusion cannot take place, and energy will be required for it to carry to that level. That is why we need to go for an alternative plant.
Plant transport system. In leaves, water and CO2 are used for photosynthesis. Plants having low energy needs will use slow transport system. In tall trees, the energy needs will be more, so it will use fast transport system.
Okay, and especially when the plant energy requirement is less, but in terms of tall plant, okay, it requires that it should carry the water much faster. So उनकी energy requirement थोड़ी ज़्यादा होगी compared to the smaller plants.
Two pathways include conducting tubes like xylem, moves water and minerals obtained from the soil, and phloem, transport products of photosynthesis from the leaves to the other parts of the plant.
Do I need to explain that? Xylem conduct water and minerals from where to where? Roots to leaves. What about phloem? It conducts food to all parts of the plant in all directions, not only leaves. It conducts food to all parts in all directions. Leaf to root, root to leaf in all directions. Okay. So basically, xylem mein the movement is unidirectional, and in phloem it is bidirectional or all direction. Okay. Next.
Water is essential for normal growth and development.
Is काम करना.
Water gives turgidity to the plants. Turgidity is necessary for non-woody tissues for mechanical support.
What is turgidity? Swelling of the cell. अगर ये cell है completely filled with water तो swell होगा. ये shrink होगा. So if the cell is swollen, completely turgid, then only it will give mechanical support to the plant. For herbaceous or non-woody plant, because non-woody plant में अगर पानी निकाल दिया तो देखते हैं ना सर droop down होता है. That is because there is no water. अपने घर पे देखो ना plants होंगे 3-4 दिन पानी मत डालो ऐसे सो जाते हैं. Are they dead? No. For longer period of time no water, then they will die. but for 2 or 3 days, you just leave the honey diya will back to be fresh condition. Is it clear? Next.
Loss of the acidity leads to building of leaves and dropping of shoots.
Then, drooping of the leaves and technically loss of water is known as हिंदी में सिंपल लैंग्वेज में मुरझा जाना is wilting. Okay, am I clear? Wilting. तो ध्यान कहाँ पे है? Next.
Absorption of water by plants.
Okay, children, absorption of water by plants. First 5-6 पॉइंट जो है वो सिर्फ बेसिक अंडरस्टैंडिंग के लिए, अपने लिए इतना इंपॉर्टेंट नहीं है, बट बुक में दिया है इसके लिए पढ़ रहे हैं. आंसर मैंने जो मार्क किया है वहां से स्टार्ट होगा. सो या
Water can enter plants through its entire surface, but maximum water is absorbed through the roots, especially at the tips.
या आई थिंक वेरी क्लियर. पानी सब जगह से जा सकता है, नो डाउट, बिकॉज़ इट कैन एब्जॉर्ब सम अमाउंट ऑफ वाटर, but maximum water will be absorbed from the roots, and that to root tips and root hairs. Next.
The root hairs increase the surface area for absorption of water.
I think already know everybody. Next.
The water enters the root hairs, and from there it moves to the epidermis, root cortex, endodermis, and reaches the xylem vessels, through which it is further transferred.
Okay. मतलब ये याद रखा तो अच्छी बात है, नहीं not required as such, but still, if I take a cross section of a root, इसका cross section लेता हूँ तो ये ऐसे नहीं. ये रूट का क्रॉस सेक्शन है. व्हाट विल बी दिस स्ट्रक्चर? रूट है, ये रूट है, और रूट है के एक्सटेंस को अपन रूट हेयर बोलते हैं. द रूट हेयर और द आउटर मोस्ट लेयर ऑफ द रूट इज एपिडर्मिस. बीच में एक लेयर होता है एंडोडर्मिस. एपी आउटर, एंडो इंटरनल. इन दोनों के बीच में पैरेन्काइमेटस सेल, 9th स्टैंडर्ड टिश्यू चैप्टर, पैरेन्काइमा present in the stem or root is known as root cortex. So water कहाँ से move कर रहा है? Water is moving from soil to root hair, epidermis, cortex, same here will be cortex, endodermis, till it reaches the xylem. Xylem ट्यूब है, अगर ये रूट है तो ट्यूब का आपकी होगी ऐसे बाहर आती होगी, स्टेम ऐसे होगा ना इस केस में, तो ये xylem ट्यूब में एंटर करेगा. So that is what the pathway of water movement is.
Water and minerals dissolved in it moves up through the conducting tissue called xylem, which is made up of tracheids and vessels.
Okay, ultimately it moves in xylem, which is made up of tracheids and vessels, because xylem parenchyma storage का function करता है, xylem sclerenchyma strength का function करता है. So main conducting tissue कौन सा होता है? Tracheids and vessels. They are pipe-like structures which conduct water and minerals upwards. Next.
In xylem tissues, vessels and tracheids of the roots, stems, and leaves are interconnected to form a continuous system of water conducting channels reaching all parts of the plants.
Tracheids and vessels are connected होते हैं from sideways and upward, so that forms a conducting tissues right up to upwards, and water moves like this upwards, sideways also go to each and every part of the plant.
The cells of the roots in contact with the soil.
Stop. Okay, यहाँ तक किसी को डाउट है? Everybody? Sure? Shall I move ahead now? Right. Okay, now for some time, I want you to close your volumes. I hope you understand what it is. Good. Leave. Okay. Now, within this, is a tube which conducts water and minerals. What is this tube? ये xylem. Okay. अब मैं क्वेश्चन टू एवरीबॉडी ध्यान से सुनना है और आंसर करना है. ये क्या है? सॉइल. ये क्या है? आया ना? सॉइल के अंदर क्या है? और so I give it as water plus minerals को अपन ions बोलते हैं. वाटर को अपन W बोलते हैं. In short, it is made up of water and ions. अब सोच के बताओ, आयंस सॉइल में ज्यादा होता है या रूट में ज्यादा? सॉइल में ज्यादा होता है. आयंस आर मोर ओवर सॉइल. तो आयंस रूट में क्या होंगे? कम होंगे. जिनमें आयंस ज्यादा होते हैं, सोल्यूशन है ये वाटर एंड आयंस का. When the ions is more, what about water concentration? Less. आयन ज्यादा जो तो पानी ज्यादा ऑक्यूपाई होगा ना, so water would be less. इसमें क्या होगा? Water will be more. Please remember, wherever ions are more, if it is solution, then water will be less. सबको डायग्राम समझ में आया? Everybody? No. Diffusion पढ़ा होगा 9th में होगा. What is the definition of diffusion? Movement of substances from a higher concentration to lower concentration. Good. Then water molecules move from higher to lower and lower to higher. Higher concentration of water to lower concentration of water. कहाँ से कहाँ मूव करेंगे?
According to diffusion, you would say soil is appreciated.
Good. According to diffusion, it tends to move from higher concentration. Now, what is higher concentration? Roots. To lower concentration soil. Perfectly right. Okay. Water will move from roots to soil according to this diagram. हाँ या ना? ऐसे हुआ तो प्लांट का क्या होगा? मुझे जाएगा और अल्टीमेटली okay, do we want this? पानी अंदर लेना है या पानी बाहर भेजना है? Okay, but is that condition okay? जिलन कंडीशन ये है, इसके वजह से पानी अंदर नहीं आएगा, पानी बाहर जाएगा. समझ में आया? Said it rightly. From inside to outside water. हमें क्या चाहिए? Outside to inside. कैसे करोगे? आयंस को कम करके. आयंस को किधर बढ़ाना है? That's the first process which happens. सबसे पहले the plant undergoes active transport. यहाँ पे एक्टिव ट्रांसपोर्ट. What is active transport? The cells of the root spends energy and take up the ions. Energy खर्च करके मुझे अंदर ले लिया. So what will happen? Ions increases. Here it decreases. So same goes over here. अब is my requirement satisfied? आया ना? अब मुझे जैसे चाहिए पानी अंदर आने के लिए वो क्राइटेरिया सेटिस्फाई हुआ. जैसे ही एक्टिव ट्रांसपोर्ट हुआ, more amount of ions enter into the root air, which leads to the second step. In order, there is a concentration gradient created, which for to minimize this concentration gradient, immediately osmosis takes place. What is osmosis? It is also type of diffusion, but only water molecules. So, यहाँ पे water ही को move करना ना. So there is a movement of water from its higher concentration to its lower concentration across a semi-permeable medium. Is known as osmosis. पानी कहाँ पे आया अभी? Roots में आया. यहाँ पे पानी पहुंचा. अब तीसरा स्टेप होता है. What is the third step occurring? पानी यहाँ पे पहुंच चुका है. मैं ग्रीन कलर दिखा रहा हूँ पानी. चलो मान लो रूट्स को मैं बड़ा कर देता हूँ. और यहाँ पे ये सेल्स हैं. पानी कहाँ पे पहुंचना है हमें? Xylem में. So what is happening? The water has reached here. अब कहाँ पे बढ़ना है? It has passed through epidermis, cortex, endodermis till it reaches the xylem. Now, water यहाँ पे आ चुका है. Now, cell to cell osmosis takes place. यहाँ से यहाँ पे, यहाँ से यहाँ पे, यहाँ से यहाँ पे, यहाँ से यहाँ पे. So osmosis will continue to occur till, and this osmosis, central osmosis, pushes the water towards the xylem. इसको बोला जाता है root pressure. Because cell to cell osmosis is a pressure gradient at the root level which pushes the water towards the xylem. समझ में आया? So due to root pressure, the water enters into the xylem bond. पानी कहाँ पे पहुंचा? xylem bond. अब पानी xylem में ऊपर आया, मान लीजिए. तो पानी इतने तक ही ऊपर है. इसके ऊपर जाना है. क्यों? एक ग्रेविटेशनल पुल पानी को नीचे खींचेगा. दूसरा, ये जो प्रेशर क्रिएट हुआ इतना भी नहीं है कि पानी को इसके ऊपर जा सके. सो इट इज जितना प्रेशर था उससे इतना ही आ चुका. ओके, कैन नॉट गो बियॉन्ड दैट. नो. सो नेक्स्ट occurs is at the fourth point. There is on top, water is continuously being lost due to transpiration. So यहाँ पे इस पॉइंट से लेके इस पॉइंट तक there is a partial vacuum created. जैसे ही partial vacuum create होगा क्या होगा? Immediately the water will be pulled upwards, and this pulling is known as transpirational pull, which pulls the water right up to the leaves, and that's the force. Root pressure is not good enough to carry the water to the tip of the plant. So what is the other option for the plant? The plant undergoes transpiration, due to which is a partial vacuum created in the xylem column, which creates a suction pump or suction force that is known as transpirational pull, which pulls the water. Is it clear? पर अगर मान लो ये प्लांट का हाइट ही इतना हो, तो पानी ऊपर तक पहुंच गया, यहाँ तक तो पानी पहुंच गया ना, टिप तक तो आ ही गया. ओके, सो ट्रांसपिरेशन पुल किस प्लांट में ज्यादा इंपॉर्टेंट होगा? So all tall trees में water simply cannot be carried forward by diffusion, osmosis or root pressure. So in taller plants, it requires alternative option, that is transpiration pull. Transpiration pull can carry water up to meters. Coconut tree, सबसे बड़ा प्लांट कौन सा है? वर्ल्ड में सबसे बड़ा प्लांट कौन सा है? Redwood, red pine wood. In Metacyclia, the botanical name, almost 363 feet. Which is okay, children, 363 feet. आपका ये शायद 10 फीट का होगा. So 300 feet का पकड़ के चलते हो 36 फ्लोर के आसपास इतना बड़ा प्लांट पानी ऑब्जॉर्ब करते हैं because of the transpirational. Am I clear? Everybody. Okay. So first active transport. Why active transport? Because ions are ज़्यादा. What I want, not outside. I want it inside. So we first actively take up ions inside the root. Once actively take up ions, it creates a concentration difference. Immediately, osmosis takes place to get the concentration difference, and then the water will push upwards because of the root pressure. But root pressure is not good enough to carry the water to the leaves in higher plants or tall plant. The plant goes for transpirational. समझ में आया? Okay. I want you to open your volumes because please number. Okay. So मैं आपको कुछ मार्क करने बोल रहा हूँ, इसके साथ मार्क कर लीजिए. हमने कौन से पॉइंट पे मार्क किया था? सेल्स ऑफ अ रूट कॉन्ट ना, वहां पे अपन इनपुट साइड किया था. सन वहां पे अंडरलाइन soil actively take up ions. Soil actively take up ions. वहां पे 1 लिखोगे. फिर which creates the difference in the concentration of the ions between the roots and the soil. The requirement था वो हो गया अचीव. फिर water continuously move into the root cell to eliminate the difference. Water continuously moves into the root cells. Underline that. Okay. वहां पे 2 लिखोगे और 2 के ब्रैकेट में डालोगे osmosis. मतलब पानी जो अंदर आ रहा है वो किसके वजह से आ रहा है? Osmosis. And creates a column of water which is steadily pushed upwards. No, pushed upwards. उसको अंडरलाइन करोगे और वहां पे 3 लिख के ब्रैकेट में डालोगे रूट प्रेशर. उसके बाद वही लिखा हुआ है, देखो नेक्स्ट लाइन में. However, this pressure by itself. उसको पता क्या करना है? Hold for a minute, let him complete fast. स्कूल में चैप्टर हो गया ये? कहाँ तक पहुंचा? थ्री पहुंचा। तुम लोग का ही है ना एक्स्ट्रा बाकी हर स्कूल में ऑनलाइन है ना, so there is no point in our sitting for online. सिंपल है ना यार, went to native place, लेटर दे देने का, went to native place या कुछ बोलने का, not comfortable, doctor is said no sitting for online due to pain in the eye. अरे यार वो चीज को सी बिकॉज सिंपल है, ऑनलाइन में क्या होगा, 4 टीचर में से एक टीचर अच्छा ऑनलाइन में डिफरेंस होता है। फिर आई एम नॉट आल्सो इंटरेस्टेड बिकॉज आधे दिन का वीडियो ऑफ होता है, क्या कर रहे क्या नहीं कर रहे पता नहीं चलता। ऑफलाइन में ऐसा ऑप्शन नहीं है ना अपने पास, कश करेगा बैठने का, कितना आलसी है तू। Next, higher pressure itself is unlikely to be enough to move water over the heights. Normally tall plants के लिए over the heights मतलब. So what is happening? Use another strategy to move water in the xylem upwards to the highest point of the plant body. Bracket में transpiration वाला है कि अतलन वहाँ पे ठीक है. Use another strategy to move water in the xylem upwards. That is वहाँ पे upwards के बाद छोटा सा asterisk करके that is ट्रांसपीरेशनल पुल। तो वो आगे का लाइन लिखने की जरूरत नहीं, दैट इज ट्रांसपीरेशनल पुल। ओके अतलन वहां पे ट्रांसपीरेशन पुल लिखा, ओके अब वहां से ना ट्रांसपीरेशन पुल मान लो यहाँ पे आ गया, वहां से एक पेंसिल से ऐसा एरो करना और वो एरो को ना ट्रांसपीरेशन के सेकंड पॉइंट पे कनेक्ट कर देना, कनेक्टेड टू ट्रांसपीरेशन का सेकंड पॉइंट। इवापोरेशन ऑफ वाटर फ्रॉम वाटर मॉलिक्यूल्स फ्रॉम सेल्स ऑफ अ लीफ क्रिएट्स अ सक्शन व्हिच पुल्स वाटर फ्रॉम द जाइलम सेल्स। हाँ ट्रांसपीरेशन पुल के आगे अब इस लाइन से कंटिन्यू करोगे। इवापोरेशन ऑफ वाटर मॉलिक्यूल्स फ्रॉम सेल्स ऑफ अ लीफ okay creates a suction which pulls water from xylem cell, this is known as transpiration pull। जब वहां पे थोड़ा चेंज करना है, वहां पे लिखा है evaporation of water molecules from cells of a leaf, वो लीफ के आगे वापस पुट आइकन और वहां पे डालना ऐड करना है ट्रांसपीरेशन ब्रैकेट में। उस प्रोसेस को क्या बोलते हैं? ट्रांसपिरेशन बोलते हैं ना। जस्ट राइट ट्रांसमिशन ओवर देयर, लीफ के आगे। अचलन यहाँ पे मैंने आपको ना उसको शॉर्ट कर दिया, वरना आपको क्या करना पड़ता था, पूरा लिखना है, फिर ट्रांसमिशन का डेफिनेशन लिखता था, फिर ट्रांसमिशन पुल लिखना पड़ता था। अपन ने एक स्टेटमेंट में लिख दिया इवापोरेशन ऑफ वाटर मॉलिक्यूल्स फ्रॉम अ सेल्स ऑफ अ लीफ ब्रैकेट में ट्रांसपिरेशन क्रिएट्स अ सक्शन व्हिच पुल्स वाटर फ्रॉम द जाइलम सेल्स नोन एज ट्रांसपीरेशनल पुल। तो ऊपर का पॉइंट अपने नीचे एक्सप्लेन भी कर दिया। आपका आंसर सिर्फ यहाँ तक ही है, तो वहां पे वो ब्रैकेट क्लोज करके ट्रांसपीरेशन पुल के आगे आप क्लोज करोगे एंड दिस इज द पॉइंट अप टिल वेयर इफ द क्वेश्चन इज आस्क्ड हाउ डज द वाटर इज एब्सॉर्ब्ड इन द प्लांट। स्टार्टिंग पॉइंट क्या होगा? एक्टिवली टेक अप आयंस, फिर ऑस्मोसिस, हाउ डज द प्लांट एब्सॉर्ब वाटर और ट्रांसपोर्ट वाटर टू द लीव्स। तो ये लिखोगे, पहला एक्टिव ट्रांसपोर्ट, फिर दूसरा ऑस्मोसिस दैट इज वाटर मूव्स इनसाइड, तीसरा द कंटिन्यू वाटर पुशिंग अपवर्ड्स इज नोन एज रूट प्रेशर व्हिच इज नॉट इनफ सो द प्लांट गोस फॉर ट्रांसपीरेशनल पुल। और फिर ट्रांसपीरेशन पुल का लास्ट एक लाइन लिखनी है। समझ में आया? ओके उसके बाद जितना भी लिखा होगा अगेन टिक टाक ओके, अभी ध्यान देना, उसके इमीडिएट नेक्स्ट लाइन के बाद क्या लिखा है? नो सॉरी, इमीडिएट लाइन में क्या लिखा है? गुड, जो भी पढ़ा था अब आपको अंडरलाइन करना है। ट्रांसपीरेशन इज अ नेसेसरी इवेंट, इतना अंडरलाइन करना है। ओके, बचपन मार्जिन में ऊपर या नीचे साइड में कहीं पे भी लिखना है, आप लिखोगे ट्रांसपीरेशन इज अ नेसेसरी इवल, लिखना है वापस, ट्रांसपीरेशन इज अ नेसेसरी इवल फॉर फोटोसिंथेसिस टू ऑकर। Transpiration is a necessary evil for photosynthesis to occur. Full stop. आगे comment. सर ये क्वेश्चन एप्लीकेशन बेस्ड फॉर्म में है। हाँ, Transpiration is a necessary evil for photosynthesis to occur. Comment. चिल्ड्रन, सबसे पहला चीज वहां पे ट्रांसमिशन इज अ नेसेसरी इविल, नेसेसरी भी है, इविल भी यूज हो रही है, मतलब क्या? ठीक है, थोड़ा बहुत ठीक है।
So in transpiration, plant uses water and it is necessary because it provides water or sun.
इविल क्यों है? तुम लोग मुझे सबसे नेसेसरी बता रहे हो, इविल का पार्टी नहीं आया इसमें।
No, because transpiration uses water that is essential for the photosynthesis.
ओके, तो मैं आपको एक सिंपल एग्जांपल देता हूँ। मान लो तुम दोनों दोस्त हो, बेस्ट फ्रेंड। हाँ, मैं एग्जांपल दे रहा हूँ, ओके? और तुम जंगल में गए, जंगल, ओके? अब इस बंदे को बहुत ज्यादा प्यास लगी, मतलब एकदम ऐसा भी पानी नहीं होना चाहिए मरने के इसमें, इतना बहुत सारे चल रहे थे, प्यास बहुत लगी है। लकीली जंगल के बीच में एक कुआं दिखता है, इट इज फिल्ड विथ वाटर एंड लक इज टू गुड फॉर यू, वहां पे रोप भी है और वेसल भी है, पानी खींचनी है, ठीक है? एंड द वाटर इज क्लीन ड्रिंकिंग वाटर, पानी साफ सुथरा भी है, पीने के लायक है। गुड, सुपर, ओके? प्यास तुम्हें लगी है, ओके? अब दोस्त है, बेस्ट फ्रेंड होता है, तू बैठ जा, थक गया है, मैं तेरे लिए पानी निकालता। अब पानी निकालना मतलब इसलिए कोई पानी निकाला नहीं था, फर्स्ट टाइम ट्राई करा, सो ही पुट द थिंग, बड़े द वेल इज टू डीप, तो पानी है, बहुत मेहनत करके और बहुत पेशेंस के बाद मटका पानी भर के आ जाता हूँ। ऐसे कैसे तो आता है, हाथ को लग रहा है वगैरह, वो रस्सी का पर खींच के निकाल ही लिया, ये पानी निकाला, देखा पानी वॉज क्लीन ड्रिंकिंग वाटर, इसने अपने दोस्त को दिया पीने के लिए, ये दोस्त ने लिया, देखा और सीधा मटके को वापस वेल के अंदर डाल दिया, लिया और डाला पानी के अंदर वापस। What would be your immediate reaction? Spontaneous तो ये होगा ना, बे साले इतना मेहनत करके निकाला, तुझे इतना हाथ को लगा और तू क्या किया, सीधा फेंक दिया। मन तो करेगा तुझको भी डाल दो कुएं के अंदर, हाँ या ना? फाइनली यही होगा। चिल्ड्रन, ट्रांसपीरेशन इज अ नेसेसरी इवन, बिना ट्रांसपीरेशन के ट्रांसपीरेशन पुल क्रिएट होता, और बिना ट्रांसपीरेशन पुल के पानी ऊपर जाता, अब बिना पानी ऊपर पहुंचे फोटोसिंथेसिस होता, तो ट्रांसपीरेशन इज नेसेसरी फॉर फोटोसिंथेसिस। इतना समझ में आया? अब इवल क्यों? पानी अंदर कैसे आया?
Active transfer.
एक्टिव ट्रांसफर, बिना एक्टिव ट्रांसफर के पानी अंदर आता नहीं। तो सेल ने क्या किया है यहाँ पे, प्लांट ने क्या किया है? एनर्जी खर्च की है ना, और एनर्जी खर्च करने के बाद इसने क्या किया? इतना एनर्जी खर्च करने के बाद पानी अंदर लिया, पानी अंदर लेने के बाद सेल प्लांट ने क्या किया? पानी फेंक दिया। 99% of water is lost in transpiration. गेन नहीं, 99% एब्जॉर्ब किया, 100 का एब्जॉर्ब किया, उसमें से 99% पानी आप फेंक दिया, मतलब इतना एनर्जी खर्च करके जो अंदर लिया। So is it evil? Yes ना, but necessary evil, because बिना इसके फोटोसिंथेसिस को पानी नहीं पहुंचता। That is why transpiration is a
जो सॉइल में है पानी अगर ट्रांसपोर्टेशन नहीं होता तो उसकी जरूरत नहीं पड़ती, वही यूज होता है।
फोटोसिंथेसिस के लिए वही यूज होने के लिए पहला पानी नीचे से तो आना चाहिए ऊपर।
पानी एक लेवल तक ही जाता है, पर ट्रांसपोर्टेशन को ले
पर पानी ऊपर है ना,
तो पानी ऊपर कहाँ से आ रहा है? वो भी नीचे से ही आ रहा है। Okay, that is we are considering कि वो प्लांट बढ़ते जा रहा है, उसके हिसाब से पानी ये तो बहुत कॉम्प्लिकेट हो जाएगा। ट्रांसपिरेशन it can absorb water, very minimum, not too much। और क्या 365 डेज पानी गिरता है, बारिश होती है तो ना, only recent में थोड़ा बहुत वीक एब्जॉर्ब करेगा, बाकी के टाइम अपन जब पानी डालेंगे तब जंगल में क्या तू पानी डालने आता है? That is where transpiration pull is necessary. Am I clear? उसके बाद manual transpiration is sufficient to raise the water up to top of tallest trees, already explained. Under optimal condition, leaf can transpire its own weight of water in less than an hour, not required. A leaf trees transpire equivalent to 100 times its weight in its lifetime, not required. Okay, other functions देखना। Transpiration helps in absorption and upward movement of water and mineral dissolved in it from roots to the leaves. It also helps in temperature regulation. The effect of root pressure in transport of water is more important at night. Then it also helps in temperature regulation. यहाँ तक function है। The effect of root pressure in transport of water is more important at night. ये नहीं है function। Okay? During the day when stomata of the transpiration pool becomes the major driving force in the movement of water in the xylem. ये function नहीं है, इसी उसका expansion part था, not required over. That completes transportation of water in लेक्चर कितने में खत्म होता है? 6 मिनट है। Next is transportation of food and other substances. Actually, यहाँ पे सबसे बेस्ट कॉमेडी इवन योर टेक्स्ट बुक में भी और यहाँ पे भी तो उन्होंने एक्सप्लेन किया ही नहीं ये प्रोसेस को। अब यहाँ पे आप देखोगे वहां पे नोट लिखा है, दिखा चिल्ड्रन, एक्चुअली पूरा एक्सप्रेशन नोट में है, बाकी सब बकवास है। मतलब जनरल नॉलेज आपको एक डायग्राम ड्रॉ करके दिखा दूंगा मैं, वो डायग्राम आप ड्रॉ करोगे ना, उसी डायग्राम को एक्सप्लेन कर दिया, आंसर आ गया। ट्रांसपोर्ट ऑफ फूड अपने वर्ड्स। सो नाउ वी आर नॉट गोइंग टू डू दैट, टुडे विल स्टॉप ओवर हियर। ओके, आई थिंक मे बी 1.5 hour मोर और व्हाटएवर। अब चिल्ड्रन थोड़ा वॉल्यूम के पीछे जाना, बताता हूँ। very short answer type questions. Okay, question number 1 से लेके question number 56. क्वेश्चन नंबर 61 से लेके पहला 1 से लेके 56, फिर नेक्स्ट 61 से लेके 88, फिर 91 से लेके 101, 101. चल क्या करोगे? Okay. Go to the questions, पढ़ो, find out the answers. बुक में मार्क करो. I am not interested in क्या लिखना, वो तुम्हारा चॉइस है. Okay. What I am more interested is, okay, then questions दिए गए, whether you are able to get the answers. जिस क्वेश्चन को आपको डाउट है, नॉट प्रॉपरली नो, आप क्या करोगे? सर्कल करोगे. सो वी विल डिस्कस. जब मेरे हिसाब से अगर आप राइट भी करना चाहते हो, ओके, यू कैन डू इट फॉर द फर्स्ट 30-40 क्वेश्चन. जो वन वर्ड के है ना, वो वन वर्ड के है, वन लाइन के. तो आप अपने एक अलग नोटबुक में लिख लो ना, वहां पे आपके क्वेश्चन बैंक बन जाएगा, आंसर बैंक. सब कुछ लिख दिया, सिर्फ उठाना है और पढ़ना है बस. ओके, नहीं आप किसी नोटबुक में बनाओ, योर चॉइस. बचलन वहां पे अगर बना रहे हो, अभी फिलहाल जगह छोड़ देना. पर एग्जाम के लिए अगर पढ़ना है, कंटेंट पार्ट पढ़ने से मतलब नहीं, क्वेश्चन आंसर ही पढ़ना. तो क्वेश्चन और आंसर साथ में होना चाहिए. क्वेश्चंस के लिए आप बाद में ये करो, आपके ऊपर, बाद में क्वेश्चन लिखते बैठना. फर्स्ट पार्ट में यू विल नॉट हैव एनी प्रॉब्लम फाइंडिंग द आंसर्स. प्रॉब्लम कहाँ पे आएगा? वेरी शॉर्ट आंसर, सॉरी शॉर्ट आंसर्स, वहां पे थोड़ा प्रॉब्लम आएगा. और लॉन्ग आंसर्स में इतना नहीं आना चाहिए. हाँ, जहाँ पे आपको वन वर्ड लगता है वहां पे लिखो. अगर आप अलग से बुक बनाओगे, तो जो अलग से नोटबुक में लिखोगे, तो आपके ऊपर चॉइस. मैंने कंपल्शन नहीं की, मैं आपके ऊपर चलोगे. रीजन ये आपको हैंडी गिरेगा व्हेन यू स्टार्ट लर्निंग फॉर द एग्जाम्स. 103 क्वेश्चन कवर करने के बाद कुछ बचता ही नहीं उस चैप्टर में से पढ़ने. पूरे आंसर्स कवर हो जाएंगे उसमें से. एग्जाम में क्या आएगा? क्वेश्चन घुमा फिरा के आएगा, आंसर्स तो वही आने वाले. सो इट वुड बी एटलीस्ट 1 मार्क और 2 मार्क के आंसर. नॉन क्वेश्चंस का लिखने का जरूरत ही नहीं पड़ेगा. ठीक है, चॉइस आई एम लिविंग इट टू यू, नो कंपल्शन फ्रॉम माय साइड. हाँ, पर मेरे कंपल्शन में आपको गो थ्रू करना है, और मुझे बाद में पता चलेगा आपने गो थ्रू किया है कि नहीं. बिकॉज नेक्स्ट लेक्चर आई वुड आस्क यू, क्वेश्चन नंबर 10 का आंसर तू दे, क्वेश्चन नंबर 11 का आंसर तू दे. मार्क किया होगा ना आपने, तभी किधर भी आपका अगर मानने से कि अपना वीआईपी फ्रेंड मेरा. क्वेश्चन नंबर 10 मैं होमवर्क ही नहीं किया है सर, नहीं पता. बड़ा सिंपल आंसर, सर सर्कल कर दो, नहीं पता. ओके, आई नो व्हिच आंसर्स यू विल बी हैविंग डाउट एंड व्हिच आंसर्स यू डोंट हैव अ डाउट. तो तू ऐसा बोलेगा कि सर व्हाट इज, वेयर इज द क्लोरोफिल प्रेजेंट, उसको डाउट हुआ, फिर तू दुनिया में रहेगा कि नहीं डाउट. Bye children, enjoy. ऑनलाइन चिल्ड्रन आपको भी ये करना है. इनको बात मत करना. जितना बोला उतना सब गो थ्रू दैट. शॉर्ट आंसर्स में तो मैं डिस्कस करता हूं, शॉर्ट और लॉन्ग आंसर भी डिस्कस. तो 1 मार्क के है ना, वो नॉर्मली आपसे आंसर.
especially after
मेरा फोन नहीं है मैं खाबू ले जाऊँ
देखिए तो योगी का क्लास भी चल रहा है हाँ इसको पता है सुपर इसकी मम्मी का टीचर है
दो बार किया ना उसको लगा वो भी करता है
Actually, 148 था, but 7390
बिना समझ में नहीं सब बोला 5 है
मुझे भी आया नहीं अरे यार आई
देख तेरे जीवन में आगे का
इधर भी रहेगा मैंने बोला 7 कब तक रहेगा ये 1 million marks 6 साल से
I scored 1,580,000.
मेरा 0
सिमरन में पढ़ते थे
सबसे सब पहले हमारे पास
वो सब्जेक्ट
अब मैं पहले आपका ऐसा होता है हाथ में ऐसे पहले मैं पीटी पे एडिटेड था
एक सेकंड तो काम से किसी डेट को है
क्योंकि काम से डेटा नहीं करता
हमें सुने देते हैं ये
जब भी करता है आपको ट्रेन आ रहा है सब कुछ शुरू है jump in there, uसी second कर दो.
तो इंटरनल टावर में एक बार में इसको मार देना है. इससे डेवलप कार्ड बीच में बचेगा. आयुष नंबर चाहिए क्या तेरे पास? इसकी बहुत जरूरत है.
Good afternoon, everyone. Appendix one, they have better. One appendix is less. Two. But they are all like that. And everywhere. So good afternoon, online students. Are you all there? Students online? Acknowledge. They have taken a lunch break. All right. कहाँ पे थे हम लोग? Where were we in the last class? Concave lens completed?
Yes.
So all the image formation completed? Okay. So 6 plus 2, 8 for mirror and 8 for lenses. Everything is complete. So we'll begin with numericals for this chapter after completing few more topics in this. Which is sign convention for spherical lenses, lens formula, magnification and power of lens. Okay. All right. Let us start. Online students, please let me see your video. I'll have to ask madam to call them then. Madam, the online students are not present. Can you call them and check and ask them to keep their videos on?
Yes, sir. Yes.
Yeah. Okay. Sign convention for spherical lenses. So it is exactly same as it was for spherical mirrors, right? So what was the sign convention? It was Cartesian sign convention. In this case, the origin is optical center. Okay, over there it was pole, right? So all the distances will be measured from optical center. Anything to the left of optical center will be negative. Anything to the right of optical center would be positive. Anything above the principal axis on both sides would be positive. Right? Anything below the principal axis will be negative. Okay, these are the sign conventions. And the incident ray will always be from left to right. Incident ray. Left to right. apart from this, another sign convention is focal length.
Arya, please start your video.
Focal length of convex lenses always positive, right? And focal length of concave lens is always negative. Concave lens is always negative. Can anybody tell me why?
So for convex lens, we consider the focal length that is on the right side of the optic.
Why do we consider that?
So because after the incident ray passes through the lens, it meets at that time.
Correct. So wherever the parallel rays meet or divert away after refraction, that's the principal focus. Okay. So for convex lens, the parallel rays would meet at F. Whereas for diverging lens, since it is diverging, the incident parallel rays would be diverted away from focus. So wherever they meet, then extended backward. Right? So for convex lens, the focal length is always positive. And for concave, it is always negative. So wherever this the direction should be away. Is it understood why for concave lens focal length is always negative and it is always positive for convex? Because wherever these incident rays meet or appear to meet after refraction. Okay, yes. All right, so these are the sign convention. Next is just like in case of mirrors. Can I erase this part? Left side? Okay. So this is above is same as partition. Which you are already familiar, x-axis, y-axis. Right? Optical center of the lens. And position. The optical center of the lens at origin. Okay, can I erase the left side? Next is magnification lens formula. So like we had mirror formula, we also have lens formula. Lens formula gives what? The relationship between? Okay, correct. So it gives the relation Again, the derivation is not in the scope, just remember the formula. Between the focal length, the object distance, and the image distance for a given lens. Okay. Okay. How is it given as 1 upon 10 is equal to 1 upon E minus 1 upon. So how is it different than mirror formula? The plus is replaced with minus. Okay. So note, when you will use this formula for solving numericals, substitute all the given quantities with proper sign. Yes.
Sir, but after finding the U is given, then U is always negative, then it will be positive.
So we will see that. No, it is not positive. If it is positive, then if you write minus U, then it will become negative.
No, but minus U will be positive always.
Minus 1 by E of minus or minus U.
That's okay, but this is a formula. So you substitute U value with minus sign all the time. Okay?
After that it becomes plus, it doesn't matter.
No, it doesn't matter. After that, it will become plus.
Always.
Always. So for solving numericals, substitute all the given quantities with proper sign. And leave the unknown quantities as it. Don't bother about the signs of unknown quantity. That will be obtained by calculation. So that's about lens formula. Then we have magnification formula for lens. So what does the magnification formula gives? What relation between?
The height of the
So relation between image size and object size. It gives relation between object size and image size. What is the formula?
H1, N
H dash by H, मतलब H dash लिख दिया मैंने. V by, no, V by, minus is for mirror. Okay, so this is what you have to remember. And M is positive for which image? Virtual image. Erect. Which image is erect? Virtual. Virtual image. Concave में हमेशा it will be like that. Right. M is. Why I am saying image, because it is true for both mirror and lens case. M is negative for real image. So all these things will be very, very handy in deciding about the signs of the given physical quantity. Why the view is not. Okay. Finally, solving some numericals. Everybody has NCERT textbook. I'll be solving numerical example 10.3 on page 182. Just write down example 10.3, page 182 of NCERT. Yes, 182. Done, that's it. Left side, done.
Yes.
182, 182, yes. I'll write it on. Textbook page 182. Exercise 10.1. Okay, shall I read it? So it says a concave lens has a focal length of 15 centimeters. So what is given? So you have tried also the nature of lens. So it is concave lens. Okay. It has a focal length of 15 centimeters. So f is equal to so in question it is given 15 centimeters. So then you will have to write the sign for it because it is concave, so it should be minus. At what distance should the object from the lens be placed so that it forms an image at 10 centimeters from the lens? So u is equal to what? Now v is equal to 10 centimeters. Now which lens it is? Concave. So the image is always virtual. And in case of lens, the virtual image is in the front of the lens. Minus.
So front of the lens means right.
That is behind the lens.
Acha.
Okay, so light is incident on the front portion of the lens. So anything on the left is front. And concave is always virtual, so it has to be negative. Okay. In case of mirror, it will be behind the mirror because all the reflected rays will meet in front of the mirror. So only thing that will meet is only diverging reflected ray will meet behind the mirror. In this case, when the rays are refracted, so they are passing through the lens. Now when they divert, so only when they are extended backward, that means they will meet at the same side of the lens. Got it? So these logics you develop. So these signs we have put ourselves because we know that it is concave lens and we are familiar about the nature of image formation for concave lens. Okay. So this for solving numerical, substitute all the given quantities with proper sign. Okay. So U we are leaving as it is. Now here, you asked me a question, right? Now here, U is unknown. So in the formula, how can you leave the U as plus? See, every time U value is not given, na? And when you are solving the unknown quantities, you will leave. That manipulation has chance of error. Now this is standard process. So whenever you are substituting U, that time you substitute with minus. But when U itself has to be calculated, then there if you put plus, then the answer you would get would be wrong. Like that. So okay. So where will be the image? Also find the magnification produced by the lens. So also find M is equal to. Okay, so these things we have to find out. Understood the question? So what will you do now? Just substitute the values. So to first find U, so it is since 1 by F is equal to 1 by V minus 1 by U. So to bring it over here, so therefore 1 by U is equal to 1 by V minus 1 by F. So 1 by V is 1 by minus 10 minus 1 by minus 15. So rearrange, this will be plus 1 by 15 minus 1 by 10. So what it will be? So 15 into 10, 10 minus 15, right? Everybody familiar with how to solve fractions? So therefore 10 minus 15 is minus 1. Therefore 1 by U is equal to minus 5 upon 15 to 10. So therefore minus 30 something. Okay, so what is U? U is minus 30. Now F is minus 15. If U is minus 30, therefore U must be center of curvature. Then straight away you can say that magnification will be 1. Will it be 1? No. Oh, it is concave lens. Yeah. So for concave lens, we don't have this concept of C, right? So now what is the magnification? So this is one answer, right? U is equal to minus 7. Now M is equal to V by. So this is minus 30 divided by
Minus 10 divided.
U is minus 10. Minus 10 by.
Minus 12.
Okay. So you get 1/3, 1 by 3. So therefore, that what is M? H dash upon H is equal to 1 by 3. This means H dash is 1/3 of H. Okay, so you are getting a diminished image, and it is, since N is positive, it is a virtual image. Everything is very good. Okay?
Sir, is there any unit for magnification?
No, because yeah, it is length over length. So no unit. It's just a factor. Magnification is the factor by which image is either magnified or reduced with respect to object size.
And we can write in refraction form also.
Yeah. So
Or the proof of what it is.
it's okay. I mean, 1/3 means it is not equal, right? It is 1/3, 30%. How much it will be? 33.3. Exactly. So it is about 33.3%, or this is how much? 0.33. Okay. It's like
You can leave it in fractional.
you can leave it in fractional or put it in decimal form. It's okay. It's fine. Or write it in statement. So what is the statement? The
Minus of thickness is
Here at magnification, you can, if it is only M, then you can find out 0.33. So it is less than 1. Therefore, image is reduced. 1 image size is same as object size. What are you going to write the explanation of it?
The height of image, sorry, the height of image.
Don't say height, just say
The
size.
size of image is 0.33 times the size of
Object, exactly. That's right. And since it is positive, it is virtual and upright. Okay. So from the sign of the magnification also, you can figure out what is the nature of the image, right? V will give you its position. So here anyway, V was known. We have to find the object position. Clear? Question number 1. Have you noted it down? Noted it down? Right. Clear. So how are you finding this long lectures like this? Good use of time. What is it? Are you finding okay? Yeah. Okay. Okay. So try to cover as much as possible before another lockdown is declared. I don't think it will be declared. What do you think? Are the cases rising in India? What is it?
In China, it is rising to, I think the price.
Don't trust the Chinese. They are not to be trusted. Their economy is thriving. How can
You know, like, in their basis are increasing.
so? What is the basis for you to know that in China cases are increasing? It's the media, right? As simple as creating some scenes and posting on television to confuse other countries. So that rather than focusing on development, they become defensive. And change their strategy. Chinese cannot be trusted. In my feeling, it's actually Americans who use Chinese to basically spread rumors. So Chinese just got a bad name, but real culprits are Americans. But you agree? Every Indian wants to go to.
Weapons to Ukraine, but
Yeah, they have basically, yeah. I mean, Ukraine is fighting the war for US. That's what they're doing. They're minding shoulders, taking the front, face somebody else. We Indians, like, we take pride in going to America to, Americans can't run their own country. They need Indians and Chinese. Nobody.
Because most of the population is not Native Americans who stay there. Some is Indian people are there, plus Chinese.
The smart ones are expats, Indians and Chinese. The red Indians are the local Americans, the actual Americans. But they have been taken over by these Europeans, UK and all, right? So they spread out to Canada and all other countries. But the problem is they were bandits since the beginning. They looted all the other countries and accumulated the wealth, and now they're calling themselves the leaders of the world. Our own countrymen should take pride, and I have traveled to many parts of the world here. I don't find anything better than India. Indians are smart as always anyway. We Indians, we perform so well when we go out. Why can't we do wonders over here? Because we are led by some corrupt politicians.
they are very much development is
They are highly populated. So many people just infiltrate in our country, and we don't mind it because they vote for us. All those things. So our kids, our students should become aware. Apart from learning, okay, they should also take pride in our country, like American dream. Why can't it be Indian dream? If we don't think about our country, who will? You think they will come and think about us? If it is our house, we only have to keep it in order. So anyway, so we need to educate ourselves well and take pride in our country. There cannot be anything better than India for Indians. This is what I mean. That's why I came there. I did not stay there. That's my personal opinion, okay? It's not binding on anybody sitting in this room. Jamshed and I had a lot of discussion last year, and he seems to agree with me. Americans tried so much that India should, join them in fight against Russia. But we did not. We did not join either Ukraine or Russia. Go finish yourself.
India never joins any war also.
It is always pushed into it.
India even never starts a war.
Because we know what is the outcome. So we have so rich tradition, all our literature is so rich. Even people who came from outside, they have adapted, right? So, and the most peaceful country is India, and that's why they are jealous. And we are blessed by all kinds of weather, everything is here. Why should we? Anyway, priority optics. Next question. 10.4. So I guess this is clear to everyone. Next. So whenever you capture data, okay, attention everybody. Whenever you capture data, you also write what type of lens or mirror we are using. That will help you decide on the sign. Now, so next question. So a 2 centimeter tall object, right? So this is object size. So h is equal to 2 centimeter. Is placed perpendicular to the principal axis of a convex lens of focal length 10 centimeter. So it is convex lens. Focal length is 10 centimeter. So what it will be? Plus or minus? Plus. The distance of the object from the lens is 15 centimeter. So u is equal to 15 centimeter and it is always minus. Find the nature, position and size of the image. So you have to find basically B is equal to what? The what? And H dash is equal to, and of course will nature as well. Nature as well. So can you find out from the given data?
Yes.
Try. So I'm saying Yug Pandey. Is he a new student?
So he's now on and he used to come here, but from vacation he has been online because he's at KK23.
But has he attended any of my class before? You can you open your video? Just want to see you. I'm not able to. Just show me your face once and.
Sir, position, size of the and nature of image.
Where is he gone? He's just gone. Yeah, he's come back. Are you with us? You? We'll start video later. May I know why? You just wanted to see your face, man. I mean, I'm not able to recognize you. That's why. Ananya, I think last time I told her about its spread and all. I hope she did not take it too seriously. Huh? Is she absent because of this? She's also at native. So during vacation. So we are doing question 10.4, right? Hope you all are doing. So I just wanted to see your video, what you are doing. Anybody got the answer?
Almost 30.
Huh?
30
Plus or minus?
plus.
Correct. Plus 30. That means where will it be?
Right.
On the right side of the convex lens. So what is the nature?
Really
Real and inverted. Very good. And enlarged.
Beyond C.
And frame size of the object is minus.
So
Minus.
focus is this. So where is it? It is between C and M. So therefore it will be enlarged. Huh?
Beyond C.
Beyond C. Correct. So therefore it is.
The size is negative there.
Yes. Negative 2. Correct. So everybody got M equal to -2?
I am not getting
So, do we have to keep 2 times the size of object, or do we have to calculate the height of the image? Do we have to calculate the height of the image as -4?
So if you get M equal to -2,
Yes, sir.
then substitute here. So H anyway is 2. So -2 into 2 will be 4 centimeters. So it will be -4. So what it shows, that image is inverted. -4 means it is below the principal axis. Image is below the principal axis. Okay. Have I answered your question? Because you have to calculate H dash. Size was asked. So what is size? Since H dash upon H, right? M is equal to this. What is H dash? H dash is equal to M into H. You found M as -2. What was H?
Minus.
So when H dash is -4, that means the image is below the principal axis on the right side. And M is negative. So what is the nature? Real. So you can find out that the image is real by value of V, which is positive. So behind the lens is always real image. Okay, in case of lens. Right? In case of mirror, it is, yeah, watch it. Okay, so with this 2 examples, I think you must have got some idea about how to work with the signs of the given data, right? What data to capture, how to get the sign, and how to use it. Let us now move on to the last topic of this chapter, which is called power of lens. Is this topic taught at your schools?
Yes.
Huh?
No, sir.
Yes, no, yes, no.
We did not start.
Oh, very good. Okay. But you did not start anything in physics. In science?
Science, biology, chemistry,
Okay. So this is a very big chapter and it has got high weightage. Okay, so we have finished, almost finished it. And for you all, it is not yet started.
It is done till lens. The lenses part is just started and then.
So you have no difficulty in understanding now that we have covered it. Done, huh? Okay. So let me ask those who have done, what is power of lens? What do you mean by power of lens?
The unit was diopter.
I'm not asking what's the unit. What do you mean by power of lens? Which lens is more powerful?
Diameter or the thickness of the lens.
If the thickness of the lens is more, it is powerful. Answer is yes. But it still doesn't answer my question. What is power of lens? The lens which is thicker has high power, but what is high power? When do we say lens has high power?
So, I think power is the inverse of focal length.
That is again definition. So what does it mean?
So, a higher power.
So more thicker than Only more refraction.
Bending. The ability of the lens to bend the light. Okay, either bending is both, either converging or diverging. If it is able to converge or diverge the incident ray more, then it is more powerful. Less bending, less power. Okay. So what is the power of the lens? In layman terms is this. Power of lens. It is the ability of the lens to bend, that is converge or diverge. The incident rays. So more the bending, more the power of lens. Right? So obviously, so suppose I give you these two examples. Right. So which lens is bending the light more? Second one. So second one is more powerful, right? So when the parallel rays are bent, they meet at a point that we call focus. So the focal length is smaller for a powerful lens, right? So power is therefore inversely proportional to the focal length. That's how you get that difference, right? Therefore, P is equal to reciprocal of focal length. This is the definition. Okay. So from this diagram, you can see that power is inversely proportional to focal length. Here the bending is less, so we are getting large focal. So power L of lens is defined as reciprocal of focal length. What is unit of power? Yeah. SI unit of power is diopter. When the focal length is expressed in in meter. Okay. So unit of power is diopter. It is written as capital D. So therefore, what is one diopter? 1D is equal to 1 by 1 meter. Right? Focal length is expressed as meter. Got it? about sign of power of lens? What is the sign of power of lens? Plus, minus. It will draw from the focal concave, right? So whatever is the sign of the focal length will be the sign of the power, right? So power is negative for diverging lens, diverging or concave lens, and is positive for converging or convex lens. The same as the sign of the focal length of the respective lenses. Okay. So if there are multiple lenses with their powers given, then the combined power will be their sum, right? So power of multiple lenses. Okay. So net power you can say of multiple lenses is sum of, is actually algebraic sum because it may have sign. It's algebraic sum of powers of individual lenses. So suppose you have a system of lenses. So one convex, one concave, another convex, right? So you'll have plus P1, minus P2, plus P3. Then what is the net power? P is equal to P1 plus P2 plus P, whatever. With power signs. Okay, so so I have a habit of keeping and making mistake with lens, right? This E is not there. Yes. Yes. So this is a funny language. Don't know what it's called. Everybody done great. Can I raise the left side? So there are 3 in-text questions in NCERT for power. So on page 184. In-text questions. Page textbook. Page 184. Question 1, it says, define one diopter of power of lens. Yes. How will you define it?
It is one diopter is the power of a lens with focal length 1 meter.
Exactly. That's it. Just discuss it. Rightly answer yourself. One diopter,
So
one diopter, one D. Diopter, see the spelling, D-I-O-P-T-R or D-E-R. T-R-E, correct. One diopter, power of lens is when the focal length of the lens is 1 meter.
lenses.
Is when the focal length of the lens is 1 meter. Or when the given of the lens, one diopter of power of lens is of focal length 1 meter. Whatever, correct English. Okay. So you understand. So now second question. Second question is, can we discuss the second question? It says a convex lens forms a real and inverted image of a needle at a distance of 50 centimeter from it. Right? So you have a convex lens. What is given? It is a convex lens. Forms a real and inverted image. Real and so all these things you write inverted image. Right? Therefore, where will be the position? Behind or front? Convex lens. Real will be behind. After refraction, they should meet. So they will meet behind only.
Towards the right.
Yes, towards the right. That's why I said V should be +2. So where is the needle placed in front of the convex lens if the image is equal to the size of the object? So H dash is equal to H. This is what is given. That is, and V. So this is M should be equal to -1. Correct? Real. Because only for virtual M is positive. Since H dash is equal to H and it is inverted, so M should be -1.
And then should be like minus H dash is equal to H.
It is the magnitude is equal to H. The size of object is equal to size, but H dash is inverted, no, real and inverted. So that's why minus. Okay.
What is asked?
What is asked, right? So what is asked? So find the power of the lens. So find P. Right? Now to find P, we need to find focal length. So how do we find the focal length from the given data? Now,
From magnitude, from
And -1 is equal to
magnification, we get zoom, and then we do that lens formula, where we get F.
so yes, that is a method, but that will be a long mathematical method. But here it says the image size is equal to object size. When it happens, when it is placed at center of curvature, which is double the focal length. So V is +50, that means R is 50. Object and image both form at C when the size is same. So which means if the object is at C, so from C you can find focal length, which is half. So focal length and it is convex lens, so it should be +25. Got it?
power is 1/.
Yes. No, 25 is centimeter. Convert centimeter in meter. Because power has to be diameter.
What I.
Point 0.25 100 by 25, 4 diameter, right? Plus 4 diameter. Got it? Everybody got it? Junction views look little bit confusing. See what it says. It says the object size is same as image size. So in convex lens, for what object position you get same size? See, there are 6 options, right? For convex lens image formation, there are 6 options. Object at infinity, then the image is at focus. Object beyond C, images between C and F, and diminish. At 2F, right? So when the object is at 2F, what is 2F? C. Only then the image size is same. So now we are saying we got the image is at 50. So this image must be at C, no? So what is F? For power, I want to calculate F. So I get from this data that H dash is equal to H and V. I come to know that object must be placed at C. So F is equal to half of, yes, exactly. Got it? So from given data, so see, this is how the image formation knowledge helps us. Sometimes you can avoid mathematics, right? From given data, F is equal to plus 25 centimeters. Okay, I will explain how we got 25. Therefore, which is equal to 0.25 meter. Therefore, P is equal to 1 by F. 1 by 0.25 is equal to 100 upon 25 equal to 4. So this is plus 4. P is equal to plus 4. Everybody else. Last question. Find the power of a concave lens of focal length 2 meters. 2 meters, yes. Question 3. E is equal to what?
It was which lens?
Concave.
Minus 0.
Yes, now it is correct. Minus 0.5. Okay? Yeah, so power you have to always provide with. So that what is key?
Minus 0.5.
0.5. Okay? So that's why knowledge of lens will help you get the correct sign. Okay. When is the break?
2 o'clock.
2 o'clock. It's a 2-hour lecture.
1:30 to 2.
12:32. That's okay. Chalo, so we'll solve now numericals. Everybody has volumes. Page number. So we are going to solve numericals. So online students, there is a chat. Yug Pandey has a network problem. Okay, fine. No issues. So Ananya, Arya, right? Who is that? Arya or Ananya? Okay, Arya. So we are going to solve numericals now. Right? So open your volume, page 8. Okay, open page number 48. We're starting with MCQs, multiple choice questions. Can you see it? This is page number 48. Okay, so is everybody with me? So how I will solve it is each one of you will take turn in reading the question and answering. Okay. So first, start with you.
Which one of the following materials cannot be used to make a lens? D.
B, which is clay. Why?
Because they cannot.
It is not transparent. For lens we need transparent medium, right? So, clay is opaque. So the correct answer is for first question, the correct answer is D. Second question, Jamshed.
Yes, sir. The image formed by a concave mirror is observed to be virtual, direct, and larger than object. What should be the position of an object?
So what is it?
Larger.
Concave mirror.
Concave mirror.
It is forming virtual image which is magnified. So what is it?
Focus and pull.
Correct. So what is the correct option? D. So everybody just take your names also so that I will remember. What's the name? Akshar. Yeah. Read question 3.
Where should an object be placed so that a real and inverted image of same size is obtained by a convex lens and twice the focus length?
Yes. Just now discussed, right? Your name?
So, Aditya.
Aditya. 1 second. You said Akshar. Akshar. Aditya. Jamshed. You? Ayush. I will again forget, but don't mind. I'll keep asking. Don't be irritated. Okay.
A spherical mirror and a spherical lens each have a focal length of -15 centimeter. In here, the mirror will be concave, but the lens will be convex. See.
Convex lens has positive focal length or negative? I showed the diagram. Today that's the first thing we discussed.
Sorry, sir. D will be.
What is D?
Mirror will be convex and lens will be concave.
Convex mirror has positive focal length or negative?
So it has
The parallel ray which is incident on convex mirror. Where is it?
so it would be diverging.
Diverted away from focus. So the diverted ray is always extended backward or forward.
So it will be extended backwards.
Convex is positive.
Yes, sir. So both will be same only.
You will now get it. Okay, so this was a good question, right? So online folks, we are discussing question 4 and the correct answer is C. Sorry, both are concave. A. Correct answer is A for question 4. Give me a thumbs up, online folks, if you have heard the question. Yes, you guys given me the thumbs up. I don't know about Arya. Arya is eating and sleeping. Okay. So question 4, correct answer is A. Was that a tricky question? Yes, that was a tricky question, right? 5.
So my name is Shraddha.
Shraddha, okay. Go ahead.
No matter how far you stand from a mirror, your image appears direct. The mirror may be
So,
either plane or convex.
yes, very good. Okay, so the image is always erect. We are not talking about size. So erect is in case of plane or convex. Question 5, correct answer is D. Question 6. Ayush.
Which of the following lenses would you prefer to use while reading small letters found in the dictionary?
Yes. Right.
Convex.
When you say pass, then only.
Convex lens of focal length, right?
Very good. Excellent. Why?
So because like
Because if you're reading, you would prefer to read straight. So the image should be magnified as well as straight. So that has to be virtual. So it is only possible when the object is within focus and optical center. So it is better that it is nearer. You want concave lens, it will be diminished, na? Would you prefer that already small letter, you would prefer it to get reduced or magnified? Got it?
So,
What is it?
C.
C, convex lens of focal length 5. Question 6, the correct option is C. You see, these are confusing questions, right? But you have to read it twice slowly, then it will settle down. Okay. Ramshay.
We wish to obtain a direct image of an object using a concave mirror of focal length 50 cm. What should be the range of distance of the object from the mirror? What is the nature of the image? Is the image larger or smaller than the object? Draw an ray diagram to show the image formation in this case.
What should be the object position? It is a concave mirror, right?
Between F and P.
Yes, between F and P. So ray diagram you have already drawn, right? And what about the image? Will it be magnified? It will be magnified. It will form behind the mirror. It will be magnified. It will form behind the mirror. So virtual, right? So the answer for question 7 is the range will be between 0 to 15 centimeters in front of the lens, in front of the mirror. 0 to 15, right? But not at 15. At 15, so it will go to infinity. Zero. Object should not be at focus, just next to focus. Between pole and you say between pole and so between 0 and 15. That's the correct answer. Between 0 and 15 in front of the mirror. Okay, and the second part is the image will be larger or smaller? Larger. Draw a ray diagram. I hope you can draw the ray diagram. You have practiced enough number of. Question 8.
Name the type of mirror used in the following situation. Headlight of a car, concave.
Yes, why?
Because to get full range of light from
Parallel beams, powerful parallel beams, correct.
right rear view mirror of a vehicle, which is convex.
Yes,
So
why?
that virtual interior may appear and facilitates safety.
Yes, and wider viewing angle.
Wider.
Right.
And solar furnaces concave.
Okay, because it will concentrate
Because to
the
focus the solar heat to generate
at focus. Yes.
Suppose to get a parallel beam of light.
No, it's the region, like A the region here. Support your answer with region. So for A, it is A, right? For B, that's the region. For C, C is the region. They have given the answer in the question itself. Like the reason they have given already. So I think we have discussed A. Any issues, online students? No issues. No response, no issues. Okay, we proceed.
One half of a convex lens is covered with black paper. Will this lens produce a complete image of the object? verify your answer experiment in your observation.
It will not.
It depends on which half we are.
So we have how many eyes? So if I close one eye, will you see only half the students or all?
So you can.
Like you are sitting here, I am saying 1, 2, 3, 4, 5. Still saying 5. No, so what? Everybody is visible to me. Come here, you don't trust me? I see everybody. He is wearing black t-shirt. He has kept his hand like this, head resting on his hand. What should be the answer? Full image will form or not?
No, sir, I think so. Not image will not form because there will be two layers need to join.
Okay.
For forming an image. So if half is covered with black, then how two layers will transform.
So how many tubelights are here? 3. I'll go on switching off one by one. What difference you will see in the image quality? Only the image quality will become poor. It will be poor. So what should happen here?
Image will be formed with quality and
The full image will be boxy. What we have reduced by covering up? We have only reduced the aperture. So aperture is from full, it has become half. So the optical information is reduced. Firstly, full optical info was passing through the lens. Now we close the gate half. So only half the light is passing. So the image will look faint. That's what will happen. Full image will be formed. Now you trust or not? Let's see the experiment. How the ray diagrams will take place. So this is question number
9.
So you have a convex lens, right? Let us say for simplicity we are keeping it at C. Now this portion is covered. It is now opaque, right? So from this part, we used to take 2 rays, right?
Yes.
Both are blocked. But are only 2 rays coming from this part? Infinite rays are coming. Just for simplicity and convenience, we were taking 2 rays. So now take this. Let us say you have focus here. Take 1 ray from the focus. Where will it go? It will go straight. I'll take another one. Right? So where it will go? It will bend here. This is a refracting medium, right? So what about this? Everything will go there. So what has happened is the 50% of the optical information is blocked. So from the bottom half of the lens, even this top point is visible. So these points, all the rays emanating from here will be reconstructed. So the full object will be, full image will be reconstructed. So understood, image will form, but it will be, what's the question number? 10.
9.
Image will form in this case, but will be faint. Okay. Next question.
An object 5 centimeters in height is held 25 centimeters away from a converging lens of focal length 10 centimeters. Draw the ray diagram and find the position, size, and nature of the image formed. Hint. To draw the diagram, consider a suitable scale for the distances. Example 5 centimeters equals 1 meter.
So this ray diagram you have to draw to the scale. So when you draw the principal axis, mark it. So 0 will be at the optical center, then mark 1 centimeter, 2 centimeter, 3 centimeter. Each centimeter is 5 centimeters. So what is the unit? 1 centimeter is equal to, that is 1 unit is equal to 5 centimeters. Then place the object. The object size it says is 5 centimeters. That means in your drawing it will appear only 1 centimeter. Okay. So where is the object kept?
Beyond C.
It's 25 centimeter away. So let's keep the marking. Right. So there will be 5 marks after optical center on the left side. So it will be kept on the 5th mark. That will make 25 centimeter from optical center.
It will be between C and F, and it will be
The object is beyond C, so image will form behind the lens between C and F. Yeah. So it will be real, inverted, and diminished.
less than
Less than.
5.
Yeah. So you have to find it out not mathematically with ray diagram. Do it. With measurement. So when you measure with the scale, the image size should be somewhere 3.3 something. Break and do this for half an hour. Right? Okay, it's a lunch break.
So I want images diminished. Then
What is the size of image?
small, sir.
You measure scales. Where are your units? Read the question. See, you have to make it like this. This is question number 10.
Yes, sir.
So you have which lens?
Convex.
Convex lens, right? So what is the scale? Scale
10 centimeter.
1 unit
5 centimeter.
is equal to 5 centimeters. Let us say this is my 1, 2, 3, 4, 5. Like this, I marked the principal axis on both sides. Okay. Now, where is the object placed?
20 minus 20, 25 centimeters.
So 1, 2, 3, 4, 5.
So 4.
And it is also 5 centimeters. So 1 centimeter in height. Okay. And at 25. This is at 20. Right? Yeah. Yeah, it is -5. So now you take the parallel bit. It should converge at focus.
Which is 10 centimeters.
Focal length is given.
Yes, 10 centimeters.
Is it focal length?
Yes,
Focal length is
focal length is 10.
Plus 10 centimeters. So less than this. So if this is focal length, then this should be twice of f, right? Now, the other way you take from O. Right? So where is it making? It is making here. So this is 5, 10, 15. And this is 20. So this should be somewhere 16 or 17. What is the image position?
Between 16 and 17.
So B should be between 16 and 17. Right? And what is the image size?
So I'm getting a 2.5.
2.5 what?
Height of the
2.5 what?
minus
2.5 centimeter.
2.
Then it is magnified. This is only 1 centimeter. Acha, this is 5 centimeter.
5, that is
2.5. Okay. This should be okay. Let me see what is the answer. Answer is on page 70. Just so it is question. Yeah. So the image distance is 16.7, which is correct, right? After 5 it is.
Yes.
So when you measure it with scale, you will exactly get it. Okay. And it is 3.3, not 2.5, 3.3. So see, mine is more appropriate. You see, it is almost quarter to 1 centimeter. So if you are drawing it with scale with proper marking, you will get the answer. When you measure this size, you will get it 3.3 in units of 1. What it will be? What is 1 millimeter? So 5 centimeter. 1 centimeter is equal to 1 centimeter corresponds to 5 centimeters. Right? Like 10 divisions. 1 centimeter is how many? 10 divisions, right? So therefore 0.5. Each division is 0.5. So when you measure with the scale, whatever division you get, multiply that by 0.5. Right? So 1 millimeter on scale is how much? 0.5 centimeter. So measure how much is this? It will be less than 10 divisions. Are you understanding what I'm trying to say?
Yes.
So here this is 0, right? So this is 5 and there is the object. These are all 1 centimeter.
Yeah.
Right? 1, 2, 3, 1. This is your object. So not parallel. There is a focus 10 centimeter. So you're not writing given in all that. So you write on that figure. So now this will be first this one focus. Focus is 10 centimeter, you are told, right? Now where will it meet from this point? right?
Yeah.
That's why you should use pencil. Already used this. Understand? One, two, four, five. Right? So something wrong. No, yes. 8, right? So 8 into 0.5. Because one division is 0.5 centimeters.
Yes, now 3.5.
Yeah.
So I got 7 millimeters.
7
millimeters.
So 1 millimeter is 0.5 centimeter.
I also got
7
7
minus 3.5.
Right? So 7 into 0.5. This is almost 3.5. And in book it is given 3 points. So mathematically, you are very clear. You understand? So if you draw appropriate diagram, you cannot go wrong. So in this question, we basically found an alternate method to obtain the object size, image size. Now this we obtained geometrically or graphically, and mathematically obtained using magnification formula and lens formula. Got it? So this was a very good question. Oh, yeah. Anyway, 5 minutes में तो break ही हो रहा है. ठीक है. Okay. So how are you finding these questions? Interesting? So the more you solve, more will become familiar. This was question number 10, right? Whose turn now? Question 11.
A concave lens of focal length 15 centimeter forms an image of 10 centimeter from the lens. How far is the object placed from the lens?
It should be easy, right? So here you can find using lens formula. So it is a concave lens. So I think this is the first question we did today, right?
Yes.
Just check. This is same as NCERT.
The concave lens f is equal to minus 15 centimeter.
And image.
Image is minus 10.
Minus 10.
So it is the same question, right? So it's already done. So question 12, yeah, come. Question 12 we have already done.
So question 11 is already done.
Oh, sorry. Question 11 is already done. Now next. So you proceed with next question. Question 12.
An object is placed at a distance of 10 centimeter from a convex sphere from focal length 15 centimeter. In the position and region of the image.
Yeah. So I think this should be easy. Using lens formula, you should be able to find out. Next question, question 13.
Magnification field by obtained mirror is +1. What does it mean?
Yes. Magnification is positive. So positive sign tells you the nature of the image. Is it virtual or real?
Real.
Magnification positive is not real. Magnification positive is virtual. Yeah. So what is the nature of the image? Virtual. And it is same size. So what does this tell you? That it must be a plane mirror. Right?
It is given that it is
So obviously, so it is virtual image and upright.
Upright.
And same size. Object size and image size are same. This is what it means. When the magnification is 1. Because h dash upon h is equal to 1. So virtual image of same size as object. Has everybody written it? Okay, so what does this mean? These are all like tricky questions and generally asked in exams. Okay. What about question 14?
An object of size 7 centimeter is placed at 27 centimeter in front of a concave mirror of open length 18 centimeter. At what distance from mirror should a screen be placed so that a sharp focused image can be obtained? Find the size and nature of it.
So I think it's also break time. So we'll tackle it after the break. Okay, and we'll take up in the remaining time more questions. So we are going to solve numericals only in this class, next class, maybe next class. Okay.
समझ जाएगा कुछ
बस में उसको हल्का पीटेंगे 10-12 मिनट से
नहीं मैं बोल रहा हूँ अभी जो भी ब्रेकफास्ट है वो खा ले फिर हम आराम से जाएंगे तो सो जाएंगे जाकर फिर हम चिल्लाएंगे मैंने जो जब कर लिया वो नहीं खाता है फिर हमें बोलेंगे नहीं मम्मी बनाती है सुबह बोलता हूँ भेज दिया अच्छा से एक वीडियो देखा नहीं है कौन सा उसने कहा
कितना टाइम लगेगा वीडियो देखने में
बोला ना That's the law.
तुम लोगों का खाना जंक्शन हमारा मैथ्स पेपर तुमने दिया था, क्या प्रॉब्लम था तुमको? हाँ, क्या प्रॉब्लम था? हाँ, श्रद्धा, एक्सीलेंट। अक्षता तुमने भी दिया है? तुम लोगों ने टाइम से दिया था?
दिमाग क्या नहीं खेलने वाला, ऊपर ही नीचे।
ये प्रोफेसर से मार्क
मिला किधर दिख रहा है? अच्छा ये performance not satisfactory can be better. Who be number 15 is not satisfactory.
नहीं वो भी वैसे ठीक है, वो भी मैं कैलकुलेट प्रेजेंटेशन करा था, आंसर
Last color.
जो चेंज हो गया ऐसे ही करना था
मेरा 1 मार्क कट गया है सिर्फ 4 मिला
किसी और में भी
दूसरा क्वेश्चन
ओके
एक मार्क
तो सबको मिला के 3 था
हाँ
कोई फर्क नहीं
3 plus 2 is 5. 5.
Yeah.
उसमें कितने लोग मुझे 69.73
कौन थी बच्चों का मिला नहीं था एक मिनट डबल ए ना
यस
आयुष
मैम इसमें एक पे मिला है मार्क्स
आया
हाँ
that means I have to receive the paper. I am not receiving. और कौन अक्षत का मिला?
Now, Aditya, I got my marks on but I didn't.
This is double A or single A?
Single.
Aditya Biswas, na? You got your marks?
Yes.
Okay, let me check. Only 2, na?
Is it time?
yes
Yes.
Okay, so let's start again. Which question we were on? 10.
14
14. Okay, so question number 14 on page 49, right? So let me do it on the board because otherwise online students will not see it. You all can try in your notebooks. Online students also, please try yourself. Question 14. So you have what is given. So it is UT volume, page 49. Question 40. So given object size of 7 centimeters, that means H is equal to 7 centimeters. Placed at 27 centimeter in front of concave mirror. So this is concave mirror. Right, and 27 centimeter in front, U is equal to minus 27 centimeter. Focal length is 18 centimeter. So F should be equal to, it is concave, so it should be minus 18 centimeter. Right, for concave mirror or concave lens, focal length is negative.
B is minus 54.
So at what distance from the mirror should the screen be placed? That means screen will be placed at position of image. So we have to find the size and nature of image. So we have to find S dash, and we have to also find nature of image. Okay, so this shouldn't be difficult. We have already solved many questions of this type. So to find V, we will use mirror formula. The solution
Minus 50.
by mirror formula. One upon F is equal to 1 upon V plus 1 upon F. Okay. Mirror formula you have plus. Now, even without solving, where is the image expected? It is a concave mirror. So for concave mirror, an object is at minus 27 centimeter. Focus is 18. So where is the object? Object is between C and F because 2 times F is 36.
We want to see here what can match.
Right. Very good. Okay. So this is how you can figure out, and then you can tell your answer. Right. So image should be beyond C, which means image should be greater than 36. Then your answer is correct, and it should be real and inverted and enlarged. So it should be plus. B should be plus. Okay. So let's solve that point out. So 1 by V is equal to 1 by F minus 1 by E.
Sir, plus from here.
So mirror में plus ही है ना?
Yes.
Now 1 by V. So this is minus 1 upon 18 minus 1 upon U is minus 27. So now therefore 1 upon V rearranging 1 by 27 minus 1 upon 18. So the way I do this, 1 by V is equal to 18 minus 27 divided by 27 into 18. So 1 by V is equal to, so what is common? 3 is common, right? Or 9 is common. So 9 2s are 18, 9 3s are 27 divided by 27 into 18. So I would say 9 3s are 27.
Minus 1.
So this is E is equal to 1 by E is equal to minus 1 by Yeah. So 1 by d is equal to -1 upon 54. Therefore, d is equal to -54.
Yes.
Yeah, so it is real image. So we should be in front of it. It is mirror, right? Okay, so not lens, it is mirror. So this is correct. Therefore, real inverted and enlarged image and enlarged.
2 times image.
Enlarged image.
So do we have to calculate here or do we just have to let it go?
What? We have to also calculate, huh? We have to find the size. So right now, write real and inverted because it is in front of the mirror. Real inverted. Real and inverted. In front of the mirror. In front of the mirror. Okay. Now to find the size, we'll find the magnification.
And equal to -2 by u.
So now, further, m is equal to h dash on h equal to e by u minus. This is mirror. Okay, so what is the size? Therefore, h dash is equal to minus h into b by. Find out what is it? Minus. X given is 7. B is we calculated -54. U is -27. So -27 2 times will be
3.
this is 2. This is -40. Okay, so the size of the image is -14 centimeter. What was the size of the object? H. So the image is 2 times the object size. Therefore, what is M? Therefore, M is equal to H dash upon H to -14 upon 7 is equal to -4. Okay, so what answer will you write? The answer, therefore,
Size of image
size of image is
2 times size of object.
Is twice the size of object. And the image is real because M is negative. So it is doubly verified. Negative M or V. So V in front of the mirror has to be real. Because for virtual image, it has to be obtained behind the mirror. Okay, this one.
So if we are writing that it's twice the size, then why did we do that step?
What?
Why did we figure out what each size is?
H into v by u. You could have just written -2 by u and that will be 2 and then h1 by h equal to u.
So anything you can do. I mean, the sequence does not matter. So from here also, since h dash is 14, I can say image is twice. So M also I have separately obtained. That's okay. First I obtained the size of the image, then I obtained M. You could have done the other way around. It does not matter. So the answer will not differ. Just the sequence. So you can do whatever you feel like. Okay, it does not matter. So this is clear. So everything boils down to capturing the data with proper sign correctly. Okay, if your data here is wrong, then your answers are going to be wrong. Whose turn? I think that is.
Find the focal length of a lens of power minus 2.0 diopter. What type of lens is
2.0?
diopter? Sorry, diopter.
Diopter. After lunch, I thought, what happened? What did you eat? Okay. Diopter. So this could be confusing to some players. Diopter. So it is normal. I still, if you ask me your names, I will model it. So, yeah, go ahead.
Yes, sir. As this is a negative power,
Yeah.
this lens would be a concave.
Correct. So the lens type is concave because the power is negative. Okay, so we are discussing question 15. Since the power is -2, so negative power means it is concave lens. And what is the focal length? 1 by -2. So minus 0.5 meter. Focal length will be minus 0.5 meter, right? So that is the question 15 here. Yeah, is equal to 1 by E. That is 1 upon -2. This will be minus 0.5. So negative means therefore negative F means concave lens. Okay, so you have to write the full description. Next. We are discussing question 16 now. Question number 16.
A doctor has prescribed the corrective lens of power plus 1.5 diopter. Find the focal length of the lens. Is the prescribed lens diverging or converging? So first, since the power is positive, so the lens is converging or convex lens.
Correct.
And the power will be equal to 1.5 is equal to 1 by f. The answer will be 1 upon 1.5.
Yes. So focal length is 1 upon 1.5, which is 10 by 15, which is 2 by 3.
Yes.
Okay. So which is 0.66 something meter. 0.67 meter. That will. So question number 16. The focal length will be plus 0.67 meter, and since it is positive, the lens is converging or convex lens. Next. Ayush.
An object 0.5 centimeter in length is placed at a distance of 20 centimeters in front of a convex radius of curvature 30 centimeters. Find the position of the mirror, its nature and size.
Okay, so I think this one should be easy, right? So question 17, page number 49 of UT volume. So given object size, that is H, is 0.5 centimeter. Given U is equal to minus 20 centimeter. Right? Given radius of curvature is plus 30 centimeter, because this is a convex mirror. So find the position of the image, its nature and size. Do it. So this will be question 17. Given there is a convex mirror. object size is 0.5, U is equal to minus 20 centimeter because U is always negative. Now radius of curvature for convex mirror is given and it should be positive. For convex it is positive, right? So therefore what will be F? Yeah. It should be plus 50. Okay, so sometimes focal length is not given, radius of curvature is given, so you have to find the focal length. So find the position of the image. So B is equal to what? Nature and size. You have to find the size of image. H dash is equal to what? And nature. So all these things you have to find. So it should be easy.
Yes.
So since it is a mirror problem, you have to use yes 1 by B plus 1 by U. Now I have to find B. Therefore, 1 by B is equal to 1 by F minus 1 by. Now substitute. So we have 1 upon B. U is 1 upon minus 20. Sorry, this is 1 by 20. Okay, so 1 upon 15 plus 1 upon 20. That's 1 by 8. Into 20 plus 15. So what is common in this? 5. Therefore, E is equal to
7 by 6.
yeah. So 5 7s are 35. 5 is a 38. So this is 60 upon 7. So how much is this?
8.
So 8, 7, 8 is
56.
4, 40.
5.
Yeah. And again 50, 7, 7 is 40. So 8.57 centimeter and it is positive. So positive is image is taking place behind the mirror. So it should be virtual, right? So let us see. And it should be diminished because for convex mirror all the images should be diminished. That we have learned in theory, right? So this is one answer. So we found the image position. Now M is equal to H dash upon H is equal to minus V upon U. This is for mirror. So therefore H dash is equal to minus H into V upon U. So this is equal to minus, what is H? 0.5. V is, we obtained 60 by 7, I am putting 60 by 7. And U was 1 by minus 5. I have taken in the fraction form for easy calculation. Okay. So I could have taken V as 8.57 also, but for easy calculation, I have kept it like that. So this is equal to, this decimal will be cut with, okay. So this minus will cut with this minus, 20 will cut with 3. Okay. So this is 1.5 upon 7. This is 0.2. 7 to the 14, then 0.21. Okay, and then one more. 14, 10, then 3. 3, 7, 4 is 28. So it is H dash is equal to 0.214 centimeter. So image is reduced by almost half, more than half. Almost half, right? So that's it anyway. So you have to find this. And H is positive. So nature is virtual. Since the image is behind the mirror, from this itself you can give the nature. B is behind the mirror. So for convex mirror, so nature is virtual. Because image is plus, H dash is positive, right? So nature is virtual. As virtual and direct. Image nature. Nature of image. I hope nobody should have any doubts in this. So we have completed all the multiple choice questions. Right. So I suggest that you should also try NCRT questions apart from questions in the volume that we are attempting. Next, let us go to review questions on page number 50. Okay. So here we will discuss selectively. So what is refraction? What causes refraction? Any challenge in this? Right? It's a theory question already covered in theory. What happens to light on striking a transparent medium? Refraction. Whenever light enters a transparent medium, it refracts, partially reflect and partially transmitted. So major portion is refracted. The one which transmits suffers change of direction. What is it called? Refraction. Okay. State Snell's law.
Ratio of sine of angle of incidence to sine of angle of reflection is a constant.
That constant is called as refractive index of the second medium with respect to first medium. What is meant by absolute refractive index of the medium?
The refractive index of the medium with respect to air or vacuum.
Yes, very good. When the first medium is air or vacuum. Okay. So we just discussed question 4. Absolute refractive index is the refractive index of the medium with respect to air or vacuum. Okay. What kind of lens is used by watchmakers and why?
Convex for doing large image.
Yes. Yes. So they have to keep the lens close, right? So it will act like a magnifying glass. Where will be the watch kept? Watch will be kept between focus and O, right? Watch should be kept between the focus and optical center of the lens, right? It should be kept very close. Very good. So why is? Because virtual and magnified image will be formed. Right? So virtual image is always upright. So it will be easy to comprehend the components of the watch. What kind of lenses produce both real and virtual images? What kind of lenses produce both real and Virtual images.
Convex.
Only convex, because concave produce only virtual. Okay, understood? So it is only convex lens. Why you don't agree? Agree, na? So read the question once again. What kind of lenses produce both real and virtual image? It's only convex. Because in concave lens you only have real, only have virtual image, not real. Question 7. How do the frequency and wavelength change when light passes from an optically rarer to optically denser medium? So let's discuss one quantity by one quantity. Will there be any change in frequency whenever the light passes from one medium to another?
Yes.
No. No. Read the question, question 7.
How do the frequency and wavelength change when light passes from optically rarer to denser?
So optically rarer to denser, the speed will reduce. Which means what? Light will cover less distance in a given unit time. So wavelength will reduce, but not frequency. The cycles will be same, just with reduced wavelength. So frequency does not change because it depends on the source of the light. Okay. So the answer to question 7 is frequency will remain constant and it only depends on the source of the light, but the wavelength will reduce because in denser medium light will travel with smaller speed. Yes, and it will bend towards the normal. So they are not asking about normal. How do the frequency and wavelength change? So wavelength will reduce because the speed is reducing in the denser medium. Okay. Question number 8. For the same angle of incidence, the angles of refraction in 3 media A, B, C are 15 degree, 25 degree and 35. In which medium is the velocity of light minimum? Where the angle is? Least means it is closest to normal. Maximum bending has taken place. So its refractive index is maximum. See, when sin r is small, that's when refractive index is high. So high refractive index means speed of light will be reduced in that medium. Okay, so what was the question? Correct. So it will be A, right? Yeah, that's all. So it was minimum. Because angle of refraction is smallest in that, so it is closest to normal. Yeah, okay. Draw the reflected ray in the given diagram. So see the diagram. How it will be? You have to. It is a reflecting mirror, or to confuse you, the mirror is placed. They place the mirror like this, and you are provided with focus, right? Now you are told that light is incident from this direction. So it will be diverted away from the focus. This is the diagram. Okay. Right? Where is your volume? You have the volume? Question 11. No, question 10. Is magnification positive, negative in the case of concave lens? Concave lens. What kind of image is formed in concave lens? So for virtual, what is magnification?
So magnification.
Positive. Yeah. Because for
How many for positive? How many?
concave.
See, this is the mistake which happens.
I
So
got confused with that.
I know. So that is why we need to solve as many questions. Okay. Now concave lens will always form virtual and upright image. So how can M be negative? Right? Object is placed above principal axis and virtual image is upright. So both are positive. Right? Okay. So answer questions like this. Okay. Question 10. Hope everyone got it. Online students. Question 10. Is magnification positive or negative in case of concave lens? So concave lens forms virtual image. Therefore, magnification will be positive. Okay. From the given information, answer the following. So you are provided with 3 transparent mediums, ice, benzene and ruby. So refractive index given is 1.31, 1.5, 1.71, right? So refractive indexes are increasing. So in which of the 3 media does light travel fastest? Ice, the least refractive index. Okay, so it will bend the light little. So it is the least denser medium. Ice is the least dense because its refractive index is least among the given substances. So the answer for A is ice because its refractive index is least. Therefore, it will offer minimum obstruction to the speed of light. Okay. And therefore, it will be less, it will bend less towards the norm in case of ice, more in benzene and most in case of ruby. The bending will be most in case of ruby. Achha, in which of these media does light travels slowest? It will be ruby. Ruby. Okay, everybody got it? Respond. Everybody got it? Okay. Right. So answer for question 11B is ruby. Okay. Because its refractive index is maximum, therefore light will travel lowest. Okay. Draw a ray diagram to show the position, nature and size of the image found by convex lens when the object is closer to the lens. Between F and O, right? So you have to draw the ray diagram when the object is placed between F and O, and we have already covered it in theory. Question 13. I wish to obtain a real and inverted image of the same size as that of the object by a lens of focal length 20 cm. Where should the object be placed and which lens do I use? Yes. Yes. So since it is real image, I will use convex lens and I will place the object at center of curvature. Okay, then the image obtained will be of same size. So I will place the object at 40 centimeter in front of the lens, right? Okay, so we discussed question 13. Now question 14. Show the path of a ray of light through a rectangular glass block and mark angles of incidence, emergence and lateral displacement of the ray. Okay, so this we have already covered in theory. We have drawn and covered in theory. Question 15. State the factors which determine lateral displacement of a ray of light passing through a glass slab. So, what will determine? Refractive index. Yes, it will be. Correct. So, that will be the factor, right? And it will also depend on the color of light.
In human eye, we study that least
Right. Okay. So it will depend on two things: the refractive index of the medium and the color of the light. Okay. So what happens is So which ray bends the most? So violet. Violet bends the most. Yes. And red bends the least.
It is also in form of theory.
So why does violet bend more than red?
So see, what is refractive index? It is C by V. Suppose the first was air, second is the medium, right? Now what is, if you express it in terms of wavelength, so C is a constant anyways, right? So what about mu? It can be expressed as mu lambda, right? So anyway, so any colored light in air, it travels with speed c, right? So its frequency will be same only. So this and these are constant. Therefore, refractive index is inversely proportional to the wavelength. Okay. Yeah. So for violet, least wavelength. Wavelength is least. And for red color is most. So if it is greater, like for red, it is greater, the refractive index is less. The opposition for light travel in the medium will be less for red color. Okay, so for red color, refractive index is less, and for violet color, it will be more. Okay, so violet will travel with least speed, red will travel with high speed. Understood? Okay. So wavelength is the color of light. Okay. So wavelength is represents
So what is the symbol?
lambda represents the color of light. It is a Greek letter lambda. Okay. So we just completed question 15. Okay. So the factors which it depends are two factors: the color of light and refractive index of the medium. Refractive index of the glass. Okay. Question number 16. So you have to complete the ray diagram for it. So there are some questions like this also. So which will also answer question 14 when you do question 16. 16 and 14 are same. So IMN represents the narrow beam of light that enters a rectangular block of glass and travels across it. Indicate in the diagram the path of the light ray after it leaves the block at N, right? So what will you do?
Bend away from
True. So at N you will draw the normal, but how will it bend away? So what you do is you keep your scale at I, okay? And then draw the dotted line in the direction of I coming from YX. Okay, so what you will do, look here. So from I, you'll keep a scale and draw a dotted line coming out from the bottom edge. And at N, you'll draw a line parallel to the dotted line. N, you'll draw a line parallel to that dotted line. The dotted line that you got in the direction of incident ray. You have to use scale, Jamshed. You are using freehand. That's not. You don't have scale right now. Huh? Didn't bring today. Okay. So carefully draw the straight line, dotted line, and it should come out of the bottom edge. Okay. So whatever is this direction, the ray at N should be parallel to that dotted ray. That's all it is. Okay, I have covered this in theory. Right? Okay. Now read question 17.
Where should the object be placed to get a real inverted image of same size as that of object in front of a convex lens of focal length 30 centimeters?
So it's repeat. At 60 centimeter. You have to keep it. So there was a similar question like that before. So same as question 13, right? Similar to question 13, right? So you can do it. Okay. Question 18. Somebody else please read.
Diagram showing diminished virtual image by lens.
So ray diagram showing diminished virtual image by lens. It is only possible in case of concave lens. Only in case of concave, because in case of convex lens, the virtual image is magnified. Got it? So you will draw two object positions at infinity and beyond beyond optical center. Beyond O, right? Any finite. Yes, at finite. So this also we have covered in theory, question 18. So it is basically image formation by concave lens, right? Okay, now question 20. Students sitting online, attending online and offline. So 20 you should do yourself right now, okay? And ask me any doubt. So online students, you can also ask me doubt. So unmute your audio and ask doubt, okay? If you have any. So try yourself, question number 20. There are 8 sub questions. So what we are doing right now is question 20. Last one. What is the page number? Page number is 51. Try. So at home, when you're practicing, use your geometrical box. Okay. 10 minutes to do this. After that, we'll take up some ray diagram session. Worksheet में जो है last class put together. You had when did you come? Now it is starting 7:30. Next class will start for you 7:30. What are your school times? Shouldn't be a challenge. Will it be a challenge for you people?
A challenge.
What can you get up?
5:30. What's comes at 6:50?
Where do you stay? Close by, huh? You know, walking out. So it's like 15 minutes. Oh. But right now schools are closed, no? That's
Yeah,
also.
after school, like after vacations.
No, it should be it should be evening time. So all the questions? Simple? No, not simple. I'm not read. I don't know. Is it difficult?
Sir, can we use the answer given here?
One second.
In the first one,
No, answer is given just for you to verify, not that this is your answer.
we can read.
No, I can verify this. Yeah. Sometimes the answers are incorrect. Okay. So in your volume, only the NCRT exercises but MCQ answers are rest are not. And in volume, one answer given is wrong. You know which one? The 17th question answer. Page number 71. This answer given is wrong. 2.14. We got 0.2. So this should be, as per the question, this should be 0.214. So this is wrong here. So how will you do the first question? See, now these are the refractive index problems, right? So let me do the first one for you. I'll do the first one.
First one, it is 1.42 divided by 1.5.
Yes. One second. So refractive index of diamond is Ri or diamond. Mu, so it is given as mu, right? Mu diamond is equal to 2.42, right? And that of window glasses, new window, new glass, this glass. Should I write just D for diamond and G for glass?
Yes.
Right? So what does this mean? This means this is equal to C by velocity of light in diamond. And this is equal to C by velocity of light in glass. Okay. So what we have to find out? We have to find out the velocities of light in glass and diamond. So I can take the ratio. This is 1, this is 2. Right? So comparing, therefore, taking ratios. We get, right? V upon mg is equal to C upon Vd divided by C upon Vg. Right? C upon Vd, because this is nothing but absolute refractive index of diamond, and this is absolute refractive index of glass, because they are only talking about diamond and glass. So since it is absolute refractive index of diamond, the ratio, the first medium should be C and the second medium should be diamond itself. Okay. So now you cross multiply, this is C upon Bd into Bg upon C, fraction. And what is mu d upon Bg? It is 2.42 divided by 1.51. So this is cancelled. Right? So therefore, Eg is equal to 2.42 upon 1.51 times Vd. Okay, so how much is this?
1.6.
Did you calculate? 1.6.
1.6 is coming close.
Almost 1.5. Almost 1.
So this is how we'll solve this question. Understood? So this is question 20, part 1. So these are refractive index questions. Okay. Have you got how we got it? Using the formula for absolute refractive index.
So
Right.
you even give us that. So the thing you did.
Taking rate for comparison, we have to take ratios away.
Yes, sir. You take that in theory as well.
Yeah, I gave that in theory, yes. Right. So this is dividing means multiply with the reciprocal, right? Fractions. Fractions divisions are multiply with the reciprocal. Right? No?
Yes.
Right or no?
Yes, sir.
So this is the numerator, then the denominator reciprocal multiply. That's how fractions calculation are done. So therefore now CC will be cancelled. This is by definition of why did I write this? Of absolute refractive index. So, okay. Try the next one. This is the answer given in the book, right? Velocity in glass is 1.6 times velocity in diamond. Okay. I use the simple mu. You could have used n also, right? Sometimes n or mu is used. Four. And this is because you won't get it out. I think choose less after that. Yeah, what is all? You don't try to answer it actually. To understand the logic, how we got those answers. So every comparison is through ratios. So you take ratios of the given quantity. So what is given? The refractive index of diamond and the refractive index of glass is given. But by definition, what is it? It is ratio of the speeds of light in those medium. So since it is absolute refractive index, first will be speed of light, second will be this is in vacuum. C is speed of light in vacuum or air. This is the speed of light in diamond, VD. And this is the speed of light in glass. But both speeds will be same in vacuum. That's how they will get cancelled out. So how's it going, online students? Arya, Yog, is it okay? Okay, good, very good. So with this class, how many hours you would have completed for this topic? 3 hours for today. And how many hours you've already taken? Earlier, how many classes? Each class was of 1 hour earlier. So how many classes?
3 or 4 classes.
You're taking 4 classes? 4 plus 3, so this will make 7 hours. So you have 3 more hours for this. Let's see. So now I want you to do as many numericals as possible before you come for next class. Okay? So get thorough with this. So are you feeling confident with what we did today?
Yes.
Fourth one. Okay.
Sir, I also got the wrong answer of that.
Okay, wait. So, so this is section 20, part 4.
Yeah.
So what is the given data? T is equal to 4 diameters. Right? Where it also says it is concave.
So minus 4.
So it is minus 4 diameters. Therefore, what is focal length? 1 upon minus 4. This will be minus 0.25 meter. Okay, this is what we will get. So therefore, F is equal to minus 25 centimeter. Okay, now what is given? If the object distance is 4 times the image distance, right? So let us say image distance is let image distance is equal to V, right? Therefore, U is equal to 4V. Okay, and both will be negative because for concave lens, all the image will be on the left side, right? So image distance is minus V, U will be minus 4V because it is concave lens, right? So now what is F? So therefore, 1 upon F is equal to, it is a lens, 1 by V minus 1 by U. What is 1 by F? It is 1 upon minus 25. This is equal to 1 upon V. So what is U? Minus 4V.
So 1 by minus
2. It is also minus.
So but then at the end to find image distance and object distance, we have to multiply by minus 1.
No, so if I know V, I will know U also.
Yeah, but if we know V, we have to get multiply by minus 1 at the end to get it.
Yes, yes, that's right. So now we'll find V with this, right? So what is V? So all the minus sign you just cancel from bottom, right? So V 1 by 25. 1 by V. Take 1 by V common. Okay, this will be 1 minus 1 by 4. Right? 1 by V is common, so 1 minus 1 by 4. So this is 4 minus 1, so this is 3 by 4. So this is 1 by 25 is equal to 1 by V. So this will be V by. Right? Okay. So what is it? So V is equal to 25 into V by 2. This is what we. So 4 6 are 24 point, right? So 4 2s are 8. 4 5s are 20. How much is this? So 3 5s are 15, 1, 18.75. So B is 18.75. So what is image distance minus B? Therefore, image distance is B is 18.75. So what is image distance? Minus 18.75. Okay. And what is U? Minus 4 into 18.75. Because B is 18.75. So substitute 18.75. Whatever is B, it will be with negative. Find out. Minus
70.
So are we getting the answer?
Yes.
So where were you going wrong? All of you. Just check. Where do you make the mistake?
I didn't get the power in 0.25. I got 15.5.
Directly minus 1.5.
This is the thing. Its focus is in meter. Everything else is. The answer is in centimeters.
Sir,
Okay. Question.
So I did the calculation, but I'm getting a different answer.
Then fifth, fourth you had got the answer.
No, sir. That's what I did.
Achha, fourth it's so you went to fifth directly. Okay. What is it?
I did the calculation. Answer is
Object is kept 6 centimeter away. So this is minus 6. U is equal to minus 6. What I'm getting U equal to. This is minus, and this is minus 6. U. So you are not writing the given data and all that. You are directly writing the formulas. Is this how I have told you? How we are doing? What is a lens and what is given data which I am writing? This is why you will shortcuts will not get you correct answer.
and so then I
No. You have to follow that. First write down the given data. Capture the data first correctly. Okay? Then write the formula. Okay. So once you have achieved some expertise, then you do this shortcuts. Initially you do the problem. You got the answer? Any difficulty with this question?
no, I
So follow the method, right? Don't go for shortcut. So actually we did one thing wrong here. We could have done it with just B and 4B. I said unknown quantities you don't put sign. Then in calculation you would have got minus. Okay. So with unknown quantity I place signs also. Then we had to do this jugaad basically. Although because of consistency we got the answer. So you can try it yourself later on without sign and see if you're getting the values.
So when we
Yeah. You got minus, right? Yes. Very good. Lens check. Whatever lens check.
then
So where is the question? -6. This is +1. Where is that? This is +1. B. This is the 1.2. Divide 6 by 5, what will you get? 5 ones are 5, then point and 10, so 5 will get. What is 6 by 5? B. 1 by B, so B is reciprocal, not 6. Completed? Okay, now proceed with. So I think only 2 questions were tough, right? First and fourth in this. Rest everything is pretty easy, okay? And so rest are homework questions, right? Okay, so on page 52 you have homework questions. Then on page 53 you have worksheet. So in the remaining time, whatever time we have, you can try worksheet. Okay, these are like workbook kind of a thing. So this is good. Lot of questions are there on ray diagrams. And I think it's taken from. So you can expect many questions from this in exam. Okay, many incomplete ray diagrams are given. Okay, so see the position and draw. You can do it. Worksheet. Okay, so worksheet. Try these questions.
In the second one, it is particles.
Where in worksheet? Light energy is transmitted as electromagnetic waves. Waves, right? I have already written something. Wave is different, particle is different. So when it is traveling as wave, so it is this kind of wave. Electron. What answer? So online students, you can proceed with worksheet questions. It is on page number 53. On page 53 now. So light is a form of energy. So since you are not asking any questions, so I assume that you are understanding everything. And some of the difficult questions I have already done on the board from 20. Rest should be easy for you.
Very small connection of light rays called
Beam of light.
so then larger connection.
Intense beam of light. So that's a funny question. I don't know. It's like I coined that intense beam of light. Anything else was not fitting it. So if you get anything else, just discuss.
Larger.
Whatever, like intense, large.
So a path of light
You didn't get the volume or you forgot to bring?
I forgot to, so I have volume, but light is not.
Volume is correct.
Sir, of certain one surface which returns the light after falling on it is called a reflecting surface.
Fourth one,
Of second.
surface which returns the light monomer reflecting surface. So I suggest fill it with pencil. Then we can, if we make a mistake, we get it. If you don't have it today, it's fine, but try to fill the outside. Sorry, question number. Very right forgetting the point of incidence is called yeah. Point of incidence is called normal. This question 3, 8 and 9. Is the second. MOA angle is 0. There is MOA. MQO dash is dash is the surface. O dash. Then this you can write in. So this is P dash. This is Q. Okay. This 2 you can cancel. So we call it RS.
So, this one, this
Yeah. So MOA is 60, and this thing is like it's right should be R will be 30. This is also this.
this is the expression. This
Same thing. It's all related. MOA is 60. Then I is 30. Then R should be 30. This should be 60. What is 90, right? So this should be 60. Because I and R will be same.
So we can have this.
That is for 7. 8 is different. 8 is starting with that way. There are different questions. Diagram is same, but questions are yes. I will take it. The diagram drawn is question number. Question 3. The diagram is something like this. So this is M dash. Okay, this is the reflecting surface. It's a plane reflecting surface. You have a source of light over here. Okay, look here. This is the source of light. Yeah. So this is the source of light. This is this is P. 1 second. This is P. So this is the source of light, and 3 rays are coming from. Okay. So now, if you draw the normal here, the angle of reflection will be equal angle of reflection. So here angle of incidence is more, so angle of reflection also will be more. So these rays are now diverging. So where will the image of it will be formed? It will be formed somewhere same point, right? So wherever it is. This is our okay. So here it is this ray is along the normal, so it will be reflected back. So its image is formed here. This is its image. This is P dash. This is Q. Okay. This point they are calling as O. This they are calling O dash. So you can take this as R and S like that. So the diagram at the lower portion of the book, consider this diagram. Okay. Now is it clear? So this diagram shows image formation by plane mirror. And this is the source of light. Or the object is here. Object is at E. Yes.
This is P dash.
Yeah, that is P dash. The P dash below. This is. What was the question? Yes, same size. Because plane mirror. So all questions related to this diagram answered? So many questions they have collected on this chapter. But you can sit peacefully and do it. In the next class also will be solving numericals only. So in the next class I think it will be completed with this chapter. And if possible we'll start with this chapter. Question number. Rectilinear propagation of light. Rectilinear. So I think it's time also. So we'll meet in the next class now.
So rectilinear.
The rectilinear propagation of light. Which question was that? Question number 5, right?
Yes, sir.
Yeah, rectilinear propagation. School, how many hours you have to spend in school?
From 6 to 1.
6 in the morning to 1.
Sorry, 7 in the morning to 1.
7 to 12 is 5 hours. Okay. All right. So, okay, I'll see you in the next class.
Yeah, same here.
नहीं यार श्री राम
अबे प्लीज
काम करते यहाँ पे बैठा यहाँ पे बैठा मुझे ये क्या है मैं इधर बैठा
यार क्यों ले चार चुटकी फिर भी एक साइड
मेरे को तुझे ही नहीं
अभी
इधर बैठ जो कर रही ऐसे बैठ जाओ क्या सर जी निकाल दिखा हो गया
इस चैनल की जगह पे बैठ चैनल को ना
मैं कैसे हमें नहीं आ रही है
यार यहाँ पे बैठ तू यहाँ पे आ जा
क्लास के लिए ना एकदम जल्दी मत आ जाओ, 5 मिनट पहले आए तो बस। और ये कहाँ है? अयान आया है? नहीं। अर्पिता है? अर्पिता भी नहीं। प्रिशा है? नूपुर, अन्या, ये तीनों का भी ना आरएमओ में सिलेक्शन तो हुआ है, और अगर लेने का है तो एडमिशन ले सकते हैं। कल ही फर्स्ट लेक्चर है। Trisha, you got 13 out of 20 physics। और ना मैं ना जोर से मार्क्स भी रीड करती हूँ। सारे बच्चे नहीं आए हैं, आने का तो मैं वेट कर रही थी, बट मैं थोड़ा जल्दी आ गई। ताकि तुम्हारा आगे से तुम्हारा मार्क्स थोड़ा इम्प्रूव हो जाएगा। इसके लिए अगर कम मार्क्स आए तो ना सबका सामने मैडम आके रीड कर दिए करके ना थोड़ा शर्म आ जाता है, तो इसके लिए थोड़ा अच्छे से पढ़ के आएंगे, है ना प्रेशा? हाँ, क्या करो, जोर से पढ़ो या नहीं? You tell me, Shriram. You tell me. जोर से करूं या स्लो करूं? मत बोलो, मत बोलो। अरे श्रीराम को तो बहुत अच्छा मार्क्स आया है। हाँ? पढ़ना चाहिए ना? I go with her. श्रीराम 18, जिसका मार्क्स 15 के ऊपर आया तो मैं पढ़ूंगी। हाँ, चेक कर। अरे जाते वक्त ले लो ना सारे, वो मोबाइल भी तुम्हारा ही है। पहले बोलने का था ना। उन्नति is there, सुहानी, तन्मय, which one? वो तो दिया ही। अर्पिता, कीर्तना, आज का दिन मैं माफ करूंगी बट आगे से नहीं होता है, और ये पेपर तुमको ना 10 टाइम में लिखवाऊंगी। अरे इतना पढ़ाने के बाद में इतना कम मार्क्स क्यों आता है? वेयर इज अनुष्का? क्यों इतना कम मार्क्स आ जाता है? पढ़ाते वक्त सुनने के लिए नहीं आता है, सुनेगा तो ही मोर देन इनफ। सोहम नहीं, नूपुर 16, ऐश्वर्या, अच्छा चारवी, शयनदीप, तो इधर किस चीज के लिए आता है? हाँ, से करने के लिए। बैग लेके आ जाओ इधर सामने, और उधर ग्रुप में नहीं बैठने का, अलग अलग बैठो। In case तुम्हारा मार्क्स कम आया, if I am not satisfied with your marks, you have to write this paper 5 times. एक दो नहीं। ओमकार, आ जाओ तुम्हारा बैग लेके इधर बैठो, इधर फर्स्ट बेंच खाली है। निहार, और एक साथ दोनों जगह पे साइन नहीं करने का, एक एक पीरियड में एक एक करो। तुम्हारा अभी का क्या है? केमिस्ट्री है, केमिस्ट्री में साइन करो। और सब लोगों ने टेस्ट दिया या कितना लोगों ने अभी भी देने का बाकी है? यू हैव नॉट चेक योर टाइम टेबल? व्हाई? उठने का आने का वैसे ही। अभी ये ना थ्रू आउट दिस मंथ यू हैव टू चेक योर टाइम टेबल, बार बार चेंज हो जाता है। सुहानी, मार्क्स देख लो कितना अच्छा है करके।
Good evening, how are you? ठीक है ना? इसका बायो लेक्चर है कि क्या है? केमिस्ट्री। गुड। और क्या लिखा आपने? कल टेस्ट है, पीरियोडिक टेबल। रिवीजन। कोवलेंट बॉन्डिंग हो गया ना? नहीं लाए हाँ सोडियम क्लोरीन और नियॉन the electron affinity of fluorine will be high. इलेक्ट्रॉन एफिनिटी पूछ रहे हैं तो नियॉन का नहीं रहेगा क्योंकि
और सर ये वाला एक क्वेश्चन
Okay, now listen to it. We are dealing with coordinate covalent bond, the third type of bonding. Okay, last का थोड़ा जो question या doubt तुम लोग पूछ लेना chapter से या नहीं last 10 minutes में या 15 मिनट में पूछ लेना जो doubt है. Now what is this coordinate covalent bonding? ठीक है, coordinate covalent bonding is basically a special type of bond here in which an atom or a group of atoms, ठीक है, atom or a group of atoms contributes, or we can say for example share both the electrons from it, but other does not, other contributes nothing, because otherwise covalent bond is what one atom shares, other also shares, both of them mutually share the electrons, but in this case one atom only contributes. Let us take an example over here. For example, here, if you see the dot structure for ammonia, the dash structure of ammonia, ammonia के पास कितने इलेक्ट्रॉन हैं? 6 इलेक्ट्रॉन इधर है, एक लोन पेयर इधर है, 8 ऑक्टेट कंप्लीट हुआ, अमोनिया स्टेबल है फिर, बट एक हाइड्रोजन है ना अप्रोचेस दिस अमोनिया कि मेरे पास कुछ नहीं है, मैं खाली हूँ, ठीक है, मेरा डुप्लेट कंप्लीट करना है. Then with this hydrogen has got, for example, shortage of 2 electrons, और इसके पास filled with octet. तो ये approach करता है nitrogen को, and nitrogen has got one pair of electron which that atom is not utilizing, which is called lone pair of electron. This lone pair is also called, okay, unshared electron pair. ये pair of electron unshared रहता है तो atom क्या करता है, अगर if any other atom is approached, last by सुनो, ध्यान दो, कल एग्जाम है, वो ये टॉपिक नया ओपन नहीं है उसका, नहीं इसका नहीं है, लेकिन एग्जाम तो है ना, हाँ तो ये आगे का टॉपिक है, उसके बाद इसका भी एग्जाम हो, तो इसको भी ध्यान देना, और बाद में बोर्ड एग्जाम है तो सब टॉपिक को ध्यान देना, ये that's what I'm driving at. लास्ट में सुनो, इधर आओ, कम हियर, इधर बैठो, क्योंकि अभी पहला टेस्ट है, पहला परफॉर्मेंस बहुत इंपॉर्टेंट, ठीक है, फर्स्ट इंप्रेशन इज द लास्ट इंप्रेशन. नाउ लुक एट, ये देखो, ये जो लोन पेयर ऑफ इलेक्ट्रॉन्स है, ठीक है, the hydrogen approaches this nitrogen which is having a lone pair of electron, इसको मैं रेड कलर से बना देता हूं, ठीक है, इसका octet complete. So what happens, this hydrogen is short of 2 electrons, so what it does, it approaches this particular nitrogen, gets the electron, ठीक है, and finally becomes what, for example, this nitrogen donates a pair of electron to hydrogen. This time the donation is coming from this atom only, which that means we show by an arrow. ये arrow kind of bonding है, और ये देखो hydrogen is actually what, sharing its electron with nitrogen equally by covalent bonding, and in the process the whole unit becomes positively charged because the hydrogen is attached हो गया. फिर भी वो actually what is having, like for example short of duplet, but because of this pair of electron being transferred to hydrogen, the head of the arrow will be towards that atom which has accepted that lone pair. The end of the arrow will be towards that atom which has donated. That means here this is a contributor or donor of electrons, and this is acceptor. तो ये 3 बॉन्ड इसमें अमोनिया, and this becomes ammonium ion. So 3 बॉन्ड कोवलेंट है और 1 बॉन्ड कोऑर्डिनेट कोवलेंट. यानी दोनों इलेक्ट्रॉन एक ही एटम कंट्रीब्यूट करते हैं, other does not contribute anything. Okay, it's like saying this way that you are actually having the tiffin box, and your friend who's just next in the next bench has got empty tiffin box. He approaches you, takes your tiffin box from your hand and starts eating from it. Once it starts eating, you look at his face and you get satisfied. At least let him eat. He's more hungrier than me, so let him eat, and you get satisfied by giving your tiffin box, and not a single piece is left out in the tiffin, because he actually what takes each and every. This type of bonding is a special type of bonding. बहुत कम लोगों के पास ऐसा स्पेशलिटी है, और टिफिन बॉक्स लेके आना घर से और दूसरे लोग खा ले रहे. Every day you get shouting. Every day you get shouting from your parents, mother especially, because she is cooking that particular nashta at 6 o'clock. अगर क्लास तुम्हारा लेक्चर अगर 7 o'clock है, तो सवेरे जल्दी बना के दिया उन्होंने. But not a single piece you had. And every day afternoon she asks, how was the taste? How was your tiffin? Okay. मुझे मालूम क्योंकि मेरा फ्रेंड ही खा ले रहा है. Special type. Okay. So this kind of bonding is said to be what? Then the people they actually question you, sir. उसने क्यों ले लिया? उसको क्यों दे रहे हो बार-बार? Like for example, the child is. I mean, this particular entity is not having any tiffin box. They get to, in order to attain stability, if suppose something like suppose you don't listen. Mainly क्या होता है? तुम क्यों नहीं actually tiffin अपना box में ऐसे खा रहे हो? Because you are not feeling like having it. You are unusing the tiffin box. Since ये लोन पर use नहीं हो रहा है, तो friend ने तुम्हारे request किया, अरे तुमने खाया तो मुझे दे दे. मुझे तो भूख लगी है. Your neighbor is having bicycle, and every day you are just walking out of your house and you are seeing that bicycle is being unused. One fine day you will find that your neighbor is just coming out from his house, and you gather strength and you अरे तू अगर साइकिल नहीं यूज कर रहे, मुझे तो स्कूल जाने के लिए अच्छा रहेगा, suppose I can get for just temporary time, and he gives to you out of nothing. In return, he is not asking for anything. He or she, it's a special type of. भले उनको गुस्सा मिल रहा है या जो भी मिल रहा है, but he is giving you, has given you the bicycle, okay, and every day you are using it, enjoying that particular ride, okay, going to school without any walking and exercise, and therefore is, and then but you remember, you are thankful to that your friend, special type of coordinate over it. Likewise, water, water is actually what, having two lone pair of electrons. अब हाइड्रोजन इधर अप्रोच कर, ठीक है कि नहीं? ओके, हाइड्रोजन के पास शॉर्टेज है 2 इलेक्ट्रॉन, तो इमीडिएटली ये जो लोन पेयर ऑफ इलेक्ट्रॉन है वाटर का, ये इसको डोनेट कर, जस्ट लाइक दैट. इफ यू रिमेंबर दैट 4 लोग अम्ब्रेला में जा रहे हैं, और ये चाइल्ड मिसप्लेस हो गया अपने पापा ने, एंड इट इज रेनिंग हेवीली.
चाहता है
ये चाइल्ड इज हैविंग, ये चाइल्ड is actually what has lost his parents, lost means misplaced, and therefore what, if you find for example that this child comes and holds the hand of the smallest child over there in the family, and without asking a single word, he takes this child along, coordinate over it. In return, he is not getting anything. It's just a question of stability.
सर ऐसा ये तो फिर वो जो बाकी हाइड्रोजन बॉन्ड हाइड्रोजन है जो नाइट्रोजन का अटैच है वो भी कोऑर्डिनेट क्यों फॉर्म करते हैं?
वो उन लोगों के पास ऑलरेडी बॉन्डिंग चला गया ना,
अगर वो कैटायन अप्रोच करते हैं नाइट्रोजन को, तो
नहीं ऑलरेडी इन लोग ने अपना इलेक्ट्रॉन शेयर कर लिया ना. नाइट्रोजन
अगर नहीं होगा तो
किधर नहीं होगा?
फिर अगर नाइट्रोजन का अनायन है और फिर हाइड्रोजन का 3 कैटायन आता है, फिर वो डोनेट कर देता है.
नहीं, दोनों नॉन मेटल है ना, नॉन मेटल तो हमेशा गेनर्स ही रहेंगे, लूजर्स
फिर वो
किधर से?
वो हाइड्रोजन
ये बिकॉज़ दे वर इन द फॉर्म ऑफ हाइड्रोजन एटम, ये हाइड्रोजन आयन है, इसके पास कुछ भी नहीं है. डुप्लीकेट करने के लिए 2 इलेक्ट्रॉन चाहिए ना, इन लोगों को 1-1 इलेक्ट्रॉन चाहिए था, तभी वो शेयर कर रहे हैं. वो कोवलेंट बॉन्ड था लास्ट टाइम का. अब ये कोऑर्डिनेट कोवलेंट जहां पे दोनों इलेक्ट्रॉन हाइड्रोजन को जरूरत है, ठीक है कि नहीं? तो ये आयन है, आयन के वेले डुप्लीकेट उसको चाहिए, 2 इलेक्ट्रॉन चाहिए, ठीक है कि नहीं? और ये सब एटम्स थे इंडिविजुअल. यानी इसके पहले क्या था, ये देखिए, इसको पहले आपको याद नहीं होगा, ओके, ये देखो ये नाइट्रोजन है, इसके पास 3 इलेक्ट्रॉन है, 3 हाइड्रोजन इसको अप्रोच कर रहे, अपना अपना इलेक्ट्रॉन लेके, बिकॉज़ इन लोगों के पास 1-1 इलेक्ट्रॉन तभी था ना. ये न्यूट्रल हाइड्रोजन, ये तो न्यूट्रल नहीं है, ये कैटायन है. कैटायन में दोनों इलेक्ट्रॉन उसके लूज हो गए, उसके पास कुछ भी नहीं है. आई फॉलो में, तो उसको दोनों इलेक्ट्रॉन चाहिए. यू अंडरस्टैंड डिफरेंस? ये एटम है और वो आयन है, तो आयन में ये कॉम्बिनेशन. टिफिन बॉक्स लेके नहीं आया कुछ, अगर तुम्हारा फ्रेंड तो कुछ भी नहीं लेके आया, तो उसको अपने को, अगर सपोज हम लोग अनयूज्ड काइंड ऑफ टिफिन बॉक्स इज देयर, वी हैव टू we have a tendency to give it, चलो उसको खाने दो, as a matter of fact. It's a question of stability, ठीक है? Because if he eats, then he will have energy and he can attend the lecture properly. Okay, okay, so this is something, and you will find, for example, जैसे ये लोन पेयर इधर ट्रांसफर हुआ, हाइड्रोजन इसके साथ अटैच हुआ, एनर्जी रिलीज हुआ ये वहां पे, he gives a smile, वो स्माइल तुम्हारे लिए इनफ है स्टेबिलिटी के लिए. मेरे वजह से ही has attained stability. पहले जो इस चेहरा उतरा हुआ था, एनर्जी एकदम डाउन था, अभी एनर्जी एटलीस्ट स्माइल has come on his face, positive thought has come in his mind, right? जब स्माइल कब कोई आदमी करता है या पर्सन करता है, जब उसका पॉजिटिव एनर्जी तुम्हारे पास है. अगर गुस्सा आ गया तो नेगेटिव एनर्जी, अगर स्माइल कर रहे हैं तो पॉजिटिव. ओके, रिमेंबर, सो दैट इज द आइडिया ओवर हियर. यहां पे देखिए, इसको ये इमीडिएटली स्माइल कर दिया क्योंकि इसको 2 इलेक्ट्रॉन मिल गए. like suppose it's like saying this desperate kind of tendency you will find in the examination. तुम अच्छी तरह प्रिपेयर्ड हो एग्जाम के लिए, बट अनफॉर्चुनेट तुम्हारा पेंसिल बॉक्स तुमने घर में छोड़ दिया। तुम डेस्परेट किससे पूछ रहे हो, अरे मेरे को एक पेन तो दे दे। और उसमें क्या हो रहा है कि फॉर एग्जांपल किसी ने तुम्हारे को पूरा एक पेंसिल बॉक्स ला के दे दिया ना विद पेन एंड अदर थिंग्स, you will get big smile to that particular person. Why? It's a question of examination. चल तू don't worry, मेरे पास दो है, मैं एक यूटिलाइज नहीं कर रहा हूँ, फॉर एग्जांपल वो यूटिलाइज नहीं कर रहा है, when you're not utilizing, you're giving it. For him it's a big thing, but for you it is a small thing. अगर मैं I was not using my bicycle, I have given it to you, no problem here, don't worry, okay? ऐसा करके तुम बोल देते हो उसको, okay? अब बाइसिकल तुम रोज देख रहे हो कोई जंग हो रहे हैं और फ्रेंड बोलने मैं नहीं दूंगा, ठीक है? उसने अपना लिए लिया है, लेकिन वो रोज तुमको और तुम उसको एंगर दिखा क्या मुझे रोज स्कूल पढ़ाते हैं, दिस इज अ ह्यूमन टेंडेंसी, ठीक है? तो अगर किसी के पास एक्सेस है और वो नहीं दे रहे हैं तो आपको एंगर आएगा। अरे क्या रोज रोज मैं देख रहा हूँ, रोज रोज जंग उसका बढ़ता जा रहा है, एक टायर हमेशा फटा रहता है, तो मैं एटलीस्ट आई विल यूज इट एंड आई विल मेंटेन इट एज अ मैटर ऑफ फैक्ट, ठीक है? दिस इज व्हाट एंड दिस इज व्हाट इज आइडिया दैट फॉर हाइड्रोनियम आयन बनता है, इसको बोलता है हाइड्रोनियम। उन्होंने देखा ना वाटर एसिड्स और बेसिस में देखेंगे हाइड्रोनियम। So one is a donor atom, another is an acceptor. इसमें donor और acceptor है, coordinate covalent bond, ठीक है? This happens with the neutral molecules and the ions, और neutral atoms हैं जिसके पास 8 इलेक्ट्रॉन है, जिसके पास डुप्लेट नहीं है, ओके डुप्लेट नहीं हो रहा तो उसमें फॉर्म होगा, जिसके पास 6 इलेक्ट्रॉन्स है उसके लिए भी ये coordinate covalent bond है, ओके? रिमेंबर जैसा भी कोई एक स्टूडेंट ने मुझे इधर पूछा था, सर ऑक्सीजन में तो आपने डबल बॉन्ड ले लिए, व्हाट अबाउट ओजोन? ओजोन का पार्ट इधर आ रहा है, चलो ये देखिए ओजोन में क्या हो रहा है, डबल बॉन्ड ऑक्सीजन के पास है, 2 पेयर ऑफ इलेक्ट्रॉन है, ठीक है? उसके साथ एक ऑक्सीजन आ गया, ऑक्सीजन अनस्टेबल है, 6 इलेक्ट्रॉन उसको इसके पास है, उसको ऑक्टेट कंप्लीट करने के लिए 2 इलेक्ट्रॉन चाहिए, इमीडिएटली ये ऑक्सीजन अपने 2 इलेक्ट्रॉन इधर दे देते हैं, ओजोन फॉर्म होता है। ये देखिए O3 जो कोवलेंट बॉन्ड में नहीं दिखा है, ऑक्सीजन में इधर है, दिस टाइप कोऑर्डिनेट कोवलेंट, यानी ओजोन में एक डबल बॉन्ड रहेगा और एक कोऑर्डिनेट कोवलेंट बॉन्ड, ओके? ये वी शेप ऐसे बना सकते हैं, बट हम लोग लीनियर बिकॉज़ यू आर इन 10th स्टैंडर्ड नाउ, ओके? 12th में विल शो वी शेप स्ट्रक्चर, दिस इज ओजोन स्ट्रक्चर। तो ओजोन में क्या है, डबल बॉन्ड एंड देन कोऑर्डिनेट कोवलेंट ऐसा रहेगा, ठीक है? ओके देखो coordinate covalent bond, Arpita read out coordinate covalent bond, but before reading out please write down, then करते बना लिया आपने, बोलिए
साइड से भी ला सकता है,
इस साइड हाँ आ सकता है, लेकिन वो अगर सपोज सब जगह से आएंगे तो क्या होगा, एटम नजदीक आएंगे, रिपल्सिव फोर्स वो अपने इलेक्ट्रॉन वापस ले सकते हैं,
अगर उसमें वाटर में ऑक्सीजन का 2 लोग हैं,
हाँ तो एक और यूज हो सकता है, बट ये ज्यादा स्टेबल बिकॉज़ जितना आएंगे नजदीक व्हाट विल हैपन, रिपल्सिव फोर्सेस विल डेवलप, सो इट विल बी दे मे ब्रेक। So what is the condition for this type of bond? One should have a lone pair, other should be deficient of lone pair. positive thoughts. Positive thoughts will lead to smile, laughter. Negative thoughts will lead to anger, crime, overthinking. Yes. So overthinking what local exam. Think about positive. Huh? The girl school का exam. Double work. अभी तो छुट्टी हो गई ना? नहीं. Now the vacation must be going on.
Yes.
No, no. क्या बात?
अभी का एग्जाम चालू है। जय स्कूल का।
कौन सा स्कूल है वेकेशन में चल रहा है?
Study leave. Study leave. Yes, it's not going.
अच्छा स्टडी लीव.
Sir, time now.
Just जाना is not mandatory.
जाना allowed नहीं है।
Online.
Project से related सब जाते हैं समझ के लिए।
परीक्षा हो गया आपका? कुछ नहीं लिखा? लिखिए वजह से एविडेंस देखिए लास्ट में आपने लिखा वेरी गुड H3O plus. H3O plus how this H plus ions are formed वो भी देखना है आपको ठीक है I'll just show you how the H plus ions are formed हाँ एक थोड़ा सा कंप्लीट कर देता हूँ टॉपिक में डिस्कशन होगा ना तभी तो लोगों को मालूम पड़ेगा अरे बोर्ड क्वेश्चन डिस्कशन होगा हो गया ना फिर से एक बार रिवाइज कर लो ठीक है तो क्या होगा कि अपना अच्छा फॉर एग्जांपल वो फाउंडेशन फॉर्म हो जाएगा Okay, now let us come to how the formation of hydroxyl atoms takes place. ठीक है जैसे water सुनो इधर देखो ध्यान से देखो ये अपना हो गया हाइड्रोनियम now we know very well water always splits up in the form of what water splits up in the form of okay H plus ion and OH minus ठीक है ये क्या हो रहा है water splits up so how will I show the reaction or how will I show the formation of the electrons which are there ठीक है for example अगर suppose देखे जाए तो H2O okay loses now this H plus ion completely loses its all the electrons इसका दोनों इलेक्ट्रॉन ठीक है अभी सपोज अगर मैंने वाटर का अगर सपोज फॉर एग्जांपल लेट अस से दिस इज एक्चुअली व्हाट द इलेक्ट्रॉन ऑफ हाइड्रोजन, दिस इज द इलेक्ट्रॉन ऑफ ऑक्सीजन. ऑक्सीजन के पास कितने इलेक्ट्रॉन है? बड़ा, ठीक है. Look at this. This oxygen is having how many electrons? 6. I mean, like how to draw the dot structure for any element. आपको मैं ये सिंपल बता देता हूँ. सिंपल. पहले ऑक्सीजन के पास 6 इलेक्ट्रॉन है, ठीक है ना? तो क्या करेंगे? 1 2 3 4 ईस्ट वेस्ट नॉर्थ साउथ सपोज ऐसा लिखते हैं. और फिर उसके बाद रिमेनिंग 4 इलेक्ट्रॉन 2 इलेक्ट्रॉन्स है, तो ऊपर अगर बिकॉज इस साइड आपको बॉन्ड बनाना है तो आप ऊपर एक लोन पेयर बनाए और नीचे 6 हो. टेक्निक फास्टेस्ट वे, ठीक है ना? ओके. और फिर यहाँ पे एक इलेक्ट्रॉन इधर एक इलेक्ट्रॉन, सो बोथ ऑफ देम विल बी शेयर्ड बाय हाइड्रोजन. ठीक है, ठीक है. नाउ सुनो इधर. अब जब ये वाटर स्प्लिट अप होगा, इधर लिख दे. नाउ वाटर जब स्प्लिट अप होगा तो क्या हो जाएगा? दिस हाइड्रो ऑक्सीजन इज हाईली इलेक्ट्रोनेगेटिव. व्हाट इट डज? पुल्स द इलेक्ट्रॉन. और इसके ऊपर प्रेशर कौन डाल रहा है? ये हाइड्रोजन. हाइड्रोजन इज अ गुड इलेक्ट्रोनेगेटिव एलिमेंट. 2.1 on the Pauling scale. एक साइंटिस्ट था Pauling, Linus Pauling. He studied the reactivity of almost all the elements in the periodic table and he gave the highest electronegative value to fluorine. इस सब के पिताजी थे fluorine. 4.0 on the Pauling. यानी इससे ऊपर कोई वैल्यू नहीं है. उसके बाद नेक्स्ट एलिमेंट आया ऑक्सीजन 3.5, नंबर 2 एलिमेंट. जिस जो इसके बाद यानी ऑक्सीजन से कोई भी इलेक्ट्रॉन नहीं ले सकता, वो पुल करेगा दूसरे से, और उससे एक ही एलिमेंट पुल कर सकता है फ्लोरीन. उसके नीचे अनदर धुरंधर एलिमेंट फ्लोरीन 3.0, ठीक है. और फिर उसके बाद ब्रोमीन आया 2.8. नाइट्रोजन भी यही पोजीशन में है 3.0. वेरी गुड इलेक्ट्रोनेगेटिव एलिमेंट नाइट्रोजन 3.0 इसका सेम. इलेक्ट्रोनेगेटिविटी मोर लेस 19-20 हो सकता है, बट यहाँ पे दोनों सेम है, ठीक है. फिर आयोडीन आया. ऑल हेलोजेंस का वैल्यू बहुत हाई रहता है, और हाइड्रोजन उनका फेवरेट एलिमेंट है हेलोजन का 2.1 on the Pauling scale. यानी सब 2 के ऊपर जो एलिमेंट्स है, लोग you know for example tendency of pulling is very high. Now what this hydrogen does? प्रेशर डाल रहा है, इधर ये हाइड्रोजन या ये हाइड्रोजन, बिकॉज़ वी आर मूविंग ऑन दिस साइड. हम लोग ये हाइड्रोजन इसके साथ ऑक्सीजन भी फुल कर रहे हैं, बिकॉज़ इट इज मोस्ट सेकंड मोस्ट इलेक्ट्रोनेगेटिव एलिमेंट. हाउ मच इज इट? 3.5 on the Pauling scale. तो क्या होता है? ये इलेक्ट्रॉन ऑफ हाइड्रोजन इज पुल्ड मोर टुवर्ड्स दिस साइड. और दिस साइड भी कोई भी साइड ले सकते हैं, बट ऑक्सीजन के साथ एक ग्रुप जाए हाइड्रोजन के साथ. हाइड्रोजन के साथ ये ऑक्सीजन इस साइड उस साइड चले जाएगा या इधर जाए, डिपेंडिंग अपॉन जहां भी आपको ये करना है. नाउ जस्ट द मोमेंट दिस हाइड्रोजन gives its own lone single electron, it becomes what charge? Positive charge. और ये ऑक्सीजन के पास अपना खुद का, फॉर एग्जांपल खुद का तो इलेक्ट्रॉन आएगा, 6 इलेक्ट्रॉन तो आएगा इसके साथ, बट एक साथ यू नो फॉर एग्जांपल द 8वां इलेक्ट्रॉन क्या है, एंड देन दिस इज नथिंग बट व्हाट? नेगेटिव OH. आई फॉलो मी. H प्लस आएगा, इसका ऑक्टेट कंप्लीट हो, बट इसका डुप्लेट भी कंप्लीट नहीं होगा. अब ये अनस्टेबल हो गया. ये अनस्टेबल एटम क्या करेगा? इमीडिएटली वाटर को अप्रोच करेगा. ये देखो मैं तो कुछ लिखा नहीं है, कोऑर्डिनेट कोवलेंट अपने आप को स्टेबलाइज करेगा और हाइड्रोजन आयन करेगा. अंडरस्टैंड दिस पॉइंट? सो दिस इज हाइड्रोक्साइड फॉर्मेशन. हाइड्रोजन को छोड़ दिया उसने, एंड फाइनली व्हाट? यहाँ पे वाटर के साथ उसने बॉन्ड फॉर्म किए एंड बिकम्स व्हाट? H3O plus, where this is coordinate covalent. That is, जो कोई पूछेगा सर ये हाइड्रोजन कैसे मिला, कैसे आपको? Dissociation. Okay, dissociation of water. Clear? Now please read out the content and then we'll go for the questions of this previous topic. ठीक है ना? और उसमें तुमको एक टेक्निक बता देता हूँ कैसे याद रखना. पीरियोडिक टेबल सामने रखना तब जाए. अर्पिता नहीं टेस्ट के समय नहीं, यानी पीरियोडिक टेबल 1 to 20 आपको याद रखना. Please read out Arpita the coordinate covalent bond.
A coordinate bond can be defined as a bond formed between an ion and the atom of a polar covalent molecule with one or more lone pair of electrons. A coordinate bond has properties of both covalent and ionic bonds. Therefore, it is also called coionic bond.
फिर से पढ़िए, बहुत ही अच्छा important point पढ़ा.
A coordinate bond has properties of both covalent and ionic bonds. Therefore, it is
चलो एक मिनट सुनो, ये बहुत ही important point है, लेकिन अपना बुक में नहीं दिया एग्जाम्पल में. Let me just explain you in short. Means ये देखिए चिल्ड्रन, actually आपको मैं बताना चाहूंगा what is actually what happening over here. देखो ये important चीज जो पहला सेंटेंस दिया है ना, it has got a deeper meaning. Let me give you this particular idea. अब देखिए चिल्ड्रन इधर देखिए, H2O. All of you listen to it carefully. इन्होंने क्या बोला? It is a combination of ionic bond and covalent bond. इसीलिए इसको बोलते हैं coordinate covalent. तो ध्यान से देखना. Actually ये दो स्टेप में प्रोसेस चलता है. ये हाइड्रोजन is having short of 2 electrons, but इसके पास ये excess electron. ठीक है ना? तो फर्स्ट स्टेप में क्या होता है? फॉर एग्जांपल ये अपना इलेक्ट्रॉन इसको दे देता है, एक ही सिंगल इलेक्ट्रॉन. ठीक है? और फिर क्या होता है? फॉर एग्जांपल ये वाटर, जैसे फॉर एग्जांपल वाटर के पास एक पॉजिटिव चार्ज है क्योंकि उसने अपना एक इलेक्ट्रॉन दे दिया. यस और नो? ओके. और ये हाइड्रोजन न्यूट्रन बन जाता है क्योंकि इसके पास इसका एक इलेक्ट्रॉन आ गया. जैसे तुम किधर गए, ठीक है, दैट इज आउट ऑफ स्टेशन, और एज अ गेस्ट यू आर एक्चुअली स्टेइंग एट समबडीज प्लेस, और वहां पे एक ओकेजन आया, किसी का उनके रिलेटिव का शादी हो रही है या कोई अपना बड़ा इवेंट है वहां पे सपोज, बट उसके लिए ब्लेजर, यू नो, कोट एंड अदर थिंग्स वर रिक्वायर्ड, बट यू डिड नॉट टेक एनी ऑफ दैट टाइप ऑफ, ये फॉर एग्जांपल क्लोथ्स. तो उन्होंने क्या बोला? तुम्हारे पास तो कुछ भी नहीं, तुम खाली शर्ट पैंट ही लेके आ गए, और यहाँ हम लोग को वो जो जहाँ पे जाना है वहां पे सब एक अपना ड्रेस कोड है जहाँ पे सब सूट वूट में है. तुम क्या करो, मेरा जो एक्स्ट्रा सूट है वो तुम इधर ले लो, तो दोनों इक्वल हो जाए. ठीक है कि नहीं? दैट इज दिस इज आयोनिक बॉन्ड. ये आयोनिक बॉन्ड, उसने अपना सूट तुम्हारे को दिया या अपना मटेरियल तुम्हारे को. इसके बाद द नेक्स्ट स्टेप विल बी व्हाट? नेक्स्ट स्टेप ये आयोनिक हो गया. अभी तुम बोलोगे सर कोवलेंट कैसे हो रहा है? अब इमीडिएटली, ठीक है, यहाँ पे इसके पास एक इलेक्ट्रॉन, ठीक है, पॉजिटिव चार्ज हो गया, इसके पास भी एक इलेक्ट्रॉन, दोनों के पास शेयरिंग के लिए कुछ है, इमीडिएटली ये अपना इलेक्ट्रॉन शेयर कर रहे हैं, दोनों अपना अपना इलेक्ट्रॉन शेयर कर रहे हैं, इसका ही इलेक्ट्रॉन ये शेयर कर रहे हैं. That means what is happening over here, this particular, okay, sharing is taking place. Actually this electron belongs to him only, this atom only. Yes or no? Okay, but now this is positively, now whole unit has become positive. So this is nothing but ionic, this is sharing हो रहा है यार. ये देखो, इसके पास एक इलेक्ट्रॉन, इसके पास, तो ये कोवलेंट काइंड ऑफ स्टेप है. And this is what ionic. एक ने दिया, दूसरे ने लिया. Means एक ने अपना पोर्ट दिया और दूसरे ने पोर्ट उसका लिया. Basically that is it. ठीक है? हाँ, फिर शेयर. फिर वो आदमी बार बार बोलता है, अरे ये जो सूट पहना है ना इसने, वो मेरा है. You know, the idea is very much important over here. ठीक है? तो उन्होंने जब becomes embarrassing for the gentleman, वो नेक्स्ट टाइम बोलना उसने शुरू, अरे जो मैं सूट पहना हूं इन्होंने दिए. Then the other person is getting embarrassed. क्यों बोलने की जरूरत है? Because when he says that everything is okay, but when I say that yes, I'm wearing, you know, for example, the suit from my friend, I borrowed from him, so I don't mind that because I did not come prepared for that occasion, no problem at all. But bonding has taken. अगर उसने बोल दिया तो bonding break हो जाएगी. इसीलिए अगर जिसने दिया और उसने बोला नहीं कि नहीं ये मेरा सूट पहना है, ठीक है, तो definitely for example bonding will persist for a longer time. So this is ionic bond, this is covalent. पहले में एक losing हुआ, दूसरे ने gain किया, दूसरे में second step में sharing हुआ. वही जो सूट दिया था उन्होंने शेयर किया. शेयर ने जो भी ओकेजन में जाना था चला गया, पहन के एक्सेप्ट कर लिया और उस टाइम शेयर किया उन्होंने. या फिर उसका फॉर एग्जांपल उसने भी अपना फॉर एग्जांपल मटेरियल जो भी रहेगा उसने शेयर किया हो, but basically it belongs to now this hydrogen atom is sharing its electron with this electron was belonging to him only or this atom, so this is something which is sharing because दोनों equalize हो गए ना अभी, ये देखिए दोनों के पास equal हो गया, so there is a sharing of for example I mean if not clothes at least जो eating material उसमें sharing हो सकता है, जो कोई भी चीज है for example यहाँ पे second part is covalent, first part is ionic, that's why it is said to be the first sentence ionic and covalent. चलो read out Arpita that sentence again.
A coordinate bond has properties of both covalent and ionic bonds, therefore it is also called covalent bond.
Huh? Coordinate covalent bond ना?
Coionic bond.
Co-ionic, yes. Co-ionic, covalent and ionic. Very good. Second point पढ़िए. नीचे उसके नीचे जो हाँ conditions.
Conditions for the formation of a coordinate bond. One of the two atoms must have at least one lone pair of electrons.
Yes, one should have lone pair, other should be deficient of lone pair. Come on.
Example, ammonia and water. Another atom should be short of at least a lone pair of electrons. Example, hydrogen atom.
Yes, hydrogen atom. Next point. नीचे देखिए उसमें.
The formation of ammonia.
हाँ पढ़िए.
The nitrogen atom in an ammonia molecule contains a lone pair of electrons. It is a donor. The hydrogen ion accepts a lone pair of electrons and forms a coordinate bond.
It's an acceptor, isn't it?
Once this bond is formed, all four
species क्या लिखा है? All four NH bonds. All four NH bonds.
All four NH bonds become identical. The
Yes.
forms an ammonium ion having a single positive charge.
Next. Formation of
Hydronium ion.
नीचे देखिए सेकंड के लिए. ऐश्वर्या पढ़िए आगे.
Formation of hydronium ion and lone pair receptor hydrogen oxygen atom. Formation of
वो क्या? Next one और एक से. वो क्या है?
oxidation.
Oxidation reduction reaction. What is oxidation reduction reaction? Yet, have you done that earlier topic में oxidation reduction किया है ना? इसमें last year. Okay. Oxidation reduction reaction क्या है? एक glass 2 मिनट लगेगा इसको. Simple है, तुमने last year भी किया है. What is oxidation reduction reaction? Redox, yes. Redox reaction. Now oxidation means what? Addition of oxygen or removal of hydrogen. Reduction क्या है?
Addition of hydrogen.
Removal of oxygen or addition of वही format में लिखिए. ठीक है? Now for example here, children listen to it carefully. Copper oxide when you react with hydrogen, very simple, becomes copper plus अब इसका स्टेट देखिए. Copper oxide becomes what? Copper. That means it has lost oxygen. Yes or no? That means अगर oxygen lose किया, that means it is reduction. That means this element, this substance is undergoing reduction. ये hydrogen has become water. That means इसमें addition oxygen add हो गया. That means hydrogen is oxidized or undergoes oxidation. Very simple.
Yes, sir.
Right? अयान क्या हो गया? सुनो इधर देखो. Children, now tell me this one. Very simple. So rusting process, rancidity of code, corrosion, they are all coming in redox reaction. ठीक है? now यहाँ पे देखिए zinc oxide becomes zinc. That means zinc oxide is reduced. Carbon is getting oxidized to what? Carbon dioxide. Now the substance which is actually undergoing reduction, okay, is supplier of oxygen. उसको क्या बोलते हैं? Oxidizing agent. जो substance oxygen supply करेगा, उसको oxidizing agent बोले. Amazon is a big supplier of materials. तो जो supplier है, वो उसको बोलते हैं oxidizer. जो supply करता है oxygen और जो उसको oxygen लेता है, Like we are customers, we are buying those Amazon materials. Okay? So we are nothing but what? Reducing it. What is happening in our case? Why we are called as reducing agent? It is because in our case they are reducing our money aspect and they are piling up the money in a way. Okay, so that means we have there are two types of people in this world: buyers and sellers. Amazon is a seller, we are buyers. So we have to become sellers. Because sellers only are going to gain. Buyers will never gain, as a matter of fact. So that is the idea. Okay? A last reaction, redox reaction. Come on, tell me which is undergoing oxidation, which is undergoing reduction. कौन बताएगा? Very simple. आंसर बताओगे कि नहीं? आंसर दोगे कि नहीं? MnO2 is becoming MnCl2, losing oxygen. That means what? Oxidizing agent. That means undergoing reduction. Oxidizing agent हमेशा reduction show करेंगे. Reducing agent हमेशा oxidize होंगे. The reduction. What about HCl? HCl chlorine में बदल रहा है. यानी hydrogen remove हो गया इधर. That means oxidation. Yes or no? That means it is reducing agent. HCl is a reducing of the hydrogen supply. Oxygen supply, oxidizing. Hydrogen supply, reducing agent. Hydrogen या metal. तुम hydrogen की जगह पे metal लिख सकते हो. Oxygen की जगह पे non-metal लिख. Because oxygen non-metal को represent करता है. Hydrogen metal को represent करता है. Remember. ठीक है? So if there are two prefects in our class, one girl and another boy, so they represent the whole class basically. ठीक है? तो लिखा लिख लीजिए. That's the end. Now we are going for the periodic table का question. अरे question वाइज देंगे तभी तुमको आइडिया मिलेगा ना. I am not asking any question. मैं खुद ही question ये बताऊंगा. ठीक है? वो question आपको देखना है.
Last reaction, Cl will be the oxidizing agent. Cl will be the oxidizing agent.
Oxidizing agent MnO2 and HCl is reducing agent. Cl नहीं, HCl. Okay. लेकिन वो substance जो change हो रहा है उसको हम लोग reducing agent. ये इधर reducing agent और oxidizing agent इधर ही मिलेगा. Redox में नहीं. Redox में तो change हो गया ना. आपको इधर देखो. तो ये oxidizing agent है तो ये reducing है. अगर ये ऑक्सीडाइजिंग है तो ये रेड्यूसिंग है. चलो कम ऑन आयान. पहला क्वेश्चन बोर्ड क्वेश्चन तो हमने कर लिया. ओके, आप कुछ नहीं करना है. ओपन कीजिए बुक और उसमें ड्यूटी वॉल्यूम का क्वेश्चन आपको याद रखना है. मल्टीपल चॉइस क्वेश्चन में देंगे. विशाल मल्टीपल चॉइस क्वेश्चन. बेस नंबर बहुत ही अच्छा क्वेश्चन पूछा. प्राइवेट कीजिए क्वेश्चन. लास्ट बेंच वेरी गुड. मल्टीपल चॉइस क्वेश्चन नहीं है. पीछे होगा ना. पेज नंबर 9. विशाल इज स्टडीइंग वेरी मच हार्ड. Anyway, now हम लोग ने ये क्वेश्चन देंगे 2014 में 14 और 13 ओके ब्रश अप चलो. What is the revision of periodic table? We will see. Periodic table में सबसे important चीज क्या है? Our modern periodic table is based on what?
Atomic number.
Atomic number. Fill in the blanks. Point number 1. Point number 2. Atomic number से क्या लिख सकते हैं? Electronic configuration. Okay. Electronic configuration से हम लोग position of an element को find out करते हैं. ठीक है? So that means the number of shells are decided by the number of the period number is actually related with what? The period number is related with the shells. Okay. Whereas the number of valence electrons, okay, valence electrons is group number. ठीक है? तब मैं ये अपना क्वेश्चन आ सकता है तुम्हारा ये मल्टीपल चॉइस या फिल इन द ब्लैंक्स क्वेश्चन में आ सकता है, बैंक द कॉलम में आ सकता है. ठीक है? थर्ड पॉइंट, सोडियम का इलेक्ट्रॉनिक कॉन्फ़िगरेशन अगर सपोज अगर एटॉमिक नंबर दिया है तो they can ask you that this element X is having an electronic configuration of atomic number of X. What will be the electronic configuration of this element? तो आपको क्या करना पड़ेगा? 281. अब आपको याद रखना है कि 281 1 to 20 का एटॉमिक नंबर का एलिमेंट याद रखना है. तो आपको सबको मालूम है सोडियम, ठीक है ना? ओके, वैसे आपको मेंटली अपना माइंड में रखना है, लिखने की जरूरत नहीं है सोडियम. ठीक है? नाउ हाउ मेनी वैलेंस इलेक्ट्रॉन्स आर देयर? 1. तो उसका वैलेंसी क्या हो जाएगा? ये क्वेश्चन आ सकता है बेस्ड ऑन इलेक्ट्रॉनिक कॉन्फ़िगरेशन. सेकंड, वैलेंसी 1 है, तो इन व्हिच ग्रुप नंबर इट इज going to be? 1. Or which shell or which period it will come? Period. Right? But this element will form ionic bond or covalent bond. ये metal है, ठीक है ना? Because इसके पास एक ही है, so it will form ionic bond. But with the element, there is a Y दिया है तुम्हारे को. कोई एलिमेंट देंगे तभी वो ionic bond, because of course ये सब ionic bond ही form करेंगे. लेकिन with an element Y which is having an electronic configuration 287 with let us say 17, just an example. Now this element, okay, is having valency of what? 1 group. Now 17th group element, 17 period 3, yes or no?
Yes, sir.
Then also with Y it will form whatever bond, ionic bond. Okay? Now what will be the formula for this compound when they actually what combine? इसका भी valency 1 है, इसका भी valency 1 है. तो अगर इसका valency, you know, for example, and this will form what? Cation or anion. So this will form cation, positive. Okay? This will form anion. तो कौन सा कैटायन बनेगा, कौन सा एनायन, वो भी क्वेश्चन आ सकता है. ठीक है? इसका वैलेंसी 1 है, इसका वैलेंसी 1 है, दोनों का वैलेंसी जब 1 है डायरेक्टली व्हाट विल बी द मॉलिक्यूलर फार्मूला फॉर दिस कंपाउंड? ऐसा तो इमीडिएटली जब वैलेंसी सेम हो गया डायरेक्टली आप लिख सकते हैं XY इज द कंपाउंड. है ना? ऐसा 4-5 क्वेश्चन आपको याद रखना. ठीक है ना? बट इलेक्ट्रॉनिक कॉन्फ़िगरेशन के बेसिस पे तो एक क्वेश्चन श्योर है, इलेक्ट्रॉनिक कॉन्फ़िगरेशन मोर लेस. ठीक है? बिकॉज़ ये मेन पार्ट है. देन they can ask you for example, okay, what is electron affinity? अच्छा, एक important this 2013 में सब ऐसा ये जो क्वेश्चन है ना, यहाँ पे आपको ऐसा दिया है, ठीक है? यहाँ पे x है, यहाँ पे y है, यहाँ पे t है, ऐसा करके जो है ना, ये सब क्वेश्चन करते जाए. इसमें एक दो क्वेश्चन श्योर रहेगा. ठीक है? क्योंकि ये फॉर्मेट में क्वेश्चन आते हैं, ये बोर्ड एग्जाम में, so you have to go for this type of questions. It will be easier for you. Now, suppose for example, अगर metal side में कोई पूछेगा sir, which is having the lowest ionization potential outer X, Y, and T, which is having smallest atomic size, so X. Between this Y, Z, let us say Z, and let us say यहाँ पे A है, which is having the smallest atomic size in this. Just ये यहाँ पे तो बिल्कुल ये नहीं रहेगा, हाँ ये आएगा. ठीक है ना? Okay. Here, which are these elements which are actually what present as what? For example, C, D, A, B, जो भी है, ठीक है? यहाँ पे they are called as. Now, what is the valency of these elements? 0. ऐसा करके पूरा periodic table में you have to dissect those elements basically. तो ये सब चीज है ना बाहर करके ने आपको ये सब.
20 elements.
बस 20 elements. Okay, लेकिन उन लोग इधर वाले जो इधर अच्छा एक important चीज याद रखना है. 20 तक याद नहीं रखना है. इसमें कैसे याद रखना है? आपको अल्काइल मेटल्स पूरा याद रखना है. लिथियम, सोडियम, पोटेशियम, रुबिडियम, सीशियम, फ्रांसियम. फ्रांसियम तो रेडियो एक्टिव इसको नहीं देते आजकल, बट सीशियम. अल्काइल अर्थ मेटल्स में बेरिलियम, ओके, मैग्नीशियम, कैल्शियम, स्ट्रोंशियम, बेरियम, रेडियम. I mean I am writing all of them. अब इसमें क्वेश्चन आ गया, which element is having the largest atomic size? Among these, you know, two groups, which is having the largest atomic size? ठीक है? अच्छा अभी लाइक फॉर एग्जांपल अगर सपोज एक क्वेश्चन इसमें आया था this element A is on the left hand side with respect to B. Now tell me which is having a larger atomic size, A or B? A. Which is having, for example, more metallic character? Which is having less metallic character? Among these two, B. वो भी ऐसा क्वेश्चन है, ठीक है? और इसमें यहाँ पे हेलोजेंस का क्या रखना है? What are the halogens? Fluorine, chlorine, bromine, iodine, astatine, ठीक है ना? और टेनेसिन तो अभी नया है उसको नहीं, ठीक है? तो ऐसे करके क्वेश्चन आ सकता है। हेलोजेंस, the halogens में what happens? Electronegativity decreases or increases? I mean in all the cases the halogens are not the trends. Trends के क्वेश्चन के साथ है। For example, as we move from left hand side to right hand side, in the second period, atomic size decrease होता है कि increase होता है? Yes, atomic size decrease हो रहा है। What about electronegativity? Increases. What about ionization potential? Increases. What about metallic character? Decreases. What about non-metallic character? Increases. What about electron affinity? यानी अभी इसमें कौन सा तुम्हारा इन्वर्ट हो रहा है? बेसिकली जो एटॉमिक साइज के साथ चलता है इधर ही लिख देगा मेटालिक कैरेक्टर, और सब ऑपोजिट चल रहे हैं, ठीक है ना? तो आपको याद रखना है खाली एक मेटालिक कैरेक्टर डायरेक्टली प्रोपोर्शनल है और सब इनवर्सली प्रोपोर्शनल, तो ये याद रखना है। और ग्रुप में सेम चीज ये ना उल्टा हो जाएगा, इसका इलेक्ट्रोनेगेटिविटी इंक्रीजेस तो ग्रुप में डिक्रीजेस, यहाँ पे डिक्रीजेस तो ग्रुप में एट द बॉटम नॉन मेटल्स विल बी फॉर्म मोर, दिस इज अ ट्रेंड, ओके? नाउ एटॉमिक साइज किसके ऊपर डिपेंड करेगा? टू इंपॉर्टेंट फैक्टर्स, न्यूक्लियर चार्ज एंड नंबर ऑफ शेल्स, and all other properties are dependent on atomic size, बस mainly nuclear charge and atomic, बस वही दो फैक्टर, ठीक है? तो इसमें बेसिकली ऐसे सब क्वेश्चन आएंगे। ट्रेंड्स में आपको खाली ये याद रखना है, और जो लास्ट में समरी बनाया ना उसको ही आपको देखना है। हाँ टेबल जो दिया ना वो वाला, वो क्वेश्चन नॉर्मली दे आस्क, ठीक है? वो एनी अदर क्वेश्चन ना, एक क्वेश्चन ले लेते हैं जिससे कि तुम्हारे को एटलीस्ट जो ब्रश अप किया है whether you are able to do it. 2013 ले लो, 2013 all of you try, let us see, ये 2 मिनट बचा है, ये काइंड ऑफ एप्लीकेशन आ जाएगी, ठीक है? चलो और लास्ट कीर्तना पढ़िए पहला क्वेश्चन। अच्छा प्लान आउट पेज नंबर, बहुत ही अच्छा क्वेश्चन सर, समरी कहां पे सर? कौन सा? समरी कहां पे? 11 12 पेज नंबर 12 2013 आया समरी कहां पे? समरी लास्ट पेज में दिया ना, इंक्रीजेस डिक्रीजेस टैबुलर कॉलम, टेक्स्ट बुक नहीं, टेक्स्ट बुक में शायद है, यस सर क्या बोल रहे हैं? और यूपी वॉल्यूम में अपना इंक्रीज डिक्रीज दिया ना। चलो आयान रीड 2013 वाला फर्स्ट क्वेश्चन क्या है?
This table it does not represent hydrogen. Some elements are given in their own symbol.
हाँ अभी सोचो उसके बारे में, 2 मिनट दे लो, all of you just try to let us see whether which answer you getting. कौन सा कंपैरिजन? वो बताए सॉल्युबिलिटी, कोवेलेंट कंपाउंड्स, ये वो बॉन्डिंग का क्वेश्चन है, इसमें नहीं है। कोवेलेंट कंपाउंड्स आर सॉल्युबल इनसॉल्युबल इन वाटर, वो एक्चुअली वो चैप्टर में इसमें देते हैं, इट बिलोंग्स टू सेकंड ये चैप्टर केमिकल बॉन्ड्स, ठीक है सर? बोलो जे फर्स्ट आंसर क्वेश्चन क्या है? हाँ मोस्ट इलेक्ट्रोनेगेटिव एलिमेंट कौन सा है? इसमें आपको भी लग रहा है राइट हैंड साइड में क्या वो एलिमेंट? हाँ राइट हैंड साइड एलिमेंट इज मोर इलेक्ट्रोनेगेटिव एंड द टॉप हाँ फ्लोरीन। चलो नेक्स्ट वन, चलो बोलो नेक्स्ट क्वेश्चन। बुक नहीं है आपके, आ जाओ डॉक्टर मैडम के पास, मुझे प्लीज बाहर बैठ के तुमको अभी रिवाइज करना, लास्ट टाइम भी तुम्हारे पास नहीं था, अभी आज उनको रिपोर्ट करना विदाउट बुक नहीं है, सैटरडे संडे सब एक ही है आज अभी तुम्हारे को लेक्चर की बात करना पड़ेगा क्योंकि तुम्हारा मार्क्स परफॉर्मेंस डिपेंड्स अपॉन बेसिक टेक्स्ट बुक मटेरियल तुम्हारे पास नहीं होगा तो प्रॉब्लम है चलो अर्पिता सेकंड क्वेश्चन क्या है
Identify the most reactive element of group
और ग्रुप 1 बॉटम में ग्रुप 1 में सबसे बॉटम में कौन सा है आर मोस्ट रिएक्टिव एलिमेंट बिकॉज़ मोस्ट मेटालिक एलिमेंट नेक्स्ट वन बोलो थर्ड क्वेश्चन पढ़िए
Identify the element from period with least atomic size.
डिग्री बोलिए वो तो क्वेश्चन आपके लिए चाइल्ड्स प्ले है बोलिए क्या लग रहा है which element it is आया हाँ D
Group 1 में
period 3 में बोल रहे हैं जो सबसे smallest atomic size वाले कौन सा आंसर है M what is saying M एकदम राइट हैंड साइड यस गुड तुम लोग को आता है लेकिन तुम लोग बोल रहे नहीं आंसर बराबर ठीक है तो लास्ट क्वेश्चन विल टेक अप Prisha I think so you are laughing so I think you must be knowing the answer बोलिए Prisha knows the answer I'm sure Q कौन सा ग्रुप में पहले वो देख लीजिए पांचवा तो 5 इलेक्ट्रॉन 15 ग्रुप में 5 इलेक्ट्रॉन 16 ग्रुप में 6 इलेक्ट्रॉन्स ओके 17 में 7 जो ग्रुप नंबर है उसमें 10 भी ऐड होगा तो 7 इलेक्ट्रॉन्स ओके सो विल स्टॉप ओवर हियर एंड ऑल द बेस्ट ठीक है और ऐसे करते क्वेश्चंस एंड कीप दैट पीरियोडिक टेबल इन फ्रंट ऑफ यू व्हेन टुडे ठीक है थैंक यू सो मच और करते जो आराम से आ जाएगा अभी तुमको डर लग रहा है फर्स्ट एग्जाम है इससे हियर विल गो
Why there's a lot of crowd that side and the first bench is crying over here? Why like that? Get up. You. If he sits there, his brain is going to get blocked. So he has to sit here. Yeah, I am territory here. Never mind, sit at the side. The girls are serious now, at least. मैं ये नहीं बोल रही कि गर्ल्स के आसपास रहो, but हाँ रहना भी नहीं है. That's a very good thing. Men should speak like this. Very good. Now. The chapter that we are done is endocrine glands. Study the chapter. Achha, when is your test starting here? Tomorrow. On this chapter, no? On this chapter. All the best. Do well. All of you should get full marks. Okay. Keep studying. Huh? Expect नहीं करना है. You should be confident. मिलेगा. Now we are going to start. Not genetics.
Ma'am, you can start anything. I finished my.
Excretory not now. Excretory why all of a sudden? So what are the chapters you all are done? So excretory circulatory पढ़ाए बिना I will not teach you excretory. When I teach you excretory, there are so many references of the circulatory. इसीलिए तो मैं उसको सामने बुलाया. अरे याद रखो. मुझे याद है. Of course. Now, no. What I said is, abhi by the time your school gets, reopened and they start new chapter, obviously we'll be finishing because now we'll be having 3 hours lecture. No, 3 hours में एक chapter 2 दिन में खत्म हो जाएगा. But on his side, none of these chapters have been done.
Yes, ma'am. Justice to Aya.
I know.
Hashtag justice to Aya. Yes, we give exam.
अच्छा तुम लोग कितने दिन से स्कूल में मिल नहीं रहे हो? 7-8 दिन. The way you are talking, जैसे बहुत कुछ डिस्कस करना है. What is this? 10th standard के हो ना? क्लास के बाद आधा घंटा रुक के बात खत्म करके जाओ कल में. But now please don't talk. Now what I'm saying is, we had finished with endocrine glands as per your demand. Abhi before your school reopens, I think another 3 weeks तो छुट्टी है ही ना? तो 3 weeks. हाँ तो in 4 weeks time we'll of course finish circulatory system and we should be able to do it.
We don't have circulatory.
These are the two candles.
Even if you compare it with not circulatory system is not there for your exam.
For now it's not. For the upcoming exam,
It is there, but when we will teach here, I will always teach circulatory first. उसके बाद तुमको excretory. It will be easier for you to study. So that hardly makes a difference of 3-4 lectures, but you study at a go. Abhi before your school exam, you are following with both the chapters. Abhi in your school test finished with endocrine, we are done. Circulate, excretory, that we are done. Don't worry. Now, what I'm saying is, we have been asking for these 2 chapters, though his exam is over, but now let us go to the first unit and finish 2 chapters. Then again, you come to absorption, transpiration, photosynthesis at one go. So all these chapters you should do together. The continuity is there. So it's something like I teach one topic, again when I teach other topic, again I have to take the references. So it takes a lot of time because in your school they will teach one particular topic in full 1 month. Here we can't do that. We have to finish the topic in hardly 4 to 5 lectures. But again covering everything. So we'll repeat तरीके से नहीं हो सकता. Understood? At any time I cannot revise those topics with you. So that's the reason. What I feel is, ये excretory system, circulatory. Let us take it after these 2 chapters, and then we can again go to the third set of chapters, that is absorption, transpiration, photosynthesis. So a continuity is maintained, so that will be better. Same the first chapter we are not doing. Same the structural and functional unit, that first chapter we are not doing. Okay, na? You will not get questions also on that chapter. That's just.
Why won't we get?
No, in your board exams you will not get questions on that. That's just for self-study.
So why study?
You don't have to study. That's why we are not teaching you.
but in our semester we have it.
This chapter, they are keeping it.
Ma'am, teacher has said that every test there is of biology there will be cell. Like we have to do it.
I will recheck the syllabus again. This chapter is never asked in the paper. For years, we have never been teaching this in the class. We don't teach this in the class. It's fake questions nahi aata hai. Okay, still then, I will recheck and if you will need, I may show you that. Is पे questions nahi aata hai, and you may check also one of the previous year ka papers, see if any question has come from this chapter. So what I want to start, these two chapters, let us do it together. Structure function of the chromosome, cell division, and then followed by genetics. Okay, genetics, if you want to do it at one go, you do it, or let your school start that time also we can do it. That you tell me later. So since that chapter is over with you, North Point also that chapter is over, no? Structure of chromosome.
We have only completed 2 chapters, that is endocrine and then excretory.
Okay. Once you understand the concept of circulation, the names of the vessels, arteries, and all these concepts, जब तुम समझ जाओगे, the process of excretion, it will be easier for you to understand. I teach you excretory system and then I can go back to circulatory system. It will be double work for me. So now, as for the other school requirements, we will start with this chapter, that is structure of chromosome, cell division. Structure of chromosome, cell division. The first part of the chapter, structure of chromosome. Let me take out that chapter, turn to those. The first part of the chapter, structure of chromosome, that is what we have to learn. Now, when we talk about the concepts of chromosome, let us start with that concept. Chromosome, structure of chromosome. A single cell, all of you, nobody will talk. In the 9th standard, little bit about chromosome you have studied, no? Some idea about chromosomes you have. A single cell, the nuclear region. Now, in this nuclear region, if I draw these type of structures, what are they called? Chromatin fibers. Very good. These are called chromatin. These are called chromatin fibers, chromatin threads. Now, whenever we are talking about the concept of chromosome, chromatin, first of all, that you have to understand. What is the difference between the two words, chromatin, chromosome? So I said that these are long, thin, thread-like structures. Are they coiled? No, they are not coiled. They are not coiled. Now, this long, thin, thread-like structure undergo various degree of coiling and condensation. So this will now undergo extensive coiling. And the same structure, we draw something this way. Now, what do we call this as? We talk about these structures as the chromosome. This is a chromosome. What are the parts? The parts are this condensed structure. This is known as the chromatid. This structure, which is even more condensed. Can you see the board? This central structure, which is even more condensed, this is what is known as centromere or the kinetochore. Centromere or the kinetochore. Remember the terms. Huh? In this chapter, there are many terms that you have to study. You have to remember. So what I said now, what is the difference between the terms chromatin fiber and chromosome? Chromatin threads, chromatin fibers are thin, long strands, uncoiled. Chromosome rather is condensed. Now let us try to understand that what type of cell will have this and what type of cell will have this. Chromatin, if I am talking about, say suppose I talk about a cell which is undergoing the process of division, and I am drawing that this is the cell which is having the chromosome. Okay. This cell goes through various stages of division and results in the formation of 2 cells. Now this chromosome again undergoes uncoiling and results in the formation of fine long thread-like structures this way. So what am I to conclude? I'm saying that this is a mature cell. Somebody is talking. This is a mature cell ready for division. Always now. A mature cell ready for division will always have condensed structures known as chromosome. But now that the cell has already divided and now young cell have formed. These are the young cells. We generally term them as daughter cells. Now these young cells will not have condensed form of the hereditary material, but they will have the uncoiled form, long, thin threads, chromatin. So the young cell will always have chromatin. What did we learn first of all? Difference between chromatin and chromosome. First difference, what will you write? Chromatin threads are extensively long, uncoiled. Chromosomes are short and condensed, extensively coiled. First difference. What is the second difference I told you? Chromosomes are always present in a mature cell. Chromatin present in a newly formed cell or a young cell. Young cell. So try to understand, try to remember. I will give you questions to write also so that you are fully prepared with what points to write for question. So the first concept that we try to understand is a difference between chromatin and chromosome. Remember the location, the difference. So now again I talk about this chromatin thread, and this chromatin thread undergoes the process of condensation. And now that they undergo the process of condensation, extensive condensation that they undergo, and the same thing I said, I am drawing it this way to talk about the concept of condensation, transformation of a chromatin to a chromosome. But now I try to find out what is the reason for coiling and what is the chemical composition inside a single chromatin thread. But if I ask you, this chromatin thread itself has got condensed to form the chromosome, no? The chromatin itself got condensed to form the chromosome. Should the composition be same or different? Same. Composition same. So I never spoke that there is a lot of chemical differences. Chemical changes nahi hai. This is just a physical change. Coiling khali ho raha hai. So now, when I talk about that, what is the difference in the structure, or what is the difference in the chemical composition? No, chemical composition, there is no difference, only that there is a physical change that I'm talking about. Now I want to magnify this particular region and try to find out what is this thread internally made up of. What is this thread internally made up of? It is made up of two helical strands put together, and these two helical strands put together. Should I draw up there? Up there. Now these two helical strands, what I'm saying is that the part of the chromatin, when I want to magnify this structure to see how it appears. It appears almost like this, two helical strands put together. Two helical strands put together and connected by certain bonds, and this is what we talk about as the DNA molecule. What are you supposed to remember? The DNA molecule, I'm sure all of you know the full form. Kya hai? Deoxy. I will write again. All of you take a note. Deoxyribonucleic acid. Deoxyribonucleic acid. Sometimes the one more question you get, the DNA molecule is acidic. The DNA molecule is basic. Pick the correct answer. Kya pick karoge? Acid from the name itself. No. Deoxyribonucleic acid. Deoxyribonucleic acid. Okay? DNA. Now, first of all, we try to understand what are some of the properties. What are some of the properties? Now, these two strands that I have drawn, these two strands we come across that agar one strand is growing in this direction, the other strand in the same orientation they would not grow, or if I draw this way, the orientation would not be the same. What do I understand? When these two strands are formed, when one strand is growing in this direction, the other strand is growing in this direction. What is the meaning? When I understand that this strand, when it was developing by the addition of units, one after the other, one after the other, one after the other, one after the other, is directioning. And this strand was growing by the addition of units, one after the other, one after the other, one after the other, in this direction, in this direction. What is this nature called? So we are talking about the characteristics of the DNA molecule. Characteristics. What are the characteristics? The first characteristic, the DNA molecule is anti-parallel. It is parallel, two strands are parallel, but anti-parallel. You have to remember this. What is the meaning of this? Anti-parallel nature ka matlab kya hai? The two strands, they are parallel, but when we talk about their formation, if one is growing upwards, the other will grow downwards. Anti-parallel, yeah. Next is, they remain connected by what type of bonds? Hydrogen bonds. They remain connected by hydrogen bonds. Which bonds I'm talking about? I'm talking about these bonds. Hydrogen bonds. Hydrogen bonds. Okay. Now, this is also known as the DNA duplex. DNA duplex. Q, because it is made up of two strands, 1 and 2. No, two strands are there. Two ribbon-like structures are there. Two thread-like structures are there, connected by bonds. So DNA duplex there. But now, as I said that yes, whether I talk about this or I talk about this, I come across that there are some repeating units connected by bonds. Now I'm interested to know what are these repeating units? What are these called? These repeating units are called nucleotides. These repeating units are called nucleotides. What are the components of nucleotides? Wait, pause for a second. Or अरे hydrogen bond nucleotide का कहाँ component होता है? When we are talking about what is a nucleotide that I'm trying to show you, nucleotide are the, what is the definition you should learn? What is the definition you should remember? What are nucleotides? Nucleotides are the repeating units, of the DNA molecule. If I have to give you an example, very simple example to make you understand. If I'm talking about proteins are made up of what? Amino acids. So if I have to talk about a protein chain made up of 200 amino acids put together by bonds. So what am I saying? What is the repeating unit of protein? Amino acids. Carbohydrate. Repeating unit is what? You have studied no transition. Carbohydrate the repeating units kya hai? You are in this planet Earth. Behave like a human. Why have you lost somewhere? The people. So, repeating unit of carbohydrate क्या है? कौन बोलेगा? किसको याद है? Right करोगे? Repeating unit of carbohydrate किसने बोला? Who said? Glucose. Very good. Repeating unit of carbohydrate is glucose. You have studied, no? In the process of digestion, carbohydrate gets converted to the simplest form known as glucose, and then they are absorbed by the villi, no? So repeating units of carbohydrate kya hai? Glucose hai, no? So that way, if I'm saying the repeating unit of the DNA molecule is what? The repeating unit of the DNA molecule is the nucleotides. That is the answer that you should always write. Define nucleotide, repeating unit of the DNA molecule. Now, when we are talking about this nucleotides, we try to understand that what are some of the components of the nucleotide. अब ये nucleotide के components क्या है? What are the components of the nucleotide? Components. The first component is nitrogenous bases. nitrogenous bases. The second component is the pentose sugar. And the third component is the phosphate group. The third component is the phosphate group. So, ये हो गया components of nucleotide. What we are learning? Nucleotides we had learned first. What is a nucleotide? Repeating units of the DNA molecule. Now we try to find out that what are these nucleotides made up of? If I'm saying that this is a nucleotide, if I'm saying that this is a nucleotide, what is this nucleotide made up of? What is this nucleotide made up of? What is this nucleotide made up of? Just now we studied that they are connected by bonds, but what are they made up of? Three components: nitrogenous bases, pentose sugar, phosphate group. अभी ये जो nitrogenous bases है, they fall in two categories. Nitrogenous bases, they fall in two categories. Double chain structures known as purines and single chain structures known as pyrimidines. Double chain structures, I am saying very slowly, yaad karo, samjho. Double chain structures, let me just show you how do they look like, double chain structure that I am talking about. If I talk about a double chain structure, something this way, and okay. So now when we are talking about the purine and the pyrimidine, that is the basic skeletal difference. I am talking about a molecule which has got single chain structure. If that is so, then we are talking about pyrimidines. And if I am talking about a structure which has got double chain, something like this, then we are talking about purines. Okay? So now, what are purines and pyrimidines? There are two categories of nitrogenous bases. Let us try to understand that what are those nitrogenous bases, then the two categories that I told you. What are the two categories, and under these two categories, what are the bases? First of all, I talk about the first one, adenine, and then guanine. So what is common in both of these? They will always have double chain structures. No, just now that I showed you, they will basically have a structure something this way, and the supporting carbon, nitrogen, hydrogen bonds. Pyrimidines. What did I tell you just now? Single ring structures. Which are those? Cytosine, and thymine. What is common? They will have a single ring structure like this. First of all, remember the nitrogenous bases. What are the types? Single ring, double ring, and based on that, we are talking about two categories: purines, pyrimidines. You are majorly to remember these four names: adenine, guanine, cytosine, thymine. Adenine, guanine belong to the same category, and cytosine and thymine belong to the other same category. So these two are, this way we can group the four bases. Bolo. Pyrimidines, pyrimidines. Then, when I talk about the pentose sugar, what do you understand about pentose sugar? Pentose sugar means a sugar molecule with 5 carbon. Pentose sugar. Phosphate group, we understand phosphate group, similar like that. Now, let us use all these to draw the structure of a nucleotide. What is it? A nucleotide should have, what are the 3 components now? A nucleotide should have nitrogenous base, any one it may have, okay? So what are the 4 nitrogenous bases you have learned? Adenine, guanine, cytosine, thymine. These are the 4 nitrogenous bases you have learned. Now I may say that this is one nucleotide, this is the second nucleotide, this is the 4th, this is the 5th. So now whenever I'm talking about any nucleotide, a single nucleotide may have any one of the 4 bases. constant, pentose and phosphate will be constant. Let us try drawing one. Whenever we are talking about the characteristics of a nucleotide, whenever we draw the characteristics of a nucleotide. So what are the parts now? We are talking about, first of all, a pentose sugar. We start with this. This is a pentose sugar. 5-carbon sugar it is. This is the first, second, third, fourth, and here is the 5th carbon. 5-carbon sugar. To the first carbon, always the nitrogenous bases remain attached. These nitrogenous bases, again, I repeat, can be adenine, can be guanine, can be cytosine, can be thymine, any of the 4 it can be. And to the 5th carbon, what will be attached? To the 5th carbon, the phosphate will be attached. So this is a complete structure of a nucleotide. This is the, there, straight. See the pattern. So this is a complete structure of a nucleotide. Now you tell me, in a nucleotide, what are the constant components or what are the constant components in the nucleotides of DNA? Constant is phosphate and pentose. Uska koi types nahi hai. It's all the same all over the DNA molecule. What is changing from time to time? Nitrogenous bases. Sometimes adenine, sometimes guanine, sometimes cytosine, sometimes thymine. Now we are going to learn one specific rule. Now we are going to learn one specific rule. Now, if I draw the DNA molecule again, I am talking about the DNA molecule again, where we try to see one special property. We try to see a special property of the DNA molecule. Now, here I said already that look into one particular region of the DNA molecule, and now I am talking about this repeating units. I am talking about this repeating units. Once you are aware about the 4 types of nitrogenous bases, so how many types of nucleotides are possible? 4 types. I just now mentioned. एक nucleotide में कोई भी एक तरह का base possible है. So if that is the case, if I talk about a nucleotide with adenine as the base, the opposite nucleotide will always have thymine as the base. That's a constant rule of the DNA molecule. If I talk about another pair of nucleotides and if I get to see that the nucleotide contains G as the base, what does G stand for? Guanine. Then what should be the opposite? Cytosine. Now, this is what is known as the base pairing. Base pairing. This has been given by a famous biologist known as Charlotte. Base pairing. What is base pairing? A will always base pair with T. G will always base pair with C. And what are these? This is the hydrogen bonds that I'm talking about. Hydrogen bonds. This I mentioned already earlier also, no? Because we generally draw the structure this way. What type of bond is this? This is the hydrogen bond. So what is the third property about the DNA molecule we learn? The specific base pairing rule. Is it possible in a DNA molecule that sometimes A pairs with C? Error. If it is, then it's an error. There are at times, it's an error at that time. So what is the base pairing rule? A always with T, G always with C. So what did we learn so far? We are talking about that what is the DNA made up of? The double-stranded DNA molecule is made up of repeating units known as nucleotide. And nucleotide has got these 3 components. But now, if I remove these components away, then this 2 components together form what is known as the nucleoside.
Now will you repeat?
I'll repeat. What is the difference? Nucleotide and nucleoside. अगर nucleotide है तो nitrogenous base, pentose sugar, phosphate group तीनों components होना जरूरी है. Nucleoside है तो phosphate नहीं रहेगा. A component or a unit of the DNA which has got only the nitrogenous base and the pentose sugar, that is a nucleoside. So how does it look like? See here, the pentose sugar and any of the nitrogenous base. So that is a nucleoside. You get in your paper difference between nucleoside and nucleotide. What will you write? Repeating unit consisting of phosphate sugar, pentose group, pentose sugar, phosphate group, and nitrogenous base, nucleotide. Unit which contains nitrogenous base and pentose sugar only and no phosphate, that's a nucleoside. Okay, so this is first of all the nucleotide, nucleoside. Now, when we try to find out, okay, first of all, I want all of you to draw this double helical structure. Let us draw this complete. I'll draw it. Proper diagram, you all have to draw. A proper diagram, DNA double helix. Draw the diagram of the double helical structure of DNA. All of it, 2 mark, you should, as it is, get in this diagram. Draw the diagram evenly. Now, most of you, when you draw the diagram, I'll just bring into your notice. Maybe you all are drawing much better. But still, I'll just bring into your notice. Most of the students generally have a habit of drawing like this. This is not the way of drawing, huh? This way you are not supposed to draw. You are supposed to draw it just like this. This is the correct structure that you should draw, like a ribbon it is. The DNA is just like a ribbon. So इस तरह से draw नहीं करना है. You are supposed to draw it this way. All of you, please take it. 2 broad S draw करना है. That's very simple. Parallelly. Then, what is the greatest artist of this planet? Pencil से draw करना है. Pen नहीं. All of it. Now, when you draw this type of a structure, you will write straight away here. So this way you can write it. That's up to your choice how you want to draw the diagram. You may write it inside also, any of the method that you can choose. You can write down here also. Inside also you can write this way or this way also you can write. Both are correct. Whichever you find comfortable. Easier, jo bhi tumhe lage, you can draw. And if you are drawing inside, you can also show 2 bonds for A and T and 3 bonds for G and C. That also you can show. 3 bonds for G and C, 2 bonds for A and T. All of you, complete the diagram. Yes. All of you are done with the diagram? चैप्टर स्कूल में हो गया, उसके बाद ये आउटकम है। हाँ? ये क्या है? लैडर बनाना भी नहीं आता। It should not look like a snake. It should rather look like a ladder. You, हमारे बैग में पेंसिल नाम का कोई सामान नहीं है। Pencils रखा करो whenever you are attending the science lectures. देखो जो मैं डायग्राम गर्ल्स है ड्रॉ दो देखो पूरा डायग्राम और तूने पेन से क्यों किया पूरा पेंसिल देखो ऐसा बना ठीक है Done. Now, two things we will learn. ऐसे होगा structure. This is how the structure should be. Now, we have put all of you done. You will be asked questions. Now, what am I trying to draw? It is a single base pair that I have drawn. What type of, What? Done.
Yes, ma'am.
1 month पहले ये चैप्टर में मुझे पता तुम्हारे स्कूल में डायग्राम ड्राइंग नहीं कराया क्या होगा अच्छा 1 मंथ में ये हालत है क्या होगा
एक साल में
क्या होगा 1 साल में तो बायोलॉजी कोई सब्जेक्ट एग्जिस्ट करता है वही भूल जाते हैं What are we trying to understand? Complete the diagrams. All of you will complete the diagrams. अगर complete नहीं हुआ है, please finish it off at home. Complete करो. समाज सेवा. समाज सेवा के लिए तुम आए हो क्लास में. अच्छा, help ले रहे हो. You are taking help.
ना बहुत आदत है.
Oh my God.
हम बहुत आदत है. हम इसमें डॉक्टर करके मेरा खुद का ठीक है
Try to do on your own. Samay, be self-sufficient. Now, कभी भी अपना ईगो किसी के सामने बेचना ही नहीं है. मैं आए नहीं यार. Practice 100 times at home, but don't ask help from anybody. And if you are his friend, never make him handicapped. Let him try, let him do. Now, here, what are we trying to learn? You get 2 questions in the paper. एक है, this entire DNA molecule, the DNA structure that we are talking about is almost like a ladder. It's almost like a ladder. Now, let us learn 2 things here. If I talk about the entire ladder-like structure, when I talk about the vertical arrangement, I talk about the vertical arrangement. So, what are these repeating units? These are nucleotides. 2 nucleotides are linked vertically. 2 nucleotides are linked vertically by a typical type of a bond. What is the name of this bond? The name of this bond is phosphodiester. Phosphodiester. Only the name of this bonds you'll have to remember. 2 nucleotides, I come here. 2 nucleotides are linked by which bonds? Horizontally? Hydrogen bond. So we are talking about 2 types of bond. एक हो गया vertical bond, एक हो गया horizontal. तो horizontal bond कौन सा है? Horizontal bond कौन सा है? 2 nucleotides are linked horizontally by which type of bond? Hydrogen. And 2 nucleotides are linked vertically by which type of bonds? Phosphodiester. Now one more feature try to understand from this structure. I'm talking about, you get a question in your paper that the entire DNA structure which appears like a ladder-like structure. The rungs of the ladder means the steps of the ladder. The rungs of the ladder are made by what type of structure? The rungs of the ladder are connected by what type of structure? Horizontally. See here, which two structures are making the horizontal link? The nitrogenous bases. This is a nitrogenous base and this is another nitrogenous base. Yes or no? So when we are talking about that in this DNA molecule, you get questions there. The rungs of the DNA molecule. The rungs of the DNA, these are the steps. The rungs of the DNA molecule are formed by which component? The rungs of the DNA molecule are formed by the nitrogenous bases. So that should be your answer. The rungs are formed. This is the ladder, no? The rungs are formed by the nitrogenous bases. Can we write the rungs are formed by the pentose sugar? No, they are far away. They are far away. Understanding, no? So now this is what is your answer. The rungs are formed by the nitrogenous bases. So this is about the DNA structure that you have to understand as per your syllabus. What are the components? Nucleoside, nucleotide means what? And how do they majorly look like? The basic composition and the writing of the structure. See that there's a diagram in your book. Figure 2.3. Take the diagram. Take the diagram. That is what I'm showing you here. Trying to show you one base pair. So this type of base pair may be many. It will one after the other together. No. So I have written here one pair A and T. Same I could have written for G and C. Another A and T. Another G and C. So you have got one diagram in your book. Take that. This type of diagram based questions you will get. Now next is after we are talking about the concept of structure of the DNA. One more feature we try to understand, that is duplication of the DNA molecule. Duplication of the DNA molecule. Now for that, we again go back to the concept of chromosomes. We again go back to the concept of chromosomes to understand that this chromatin thread which undergoes the process of coiling. Now why does it undergo coiling? Why does it undergo coiling? How does it undergo coiling? It is something like whenever we are talking about the chromatin thread, at different stages of cell growth, the chromatin fiber will behave differently. In this cytoplasm, what is the cytoplasm inside the nucleus known as? Nucleoplasm. Very good. Nucleoplasm. Now, the nucleoplasm has got specialized proteins in abundance. What are these proteins called? These proteins are called histone proteins. And I just now asked you, if I have to talk about a protein is repeatedly made up of some of the other amino acids. Now, if I have to talk about the histone proteins which are abundantly present in the nucleoplasm, they are majorly made up of positively charged amino acids. So you have already learned a basic concept of amino acids, but try to understand that amino acids can be negatively charged also, positively charged also. So here I'm saying the nucleoplasm, which is abundantly filled with histone proteins, are positively charged. The DNA is negatively charged. It's an acid. The DNA has got an overall negative charge. These molecules that I'm talking about now as histones have got a positive charge. Should they be having an affinity? Of course, no. They should be having an affinity, and that is the major reason why the DNA gets coiled. That is the major reason why the DNA gets coiled. So if I am trying to show this type of a protein, the histone protein, this histone protein is present in the nucleoplasm and the DNA starts coiling. The DNA starts coiling like this and the DNA starts coiling like this. So this is how we understand the process of coiling of the DNA. This histone and the DNA wrapped around. The histone and the DNA wrapped around. This entire unit, if I see, this entire unit, if I see, what is this called? This is called the nucleosome. What am I saying? I'll repeat. We are trying to understand why does the chromatin coil? I told you chromatin coils to get converted into chromosomes, but what is the reason? Why should they coil? They do coil because the chromatin is made up of DNA. Just now we were talking about the DNA. And we had learned that the DNA overall charge is acidic, negative. And since the DNA is a negatively charged molecule, also develops a affinity for these positively charged proteins, and they start coiling and coiling and coiling. The filament or the ribbon, that is the DNA, starts coiling around these proteins, resulting in the formation of a complex coiled molecule. So if I have to talk about a portion of the DNA wrapped around this histone proteins, what is this packing unit called? The packing unit is called the nucleosome. So such type of histone proteins are numerous in the nucleosome, and the DNA thread is constantly packing, constantly packing. So in this case, when I understand that this nucleosome is actually made up of certain units, if I say that this is the nucleosome and the DNA is wrapping around, the DNA is wrapping around, and then it will go to the next, and then it is wrapping around, and then it will go to the next. So there are some DNA regions which are free, and certain amount of the DNA wrapped around the nucleus. So this packing unit, which has got DNA and the histone together, what will be named as nucleosome. But if I check, is this a single protein unit? No, it has got 4 types of units, and that also in 2 doses. What do I mean? This entire structure, which is known as the core histone, it is known as the core histone. Let us learn about the core histone now, or the histone core. What is it? It is the main protein around which the DNA is wrapping around. But just now I said, this histone core is not a single protein or not a single subunit. Rather, it is made up of 4 subunits, but every subunit of 2 doses. What do I understand? What are the subunits? Subunits are H2A, H2B, H3, and H4. And I said each one of them, 2 times they are present, 2 times they are present. So total how many units are there? Total how many units are there? 8 units are there. That's the reason this histone molecule is known as a octamer. You get questions. Why is the histone protein known as an octamer? Because the histone protein around which the DNA wraps is actually made up of 8 units. But how many types of subunits? 4. Pick those words. All these words are important for you. Octamer. Why is it known as an octamer? Because the histone protein has got 4 subunits. Every subunit 2 times they are present. Every subunit 2 times they are present. Now the names are optional for you. The names are optional for you. The names you may not. Just check if the names are there. I don't think the names are there. Okay, the names are optional for you. The names you may not need, but I just showed you. You want to only understand why is the histone known as an octamer. Now, if I ask you that with reference to the DNA, can you tell me that a single region of the DNA, what will you term this single region of the DNA as? A single segment of the DNA. What is this known as? What is this known as? What is this known as? You've studied in the last year's syllabus. It was there. A little bit of it you have studied. A segment of the DNA. What do we call that segment of the DNA as? जो हमारे characters regulate करता है. What do you call genes? What is a gene? What is the definition that you should always remember now? Functional segment of DNA that regulates a character. Can you tell me 5 characters? Any 5 characters that we show? Biologically, what is the meaning of the character? Biologically, the meaning of character is not only our behavioral characters, remember. Example of our characters? The color of the eyes, the texture of the hair, the height. And on top of that are behavioral features. Normally, when we talk about the word character, we understand only our behavioral features. No, but no, under the concept of biology, the physical character, the physical features is also a character. So typically, you are asked the definition of gene quite often. Define a gene. And this again, elaborate in the next chapter when we talk about. But right now, we are only confined to that word gene. What is the word gene? How do you define? Functional segment of the DNA. Please write it down. Functional segment. That's a very common definition you get to see in your paper. Functional segment of DNA that regulates a character. Functional segment of DNA that regulates a character. Functional segment of DNA that regulates a character. Okay, take out your textbook now, page 19. Let me see if you're able to solve this. B, fatafat karo. B, only B, name the following only I want you to do. B, name the following only. B, name the following only karo. Fatafat. 2 minutes. Till here, any doubt, any confusion, anything that is not understood, please raise questions. अगर confusing है, समझ नहीं आ रहा है, don't remain with your doubts and confusion. Three questions only, no? 3 to 4 questions. Complete करो. हाँ, 4. Complete करो. Those of you haven't finished drawing the DNA diagram, you will complete it at home. पर पेन नहीं यूज करो. हाँ, कलर नहीं करना है, ओनली पेंसिल से ड्रॉ करना है, पेंसिल से लेबल करना है. Are the answers all of you? What is it? Which one? The type of bond which joins the complementary nitrogenous bases. I told you, no? Complementary, that word I haven't used. I taught you this, no? Just now I told you that A will always pair with T and G will always pair with C. This is what is known as the base pair rule I told you. On top of that, G and C, they are known as complementary bases. Complementary, meaning G is always supposed to bond with C. T is always supposed to bond with A or vice versa. These are called complementary. So what is the answer? The type of bond, hydrogen bond. Horizontal bond, I told you, hydrogen it is. Hydrogen bond. Hydrogen bond. Write one difference, write one question. Write 2 differences between nucleosome and nucleotides in your notebook. 2 differences between nucleosome, write on the chapter name. And the first question that you are writing in the notebook, 2 differences between nucleosome, nucleotide. 2 differences between nucleosome and nucleotide. Which one? D. The complex structure of DNA, strand, and a core of nucleosome. Just how I explained, you know, the DNA and histone, nucleosome. DNA wrapping around histone. Histone made up of 4 units. Every unit present in 2 doses, 2 times. I said, write 2 differences between nucleosome and nucleotide. Whatever. In your paper, basically you will get a difference with a condition in the bracket. Okay, now I am asking you to generally write 2 differences so that you know both the differences what I taught you right now. What is the first difference that you will write? Nucleotide. What is the first difference that you will write? Nucleotide is a repeating unit of the DNA molecule. Nucleosome is the packing unit of the DNA molecule. Packing unit. Matlab, packing unit matlab the reason for the coiling. So that is what you should write. Nucleosome is the packing unit of the DNA molecule. Nucleotide, repeating unit of DNA molecule. What can be the second difference that you should write? Now you can write based on its composition. Nucleotide made of components. You know now what are the three components, and nucleosome what are the components? DNA and histone. Because the second difference can be composition wise. First difference, you know, first difference you have written. I will be. First difference you are doing. First likha. Second difference likho based on the composition. Nucleotide is made of three components, that is nitrogenous bases, phosphate and pentose sugar. I repeat, nucleotide is made of three components: nitrogenous base, phosphate group, pentose sugar. And nucleosome now, the other side, made of histone protein and DNA. Made of histone protein and DNA. Histone core, what did I tell you? What is meant by histone core? Made of 8 subunits of 4 types. 4 types, total 8 subunits. ये याद रखना है. Now, next we are going to come across, once you are found with this concept of DNA, composition, structure, nucleosome, nucleotide, nucleoside. Okay, all these words we should write. Let us write one more difference. Write 2 differences between chromatin and chromosome. Very common this question. I have drawn the structure of the chromosome this way. All of you please take a note again. I have drawn this structure of the chromosome. Where I said, what is this I said? Chromatid. Chromatid. I repeat again. Always students are confused. This is chromatid. When I gave a name for this, I said this is centromere here. Now I am asking you to write difference between chromosome and chromatin. Chromosome and chromatin. Chromosome and chromatin. तो एक क्रोमोसोम में कितना क्रोमैटिड्स होता है? 2. One chromosome has got 2 arms, 2 chromatids. हाँ? नहीं इसको बाद में आते हैं. इसको सिस्टर क्रोमैटिड नहीं बोलते. चलो फटाफट लिखेंगे. Chromosome and chromatin. What is the first difference? What will you write? What is the first difference? Chromatin is very long tail, uncoiled. Extensively coiled, highly coiled. देखो इसमें कितना difference आ गया. Long tail, uncoiled. Uncoiled, that is for chromatin. Chromosome, highly coiled. Highly coiled and appears broader, shorter. Appears broader, shorter. तो ये हो गया first difference. Second difference क्या बोला था मैंने? Stage of the cell. That is the second difference that you should remember. You get difference between chromatin, chromosome, bracket में stage of the cell लिखना है. तो stage of the cell क्या लिखोगे? Students start writing down the stage, mitosis, not that stage of the cell. In which type of a cell you get? Chromatin you will always find in a young cell. हाँ, ये भी तुम्हारा गलती है. Chromatin you should not students always write. Chromatin is present in interphase, chromosome is present in prophase. So chromatin तो metaphase में भी है, anaphase में भी है, telophase में भी है. Not that. That's not the state of the cell that you are supposed to write. Chromatin लिखोगे present in a young cell या तो newly formed cell. Chromosome you will write present in a mature cell ready for division. Mature cell, that is the answer that you should write. Chromosome present in a mature cell ready for division. Next, when you study the stages, you will come across क्रोमोसोम सारे स्टेजेस ऑफ सेल डिवीजन में प्रेजेंट होता है एंड क्रोमैटिन जस्ट व्हेन अ सेल इज फॉर्म उसी पार्टिकुलर स्टेज में प्रेजेंट होता है. सो व्हाट आर यू राइटिंग? क्रोमैटिन प्रेजेंट इन अ यंग सेल or newly formed cell, chromosome always in a mature cell, ready for division. Now, after this, in the next class, we are going to study the stages of cell division. What are the stages of cell division? To introduce, what is the answer for name the following? I'll tell you. All of you can check the answer. All of you can check if the answer that you written is correct. The first one is nucleotides. The second one is nucleosome, right? Second one is nucleosome. Third क्या है? Hydrogen bond. And the three components you know already, right? Next class, all of you should have pencils. You have to draw stages of cell division. Very serious. All of you, good luck for your exam. Do very well.
Ma'am, I just wrote biology.
Which one of the gland?
भाई उसको ट्रोल किया नहीं किसी का बन रहा
ये कौन सा वन स्टेशन ना अंदर ये एक सेल के अंदर की वो भी इतना है ना उसकी वो एक्सपोर्ट हो रही है ना अंदर
हिस्ट्री पे क्यों पढ़ते हो तुम? और पुराना देख रहे वैसे फाइल
कितना बता ये
ऑब्वियसली
चलो मैंने ना कि फाइल
इसके बाद
नहीं बट अच्छा है और इतना अच्छा है नदी आगे का कोई ख्याल है तेरे को
पण अरे आमचं ते हे आहे ना त्याच्यामुळे हा काय बोलायचं ते आमच्याकडं इकडं वेगळं आहे जरा थांबा 2 मिनिटं 2च मिनिटं थांबा